personalized oncolytic immunotherapy. So what are those platforms and how we came up with it? So you should know that oncolytic viruses are viruses that have at least a dual mode of action. One mode of action is direct oncolysis. So basically a virus can infect the cells, can replicate within the cells, and can kill the cancer cells or any cells for what matters. But in addition to that, those oncolytic viruses, they also have the possibility to actually stimulate the immune system and generate some kind of anti-tumor immune response. So this is the second mode of action. Now, this is a clinical trial that we did very long time ago, where actually I'm showing this to you just to let you appreciate how massive is the T-cell influx, so the T-cell recruitment in the tumor after the viral administration. This is, for instance, in this example, is an ovarian cancer patient or a mesothelioma patient before the treatment, after the treatment, and in brown you see the staining of the T-cell. So actually when a virus is in the tumor or when a virus is in any tissue, there is a massive influx of T-cells that are going to they are migrating to the tissue. The problem with that, or the problem that I had with that, is that the majority of those T cells, as you can see, are going there because of the virus. So they want the antiviral T cell are going there to clear the viral infection. They are not really going there to clear the tumor infection. That was my problem, and I actually wanted to solve this by actually changing this balance from this situation to actually this situation. So I wanted to find a system where we could actually decrease the amount of antiviral T cell and increase the amount of anti-tumor T cell. So we came up with a system that it's called PeptiCrAD. PeptiCrAD is an oncolytic virus. In this example, is an oncolytic adenovirus, which is genetically engineered to be oncolytic and genetically engineered to express immunostimulatory molecules. In addition, and that's the novelty, is coated, is decorated with the tumor-specific antigens in the form of MHC class 1 restricted peptides, which are slightly modified to attach onto the surface of the virus by electrostatic interaction. So this interaction occurs at the bedside, and it occurs in a couple of minutes. So I put here 15 minutes, but to be honest, that is a couple of seconds. We have done it with, we have developed this technology for oncolytic adenoviruses. We call it PeptiCrAD. We have done it for enveloped viruses like vaccinia virus or herpes virus. We call it, we call it PeptiEnv. We have done it for some bacteria, like for instance BCG. We call it PeptiBac. We have done even something a little bit, I would say, less orthodox, in the sense that we have used directly membrane from cancer cells to build up artificial envelope around unenveloped viruses. We call it ExtraCrAD. And then, of course, we have translated this technology that were made and developed for cancer therapy, also for infectious disease. So we have developed several vaccines for infectious disease, including COVID, but not necessarily just COVID. I'll give you some example. This is the first one, and it's PeptiCrAD, done by Chris Capasso, one of actually the first PhD student in my lab. In this example, I show you how a naked oncolytic adenovirus or coated with Synfecal is actually treating B16 ova tumors. As I said, we attach these peptides by electrostatic interaction. In this case, adenovirus is a very negatively charged virus. Actually, the charge of the particle is minus 30 millivolt. So you can think of adeno like a small negative ball, and we can attach those small peptides that we just made it positive, and they attach quite nicely. So in this example, as you see, we have a tumor. This is the mock tumor is growing. The control virus is actually growing. Just in parentheses, this is a human adenovirus. It's not really giving any oncolysis in the murine tumor. But nevertheless, there is a little bit of adjuvant effect, so that it's working a little bit better than mock. Then when you mix the control virus with the OVA-specific peptides, Synfecal in this case, there is a slight improvement, but not significant, with the control virus. In this group, the control virus and the OVA peptides, in fact, they were just not pre-mixed, but they were mixed directly in the tumor. So there are basically 2 syringes injecting both. But when you actually take the same amount of this virus and the same amount of those peptides, but you premix it, so the only difference between this group and this group, that in this case they are not premixed, and in this case you premix, then basically you go from this situation to this situation. There is actually a very nice induction of a peptide-specific T-cell response, and those T-cell response is converted directly into a much nicer anti-tumor response. We have published several papers with the same technology in different tumor type, with or without checkpoint inhibitors, and also by combining different type of peptides restricted for MHC class 1 and 2. In particular, this technology, Peptigrad, is now in clinical trial, and this is basically a simple schematic of the clinical trial that is actually ongoing at the moment, but I'm not going to talk about it. In another example is PeptiEnv. So basically, we have translated the Peptigrad technology to enveloped viruses, basically making different anchors to attach peptides onto the envelope of viruses like vaccinia or herpes virus. Erkko Ilosmagi, postdoc in the lab, has designed this, published a couple of years ago. Here you can see in this example is vaccinia virus, this example is herpes virus. As you can see, the vaccinia virus, 14% of tumor control when it's alone, and when the same virus is coated with peptides, OVA peptides in this case again, then goes from 14 to 71, and herpes virus goes from 12% to 28%. So by simply mixing or attaching anti-tumor or tumor that a peptide that the tumor is presenting onto the surface of an oncolytic virus, we make the oncolytic virus work better by strong induction of peptide-specific T-cell response. As you can see also here, the OVA T-cell response, very nice induction with herpes, no with naked, and with vaccinia, again, not with naked. And of course, the antiviral response pretty much remains the same. We've done it also for BCG, again, Erkko in the lab, BCG, an anti-tuberculosis vaccine, but also used for bladder cancer. And it has, the mode of action is simply as an adjuvant, but of course, just by coating the BCG with tumor-specific peptides, basically, we could basically improve the efficacy. As you can see, this is MOC and checkpoint inhibitors alone. Of course, MOC there is more control of the tumor, checkpoint inhibitors in this model about 27%. Then there is the BCG naked, 30% tumor control, BCG plus checkpoint inhibitors, not a real improvement in this model, in this experiment that I'm showing you. But when you coat the BCG with tumor-specific peptides, you go from, in this case, TRP2, you go from 30% to 56% without checkpoint inhibitors, and 27% to 70% when there is a combination with checkpoint inhibitors. Again, by simply attaching the peptides onto the surface of the platform is working better. And of course, nice induction of and recruitment of CD8 positive T cell and TRP2 specific CD8 positive T cells. So again, the platform is inducing a peptide specific T cell response that is going to the tumor and is helping the virus. So again, dual precise mode of action. As I said also, in fourth example, Maglio Fuscello in the lab, what we have done, we have taken directly the membrane of the tumor, and we have built up artificial envelope around unenveloped viruses, like adenovirus. Here you can see how electron microscopy of adeno naked, membrane naked, alone, sorry, and this is when the adeno is within the membrane. Here this is just some example of a paper we published a couple of years ago, B16-OVA, very nice control of the tumor with a technology called extracrad in B16F10, which is a little bit more immunosuppressive than OVA. Again, nice control of the tumor. And in this experiment in lung carcinoma, we could actually see that the membrane, in order to maximize the efficacy, the membrane has to be from the same tumor to maximize the recruitment of the virus to the tumor and also the induction of the T-cell response. Hopefully, in this first part, in these first few slides, I convinced you that basically if you work on oncolytic viruses of any kind, or if you have a platform, you can actually just simply coat your platform with tumor-specific antigens, and that will basically become a much more precise and efficient way of treating tumor. But the big question here is, what are those peptides that we want to attach onto the surface of these viruses? So to basically tackle this problem, we have a second line of research in the lab, which is mainly or 100% focused on tumor antigen discovery. We mainly do it with a technique that is called the ligandome. In the ligandome, basically, you take all the MHC class I from a given tumor, and then you detach the peptides that from the groove of MHC class I, and by mass spectrometry, you basically analyze the sequence and the abundance of every single peptides that in any specific time is presented by the tumor in that moment. The technique is called the ligandome or immunopeptidomic, and that can be done with a classical approach, which takes a little bit of time, but most importantly, the classical approach requires a huge amount of cells. I'm talking in the order of billions of cells, which is doable in the lab, but when it becomes but it's very difficult to translate to clinic, because very rarely in the clinic you have tumors that are so big that you can actually, for yourself, of course, because there is the pathologist and a lot of people involved. So usually you are left with a very small amount of tumors. To overcome this problem, we have developed and patented our own technology, which is called PeptiChip, that is a microfluidic-based chip, very, very small. I have a couple of chips with me, if you want to see later, where basically they do exactly what the classical approach would do, but starting with a very, very small amount of material. So we went down from 1 billion cells requirement in the classical approach to 1 million, and sometimes with some collaborators, we even went down to 250,000 cells, I think. Right? Yes. So the PeptiChip has been developed mainly by Sara Feola, postdoc in the lab, and Jacopo Chiaro, PhD student. It's as I said, it's a microfluidic-based chip where there are millions of micropillar in the chip that are actually taking the MHC class I from the tumor, retaining it. There is a couple of wash, and then after the wash, there is a acidification of the solution that detach the peptides from the groove of MHC class I. The peptides, they go straight to the mass spectrometry for the analysis. This is just an example where we have compared the standard method, classical approach with PeptiChip. With the 10 million cells, you see that it's already losing the amount of peptides that you can retrieve, and with 1 million cells, there is almost nothing with the classical approach, while PeptiChip, you still retrieve the same amount of peptides, which are about anything between 2,000 to 10,000 peptides. We have published this on ACS now I think 2 years ago, so if you want to look more. But anyway, after the PeptiChip analysis, usually when you looked at the length of the peptides, you have this distribution. This distribution tells you that the peptides that you are retrieving are actually real MHC class I restricted peptides. In fact, there is an enrichment in 9 mers, which is exactly the perfect length of peptides that is presented on MHC class I. So anytime that you do either classical approach or PeptiChip approach, you need to have this distribution to make sure that you are working with real MHC class I restricted peptides. But nevertheless, you still have thousands of peptides that are actually coming out from this analysis, and you need to decide in a short time what are the peptides that are more relevant, let's say more immunogenic, to actually develop your biological drug. And we come up with, actually, Jacopo in the lab came up with a very nice software that is called HEX, which was published, again, a couple of years ago, and it's free software available for everybody. The software, in addition to do a lot of physical, chemical, characterization of the peptides, but also change the paradigm or push the boundaries of what immunogenicity means. And in fact, the software is based, the concept of immunogenicity on molecular mimicry between tumor-specific antigens and pathogen-specific antigens. So the software looks at how similar a tumor-specific antigen is with a pathogen-specific antigen and assign a very high score to a tumor antigen that looks like a pathogen antigens. The scope is actually to look at tumor antigens that can somehow engage pre-existing T-cell clone that we have developed during our entire life. And by using this software, we can actually accelerate significantly our choice of the peptides, which basically allow us to develop the cancer vaccine in a relatively short time. But of course, when you do it in mice, it's very, very simple because mice, they only have one HLA. But when you need to test this in human, it gets much more complicated because we have a completely different set of HLAs. In order to do that, we have developed our immunocomplex organoid pipeline, basically and Miku in the lab together with Mela and Gabri have mainly done that. We can use organoid not only to look at the direct killing of oncolytic viruses. For instance, here, from the same patient, RCC organoid, you can see that there is the real tumor that pathologists have actually identified. It's very, very sensitive to the virus. The virus is actually really tumor-specific, replicating and killing selectively the cancer cells. But the marginal part, what the pathology called the benign tumor, I don't know what that means, but they call it benign tumor, you can see that the virus is actually not killing the benign tumor, so that the part was a little bit more marginal when the tumor was resected. At the same time, we could also, so you can see this very clearly, and again, also here, you can see that when the tumor is mixed with oncolytic virus, the tumor is actually not growing or is basically being killed. When the same tumor is actually without virus, the tumor is just growing, but there is no effect on the benign part of this tumor. But most importantly, but of course, this is one side, the oncolytic part. But what is even more important for me and for my group is the immunological side of it. And in order to do that, we had to develop immunocomplex organoid. We do it in two ways. The first one is by utilizing a microfluidic-based chip, where basically some organoids are put, patient organoids are cultured in different parts of the chip, and the T cell from the same patient is basically located in different other parts of the chip, and we can look at the recruitment of T cell here, for instance, in green, to the organoid, and we can look how many cells are recruited and whether or not those cells are actually killing the tumor. We have also another system which is based on chicken embryo, where we can actually culture the human tumor organoid in the coralontoic membrane of the chicken. And this is a very cool system because during the chicken development, at some point, there will be the blood vessels are forming, and the blood vessels are actually going through all the organoids, and you can put in one egg as many organoids you want. Some of them are treated, some of them are not treated. And of course, you can look at the virus going to the organoid, but most importantly, you can actually, and I have a video later, where you can actually inject the PBMCs of a patient that have been eventually enriched for a specific peptides you are interested in, and then you can inject in the heart of the chicken that is start to pumping blood, and then you can actually see whether or not the human PBMCs are actually going to the human organoids, and whether or not the organoid has been treated. You can look whether you can look at recruitment and T cell recruitment and T cell killing. But I have a video later on that. So now, if those are the three parts that basically they are the three main line of research of my lab, but how do they work all together when you put for developing the real personalized oncolytic vaccine? I'll give you just two examples in these last 5 minutes. One example done by Sarah, published last year, where we have taken CT26 colon cancer. We have done the entire pipeline in 1 or 2 weeks, and then we have treated CT26 tumor. So here Sarah is done the ligandome analysis, screening with the hex of the peptides, formulating of the vaccine, and treating the mice. This tumor has been treated, this tumor has not been treated. Here in green, you have the pepticrad coated with our own set of peptides, and in black you have pepticrad coated with what was considered the golden standard antigens for that specific tumor. And as you can see, for the pepticrad treated coated with our own peptides is performing much better, significantly better than the pepticrad coated with a set of peptides that were supposed to be, the best that we knew before this experiment. And this was observed not only in the treated tumor, but also in the tumor that were not treated. So eventually, again, remarking eventually the fact that the T cells are actually migrating to non-treated tumor and actually helping. We have done pretty much, Jacopo and Gabriella in the lab have done pretty much the same exercise for human and murine mesothelioma. This is an example of murine mesothelioma, very nice distribution of the replicates, very nice enrichment on 9-mers. Gibbs clustering, basically look at what kind of shapes your peptides have, and you see that this is basically the peptides motif that we have found, which basically completely overlap with the reference of the HLA of the mice. Basically, again, it means that all these 9-mers are basically perfect 9-mers that were in MHC class 1. We have done the screening with our X software, and then we have looked at whether or not the selected peptides would give any immune response in mice. Of course, we have picked the best one. We have formulated pepticrad, this is surface plasma resonance, just to make sure that the peptides that we have selected can attach quite nicely on the surface of the virus. And then we have performed an experiment, or we have performed several experiments, but in this example, for instance, this is the experiment that was actually performed. And as you can see, in the mesothelioma example, or in this example of mesothelioma, murine mesothelioma, this tumor that was incurable in murine becomes very curable when we use the same virus coated with one of the peptides that we have actually selected throughout our pipeline, with very nice activation of a specific T-cell response in the group of mice. The recruitment, as you can see, is pretty much the same. So whether there is a virus coated or not, the virus always recruits cells. But when the virus is coated, it's actually recruited more specific T-cell, and those more specific T-cell are actually able to kill the tumor, as you can see also, because they are a little bit more exhausted. Done the same thing also with the human mesothelioma, direct from the 4-cell line, and a couple of patients from the clinic. Very nice, again, biodistribution, enrichment of 9-mers. The majority of the 9-mers that we have selected, as you can see, they are very good binders. So basically, again, showing that those are very nice and good peptides. We have expanded T-cell clone by either pulsing this is, in this example, this is just an healthy donor example, expanded with peptide pulsing, and we have also expanded with the tumor lysate just to select the best peptides. We have basically taking the best peptides and with these peptides, we have looked at tumor cell killing, and as you can see now, the peptigrad coated with set of peptides that we have selected was killing the target much better than our positive control, that is SMART1, which was, just was used as positive control in this specific case. And here with this enriched T-cell clone, we could actually also basically inject those into the, as I said before, into the heart or pulsing heart of the chicken embryo that is developing but does not have its own immunosystem yet, so that we could actually see how those PBMCs were actually recruited to the tumor and were killing the tumor. This was my last slide because I think I'm a little bit late. So in summary, this is the pipeline that we have developed in the lab for personalized oncolytic approach. And big thank to all the people in the lab. As I said several times, this is unfortunately not really up to date. There are people missing here, but I need to find the time to draw more, which I don't have at the moment. And this is the same people in real life. Thank, of course, to all our funders, and thank you for your attention.
Thank you, Vincenzo, for the fantastic talk. Are there any questions from the audience?
That was fantastic, Vince. I would like to I have two questions. Maybe I missed it, the first one. How much time do you need to identify to have the full screen of peptides, the tumor peptides? And the other one is if you also can target by your approach metastatic lesions, since you with your approach, you trigger a generalized immune response.
Yes. So the first question was the time. The time in theory is very, very short, less than a week. If we had our own mass spectrometry, at the moment in real life, the bottleneck is the mass spectrometry. So usually when we have the samples, we send the samples to the mass spectrometry core facility or collaborators, and of course there is time to wait just there. But in theory, if we had our mass spectrometry, this cannot be done in a week. The second, yes, absolutely. I mean, I did not go into details because it would have required too much time, but the coated oncolytic virus is really multi-step, multi-layer mode of actions. So you do trigger an immune response that goes also to far metastasis. And of course, I'm not sure you can, and that's one part of the answer. The second part of the answer is if you would like to do the ligandome analysis on the metastasis, then you will need to have access to the metastasis. Whether or not that is required, I don't know. It depends how different the metastases are from the primary tumor.
Thank you very much, Vince, for guiding us through these fascinating technologies that you develop. Between your data slides, you had these beautiful illustrations, right? And your peptides that you showed there seem to be distributed in a random way, right? I was wondering, the regulators, how much do they ask about, this nature of the drug, the finally formulated drug, the combination?
Well, all regulatory has been already approved, and I think now, as I said, the clinical trial is already ongoing. At the very, very beginning, in the first consultation that we had in the majority of the country in Europe, the mixing peptides with the virus is seen as reconstitution step, which basically means that it can be done on the bedside. in the morning by anybody. It does not require basically anything, exactly when you mix the antibiotics with the water. And this applies for 90% of the EU country, especially, except Germany, where the clinical trial is now running. At the very last minute, somebody in the PI decided that this was not a reconstitution steps, but it's rather a manufacturing steps. Which basically changed completely. That's why we were also delayed for almost 1 year, because if it's manufacturing step needs to be done in BSL2 facility, and some of the hospitals they did not have the BSL2 facility by the bedside. So the company has built the BSL2 facility in those hospitals in order to start the clinical trial. But again, this is, that's the worst case scenario. The majority of the other consultation we have had, it was seen as a reconstitution step.
Thank you.
Great, thanks for this wonderful data.
Thank you.
One of the challenges we have in TILS treatment, in TILS treatment, tumor-infiltrating lymphocytes, is the killing assay. So how to prove that the specific tumor TILs are really attacking the tumor cells. Is this being possible to do in your chip organoid model?
I would say yes, but the real answer has been extremely challenging, and a couple of my students, they've also traveled a little bit here and there, joining some groups in Milan, probably Chiara, but also Maria Recigno's group, to learn techniques on how to really expand those TILs before really looking. So now I would say that it's everything is working quite nicely in the lab. So there is a pre-expansion of the PBMCs and then co-culturing, and then we see the killing very well. Sometimes we do it with Xeligens, which is this. It's a machine that we have it in the lab. That is, it's a nice machine. It's a golden chip where the cells are seeded, and the impedance is actually registered on the golden chip 24 hours a day, and the data are sent to an iPad that is close to the incubator. And when the target cells are dying, the impedance on the chip is actually changing, and signal is sent to the iPad, and then it builds up the curve that I show, that the curve is going down. So it's actually not measured by hand, but it's just the golden chip that loses impedance when the cells are actually dying. That's how we have done it. It's called Xeligens, the machine.
I'm afraid we are running out of time, but I would like to ask one short question. Vincenzo, stay there. Vincenzo, one more question from me. It's a short one, sorry.
A short answer.
Yeah. So how many peptides can we load, and can we also load peptides that are recognized by MHC class 2 for CD4?
Very short, up to 19 different peptides, but the number is much higher, of course, because every single peptide several times. And we have a very nice paper published in 2020 on cancer research where we have loaded MHC class 1 and 2 peptides. But we can talk more about it.
Thank you.
Much more interesting.
Thank you very much.
Moving on to our next speaker. Our next speaker is Dr. Maria Themeli. She is an assistant professor at the Department of Hematology at VU Max Cancer Center Amsterdam, the Netherlands. In her lab, she focuses on the development and optimization of CAR T-cell therapy for multiple myeloma. And today she will talk about multi-targeting CAR T-cells for multiple myeloma. Thank you.
Thank you. First, thank you for allowing me to be here and enjoying the sun, because I miss it. So I will talk to you about multi-targeting, combinatorial co-stimulation of chimeric antigen receptors, and a little bit about, in the end, about how we initiated logic gating also in CAR T cells in our lab. This is my disclosures. I'm receiving royalties from Fate Therapeutics, but for a product that is not related with what I'm going to talk about. So a little bit of a recap on chimeric antigen receptors. We heard some things yesterday, so a little refreshing your memory. So chimeric antigen receptors are synthetic molecules. They are custom made in the lab, and they are comprising of an extracellular binding domain, binding surface antigens, and it's derived from, most of the times, monoclonal antibodies, single-chain variable fragments. And this extracellular domain is fused to intracellular parts that are borrowed by the physiological T-cell receptor machinery of a T-cell. So we have borrowed the CD3 zeta chain to endow CAR T-cells with activation, and costimulatory domains from the most popular costimulatory molecules of T-cell, CD28 or 4-1BB. So these are what we call the second generation CARs, which are now applied into the clinic. So, as I said, clinical application of chimeric antigen receptors has revolutionized, especially the therapeutic landscape in hematological malignancies, leading to FDA approved and some EMA-approved products for B-cell leukemias and lymphomas and multiple myeloma. But as you can observe in these survival curves from the first pivotal clinical trials, you can see that although there is a clear benefit for the patients, most of the patients eventually relapse. And these relapses are giving us the information that we need in order to improve the design of chimeric antigen receptors. So what are the mechanisms, the main mechanisms of CAR T-cell therapy failure that have been identified up to now? One of the major mechanisms is what we call antigen loss or low expression. What happens is that we have escapes of tumor cells that do not express the target antigen, or they have modified the target antigen, or they have for short time downregulate the expression of this antigen. We also have tumor heterogeneity, which is mostly applies to solid tumors. So not all tumor cells express the target antigen, or they express different antigens. Here we have examples showing how important is the target antigen expression for CAR T-cell therapy efficacy. This is an example of a trial for B-cell lymphoma, DLBCL. You can see here the different expression in 2 patients of the CD19 antigen. And in this study, they found that if the target antigen is expressed below a threshold of 3,000 molecules per cell, then the risk of non-responding to the therapy was higher. Here you can see also how the BCMA, another target for multiple myeloma, can be downregulated during therapy, and then after CAR T-cells are not present in the blood, then you can see the BCMA comes back again, but not completely in the levels pre-therapy. So in order to address this issue, several strategies have been proposed. Most popular are the strategies of targeting 2 antigens instead of 1, what we call OR gates, because when you target antigen A or antigen B, it will result in T-cell activation. This can be achieved either by using 2 chimeric antigen receptors or 1 that bears 2 binding domains in the same molecule, what we call tandem binding. As you can understand, antigen A and B have to be tumor-specific antigens, both of them. So the combination is very difficult to find. Also, in several studies targeting right now CD19, CD20, CD22, there is no real clinical proof that we can overcome the antigen low relapses. And we know that when we target 2 antigens, we will have better anti-tumor effect, but we don't really know what is the mechanisms, how we have to design this double targeting, and what are the requirements, so how we have to address this. Another mechanism of CAR T cell failure, which we know since the very first clinical trial, which is depicted here, is that, as you can see with the red circle, when the CAR T cells are not there anymore, then the instance of relapse is higher. So persistence, a functional persistence of CAR T cells is very important, and what we have learned from the preclinical and clinical data is that when a CAR has the 4-1BB co-stimulatory domain, then the CAR T cells persist longer. It benefits survival. But when we have a CD28 co-stimulatory domain, then the cells kill better. They have a better cytotoxic function. So previously, what we have done is we tried to accommodate these two properties of co-stimulatory domains or co-stimulatory signaling in the T cell into one T cell. So not prefer a CD28 or a 4-1BB CAR, but put these things together. And we tested this in a chimeric antigen receptor targeting CD38 for multiple myeloma. So we combined a second generation anti-CD38 CAR with a 4-1BB ligand. So the 4-1BB ligand will bind to 4-1BB, and so these cells will have both signaling. We found that CAR T cells having this design showed much better in vivo anti-tumor function compared to the ordinary designs of CARs. And when we looked at the mice post-mortem, we found that these cells indeed persisted much longer in higher numbers than the two conventional CAR designs. Why this was happening? A possible explanation that we found, apart from transcriptional changes that happen when these two signaling pathways are active. Another explanation that we found is that, as you can see here with the red, when a 28 zeta CAR is present, then the signaling within the cell is marked by phosphorylation of JAP70 is much higher, much stronger than the signaling when we have a 4-1BB CAR. When this is combined, it seems that the signaling from 4-1BB is dampening, is calibrating the overactivation of CD28 and thus protecting the cells from exhaustion. So having this in mind, we thought, why can't we combine this multi-targeting, this OR gating, with our findings that CD28 and 4-1BB signaling are beneficial for the cell. So in order to do that, we tested the strategy in multiple myeloma, and we combined a CAR targeting BCMA, which is now the target that's used in the clinic for multiple myeloma, with a chimeric receptor targeting CD38, but this time this receptor does not have an activation domain, a zeta chain, but has only a co-stimulation domain. And in this way, we can combine two co-stimulatory domains and two receptors. We hypothesized that in this way, by double targeting, we will increase the avidity of the synapse, and then with two co-stimulatory receptors, we'll improve the cytotoxic and proliferative potential of the cells. And indeed, we tested several combinations of how these two molecules could be designed and could be combined, from a first-generation CAR with a CCR having only one co-stimulatory domain, to two co-stimulatory domains, to a second generation with two different separate co-stimulatory domains. And in all cases, we found that in a simple cytotoxicity assays, we had impressive benefit in the lysis of our tumor cells compared to first generation, second generation, and even third generation CARs. In order to find out how we have to design, then what are the properties that we have to have in mind when we design this double targeting, we had to dissect the two parts of the costimulatory receptor, the CCR, the binding domain and the costimulatory domain. So which is the one that is responsible for the increase in cytotoxicity? For this reason, we made this delta version, which lacks any costimulation, and there is only binder of CD38. And we found that only binding CD38 was enough to increase the cytotoxicity and grants MB secretion from CAR T cells. So even this extra synapse is increasing the cytotoxicity. And we then question, does the binding domain matter? Which one we choose? Does it have to have a high affinity, a low affinity for the target? And indeed, we found that when we lowered the affinity of the target, we had a series of different CD38 binders for the same epitope, and lowering the affinity really abrogated this benefit in cytotoxicity. So a highly expressed antigen, high affinity is required for having this result. We tested again our hypothesis that really it was the synaptic strength that was strengthened by using this machine that is measuring cell avidity. Briefly, the tumor cells, they are put on a chip, and then the CAR T cells on top, and then acoustic forces are applied, and we measure what is the force that is needed to dissociate the T cells from the target. And as you can see here, as expected, when we have double targeting, a higher force is required to dissociate the T cells from the tumor. And that was even seen with the delta, as you can see here. Now, the big question was, is this increase in avidity also help us to recognize lower expression of the antigen? And here we designed K562, so cell lines transduced with a target in levels, different levels, mimicking the levels of expression from the patients. And as you can see here, the lowest expression of BCMA in our cell lines is similar to the median expression on patients with multiple myeloma. And what we found is that indeed, as we have seen with higher expression, we do see with double targeting the increase in cytotoxicity, but most impressively is that when we go to low expression, the conventional CAR designs fail to induce lysis of the tumor cells, while the double targeted CAR T cells still keep maintaining a level of cytotoxicity. Now going to the costimulatory part of our CCR, we found that the double targeting CAR T cells had significantly increased cytokine secretion, IL-2, interferon gamma, and TNF, which you can see here that is not secreted with the delta version, meaning that for cytokine secretion and proliferation of the cells, the costimulatory part is essential. And because we got a lot of questions on what will happen if the tumor cell at some point downregulates or doesn't have the target for the CCR, we designed a system where the tumor cell does not express the target of the CCR, but the microenvironment does. So what happens if we design a CCR that is actually receiving signals from the microenvironment? And we performed this co-culture assay with myeloma cells expressing only BCMA and third-party cells. In this case, there are 3T3 cells, but we have done the same with CD14 positive CD38 monocytes. And then we measured, would the myeloma cells be killed? And would the CCR act in trance, receiving signals from the microenvironment? And indeed, this was the case. The myeloma cells were still being lysed. Our third-party cells were not lysed because the CD38 CCR was not activating, was not mediating lysis. But it could mediate increase of the costimulatory signals within the cell, increasing IL-2 production and proliferation of the cells. So we still can use this strategy by taking signals from the microenvironment. So cumulatively, we tested these benefits of using a CCR in a multiple myeloma mouse model. Here, this design is actually the same CAR that is now FDA approved. You can see that in our mouse model, we made it as a really hard to treat, and this CAR is actually failing. But when we targeted with a CAR and a CCR, we had impressive control of tumor growth and a uniform persistence, increased persistence of our double-targeted CAR T cells compared to the two conventional designs. And that was correlated with a reduced expression of the known checkpoint inhibitors, PD-1, TIM-3, and especially LAG-3. We tested the same strategy in a model of ALL. But in this case, we collaborated with the lab of Michel Sadeline, where they have generated ALL cell lines with very, very low expression of CD19. We went to as low as 20 molecules per cell. And we found that we tested several combinations, and I have to say that the last combination was actually a mistake, because it was not very used. It's not very used that someone would put a second generation 28 zeta CAR with a CCR that has a second CD28. Why would someone put the double CD28? But it was a mistake. But from this mistake, we learned that actually this was the best group performing with very long-term remissions in mice. We found that this was the best combination for double targeting low antigen expression, so using two CD28 domains and the 4-1BB. Now, another question that we received for this strategy is that very well, we also found that it can be also combined with a T-cell receptor, so a T-cell receptor and a CCR would have this a similar benefit. But what happens if you use a high affinity CCR, and when you inject into a patient, there is a chance that the CCR will bind with a higher affinity somewhere else in the body, and not go to the tumor. So we started investigating the idea of having an inducible CCR. So how can we induce the CCR that is being expressed only when is needed locally, and we can have a selective high efficacy of targeting locally. So this is our strategy, and we borrowed elements of the strategy that has been described before, using a CAR and a CCR to selectively target tumors. So by optimizing the affinity of the CAR to recognize only highly expressed antigens, so then only be activated. We designed another version where we would use a lower affinity, so lower expression of the target antigen A would not be recognized, but only high expression, so we would avoid on-tumor off-target effects. This would be combined with the CD3 zeta chain, and activation only of this receptor would lead to the expression of an CCR locally, with it multiplying the effect of binding. In this case, tumor antigen A and B can be theoretically tumor associated. So we designed a strategy where the activation of this first receptor would lead to the expression, the surface expression of a CAR, which is already expressed in the cell. So we're not talking about a transcriptional activation, which is very used, but what we did is that we added an extra domain in the CAR, which keeps the CAR inside the cytoplasm, and only activation would bring then the CCR on the surface. So it's we hope it is a very quick response. And we tested this strategy by activating CCR transduced cells with beads. And we found that even with 6 hours, the cells start expressing the receptor with peaking at 48 hours. And when we remove the beads, then after 16 or 40 hours, the cells would internalize the CCR again. When we activated with cells, so irradiated cells, we found the same results. In 24 hours, the cells start express the CCR, and after removing the cells, again, the same kinetics. So we are very excited about the potential of this strategy to locally targeting tumors with full double targeting and co-stimulation. And I will summarize somewhere here. I showed you that combining a CAR and a CCR increases the avidity of binding. We have optimal co-stimulation when we combine the two CD28 and 4-1BB domains. And the CCR does not mediate cytotoxicity, so our target selection is unrestricted, and we can use also microenvironmental expressed targets. We have applied this strategy in multiple myeloma and ALL models, and we are currently into an implementation trajectory for starting a phase 1 clinical trial with a BCMA and CD30 CAR and CD38 CCR for multiple myeloma, but because CD38 is expressed in all hematological malignancies, we think that it can be applied across other malignancies as well. And we are currently investigating this novel system for activation-inducible receptors. And we hope that in the next months we will have more data on the potential of this strategy. So somewhere here I would like to thank students that have worked on these projects, Aphrodite, George, and Dr. Richard Groen for helping with the mouse models, and Michel Sadelein and his group for helping with the ALL low expression, antigen expression model, and all other collaborators and funders. Thank you.
Thank you very much for this very interesting talk. It's actually very exciting that you can target cells with very low antigen expression, especially in multiple myeloma patients, the most of whom have already received anti-CD38 for sure, and maybe also anti-BCMA. Any questions from the audience? Dr. Mouliakakos. Can you go to the microphone, please? Because there's also an online.
Great talk. I really liked your concept with your end concept where it's not transcriptionally or translationally dependent. Have you actually thought to expand your analysis on looking whether this provides an advantage also in terms of tumor-induced suppression? Because an end signal that really or might depend on transcription translation might be also more, let's say, sensitive towards tumor-induced suppression, because we know, for example, if they drive the T cell, not the CAR T cell, into a metabolic senescence, that also transcription translation goes down, which in your case would not happen.
We have not looked at that. Very good suggestion, actually. But what we have looked, and we are, so maybe that would be a part of our investigation. Thank you for the tip. What we have found is that, indeed, we find that these cells, even compared to our CAR plus CCR, which were the best performing in persistence, the cells with the inducible receptor, they had even better persistence. So they do even better. So that might be one guess. on why this is happening. We are mostly thinking on the tuned signaling, that it's not the continuous signaling that the cells have, because it comes on the surface and then it's internalized. But I hadn't thought that because of their non-transcriptional. That's a good tip. Thank you.
Another question, actually a technical question regarding this loss of antigen, because I mean you know this paper probably better than me from Hudecek, with this high-end microscopy where you could really find the single antigens. Do you have any idea how the development is regarding assessing the specimens? Because even our agency that allows or does not allow financing the CAR therapy now is really allowing us to treat patients that are CD19 negative in the relapse with CD19-directed CAR T cells, because we have the clinical data that our immunohistochemistry is not good enough to really assess it. So in case they have not received a previous anti-CD19-directed therapy, they are allowed to get the CARs that are against CD19.
Yes, and I actually think that indeed, and most of the studies, also in myeloma with BCMA, they say, level of expression of BCMA, if you see in the clinical sites, was not correlated with outcome. But all this evaluation of BCMA was done with immunohistochemistry. So I think, and now studies showing with molecules per cell, which is a much more sensitive technique, they show that indeed we know that the CAR is very sensitive to target antigen expression. So I think that, I don't know if it can be applied in the clinic, the more sensitive detection of antigens, but I think that with strategies like that, at least the level of expression that we measure with FACS or with immunohistochemistry is not very relevant.
Yeah, thank you.
Maria, it was great. I would like to ask if you anticipate an increased toxicity by using a double CD28 and 4-1BB?
So there is one cytotoxicity that we might anticipate that would increase the off-target, so the CD38-mediated cytotoxicity. We have tested that, so it depends on the binder. So it depends on the scFv that is being used. So we have selected specific scFvs that there is no cytotoxicity against normal hematopoietic cells, because we know that the normal hematopoietic cells also have lower CD38 expression than multiple myeloma. A second toxicity that we anticipate is from the cytokine secretion, so higher CRS incidence. So in our phase 1 study, we will start with as low as 20 million or 25 million, if I'm correct. So we will do a dose escalation and see what is the effect.
Thank you very much. If there are no more questions, then thank you very much, Maria.
And our next speaker is Dr. Eliana Ruggiero. Eliana is a project leader at the experimental hematology unit in the San Raffaele at Milan, and she will talk on TCR-based T-cell products for solid tumors.
So good morning, everyone. First of all, I would like to thank the organizer. I was never in Greece, so it was my first time, and despite the delay, it's a really nice city. I had a very problem with flights, but I'm really happy to be here. So today I would like to give an overview of what we are currently doing in the lab in the context of TCR gene therapy. So I would like to start by pointing out that how now immunotherapy is really a fifth pillar for patients' care. And in particular, we can today modify patients' T cells at multiple levels to generate personalized cellular-based drugs. What I will be focusing on today is how we can work on the recognition of the tumor cells, and how we can work on counteracting the immunosuppressive tumor microenvironment. So you know that we can provide T cells with the ability to recognize the tumor by employing T cell receptors or chimeric antigen receptors. If you look at what is going on, this is an old picture from 2019, but already gives you an idea. The clinical trials that are out there right now, you see that the majority of clinical trials are mainly based on use of CAR T cells. And actually, it's not difficult to understand why if you look at the data and the success of CAR T cells in hematological diseases. In fact, if you look here at the complete remission rate, you see that it's very, very high, especially for B-cell lymphoma, BALL. Unfortunately, you have also to consider that the relapsing rate still maintain quite high. And in addition, you also need to consider that, unfortunately, for solid tumor, there are still some issues with CAR T cells, but also with TCR T cells, of course, and still more needs to be done to improve the outcome of patients affected by solid tumors. So what are why there are these disappointing clinical benefits? There are several reasons. One might be the limited number and heterogeneous expression or loss of antigens that can be targeted by the therapies, the paucity of tumor-specific receptors, the poor T-cell persistence and the fitness in the tumor microenvironment, the impaired trafficking to the tumor site, but also the low mutational burden and the high heterogeneity of the disease and the cancer cell acquisition of epigenetic or genetic alterations. So despite, as I mentioned before, despite the great success of CARs, actually now in our lab we are a little bit outsiders and we're working with TCR therapies. So why we decided to work with TCR therapies? Actually, I would like to start with the main problem with TCR therapies, that is that TCR can recognize antigens in HLA-dependent manner. That means that patients that are selected for TCR therapy, they need to express not only the antigen but also the HLA. However, there are still some good side of the TCR therapies that I think might offer some unique opportunities for TCR field. The first one is that T-cell receptor can virtually recognize every tumor protein. So not only the top of the iceberg that are represented by the surface molecules that only CAR T cells can recognize, so CAR T cells can recognize only surface protein, but also all the hidden part of the iceberg represented by the intracellular molecule. And those account for 70% of the human proteome. In addition, we need to consider that the T-cell receptor have an increased antigen sensitivity compared to CARs, while they are able to recognize even as low as one antigen on target cells, while for CAR you need much higher number of antigens. And the most important thing, I think, that even if there are few clinical trials, the few ones that are out there, they show that for solid tumors, they might have some beneficial effects of TCR therapies, as shown here in different papers for colorectal cancer, PDAC. But the most important thing that we learned from these studies is that the T-cell can traffic to the tumor sites, but the majority of these beneficial effects were observed with TCR therapies that were mainly targeting neoantigens. And so this means that you need to have a personalized therapy that is also cost-effective. So we know right now that TCR gene therapy needs to face several challenges to fulfill its potential. When you start from a patient, you need to, first of all, identify the proper target antigen that you want to use as a tumor antigen. You need to identify a proper T-cell receptor that you want to employ for your therapy. You need to engineer lymphocytes and then perform in vitro and in vivo all the preclinical safety and efficacy testing. So the first question that you need to ask is which target antigen do you want to use as a tumor antigen? And this is the first thing that you need to address. And we know, as I mentioned before, that for TCR you can have a variety of antigens because you can exploit full amount of surface and intracellular molecules. However, you can distinguish two main categories: the tumor-specific antigens, that are the viral antigens, the neoantigens, or the cancer germline antigen, that have a very good safety profile because they are only expressed on cancer cells. However, you will end up with a personalized therapy in the majority of cases. And you also have then the tumor-associated antigens that might have the issue of the expression also on healthy tissue because they are overexpressed on tumors, but they are still present on healthy tissue. But however, they will enable you to treat a high majority of patients. So this question of which target antigen we need to use is a long-term question, and the already back in 2009, the NCI developed this priority ranked list of cancer antigens based on the likelihood for efficacy in cancer therapy. You see here all the different criteria that were considered to define this list. And by applying this list, they could identify WT1 as a top-ranking ideal cancer antigen for immunotherapy purposes. But what is WT1? This is a zinc finger transcription factor that plays a key role in tumorigenesis, whose expression is maintained in relapsing diseases and is often used as a marker for minimal residual disease. And nicely, it is expressed on a variety of tumors, not only hematological tumors, but also solid tumors. So we started to address the value of WT1 for leukemia, and in particular for acute myeloid leukemia. And we needed to identify proper T-cell receptor to generate our therapy. So we set up this pipeline in the lab that enables, starting from healthy donors or from patients, to identify anti-tumor T cells, expand them from healthy donor from patients, and then isolate and sequence the T cell receptor. And by applying this pipeline, we could then identify, starting from 14 different healthy donors, 19 different receptors that are now in our library, and we could then define which one are the features that are more relevant to identify the lead candidate. So by applying a funnel approach, we could then end up starting from 19 different T-cell receptors to one TCR that is now our lead candidate and that we are exploiting for clinical validation. However, you know that we need to engineer lymphocytes, and the classical approach to do that is the TCR transfer, so meaning that you start from healthy donor T cells and you can transduce them with a lentiviral or retroviral vector encoding for the tumor-specific TCR of choice, and you will end up with such a situation. So you will have T cells that express on the surface not only the endogenous T cell receptor that was already present, but also the transgenic TCR that you inserted, and you might have the generation of hybrid receptor generated by the mispairing of endogenous and exogenous chains. So to overcome this issue, Chiara's lab, together with Luigi Naldini's lab, developed the TCR gene editing approach that is based on the idea that you can destroy the endogenous TCR repertoire and have an army of T cells that only express on the surface the anti-tumor receptor. You see here the feature of the different approaches. So with the TCR transfer, you might have the problem of hybrid TCR generation. However, you have the good thing that the T cells are manipulated in 2 weeks of culture. With the TCR gene editing in the beginning of the procedure that was performed with zinc finger, as nucleases to destroy the endogenous TCR repertoire, the procedure was taking up to 5 weeks, so quite a long time. However, thanks to the CRISPR-Cas9 and the base editing procedure, now you can generate engineered T cells in 2-3 weeks of culture. So we decided in the lab to exploit the TCR gene editing approach in collaboration with Intellia Therapeutics, and in particular we decided to engineer T cells by destroying the endogenous TCR repertoire and inserting the therapeutic T cell receptor not randomly into the genome but into the alpha chain locus. And in this way, you will avoid the insertional mutagenesis risk and generate a more homogeneous T cell product, because the T cell receptor expression will always be driven by the same promoter. So this approach was shown to be promising in CAR T cells, but also in TCR T cells. And in fact, when we tested it in acute myeloid leukemia model, so mice were infused with leukemic blast and treated with T cells, we could see that mice receiving T cells generated with this approach, they could nearly completely control leukemia outgrowth compared to mice treated with a standard protocol in which the TCR was inserted randomly into the genome. So we decided to exploit this procedure and push even further, and so we tested this cellular product specific for WT1 in 3 different in vivo models. In AML, you see here the complete eradication of leukemic outgrowth in mice treated with our TCR engineered T cells in red compared to the control mice, but also in acute lymphoblastic leukemia and also in glioblastoma, we had an increased survival of mice treated with our engineered T cell product. So we have now the pipeline, we have the TCR gene editing set, and we decided to further exploit all our pipeline to identify novel T-cell receptors, specific not only for hematological but also for solid tumors. And now this is our library. We have around 56 anti-tumor T-cell receptors that are specific for different diseases, hematological and solid tumors. And if you look at the ribbon connecting the TCR with the disease, you can see that each T-cell receptor can be used for the treatment of several diseases depending on the expression of the antigen. And nicely, those TCR are mainly recognizing epitopes that are restricted to HLA alleles that are common in the Caucasian population to increase the applicability of the therapy. In case of the solid tumors, we also started to work mainly with gastrointestinal cancer cells. So we started with colorectal cancer and PDAC, and to identify the proper target antigen that we wanted to use for our TCR approach, we started from studying differential gene expression analysis, identifying overexpressed genes, immunogenic peptides that then were used for our TCR hunting pipeline. And by doing that, we identified several T-cell receptors recognizing different antigens, and that then were tested for TCR gene editing, and we could see the ability of our engineered T cells to kill PDAC cell lines, but also colorectal cancer organoids or PDAC organoids. You see here, these are InQSite graphs, generated graph. You see that while you do not have any death of the PDAC organoids or colorectal cancer organoids when they are cultured alone, when they are co-cultured with our engineered T cells, we start to see an increase of the killing over time. So we have now this library, but we know that is not enough, especially in the solid tumor context, because the T cells will migrate to the tumor and will also be inhibited because there is an immunosuppressive tumor microenvironment. And in particular, we know that there are inhibitory receptors that are counteracting the action of the T cells. We know that from literature, but we also know from our experience in hematological diseases, in which we could track in patients upon transplant tumor-specific T cells by employing dextramers, specific for different tumor antigens. And we could see that when we looked at the expression of inhibitory receptors over time post-transplant, we could observe an increase of these warm colors bar, indicating co-expression of inhibitory receptors over time. And as a control, we had viral specific T cells, and we could not see these very warm colors here, indicating that those viral specific T cells do not get as exhausted as the tumor specific one. But we also know that from solid tumors, in fact, we had the chance to work with the primary and metastatic colorectal cancer specimen from patients, and to analyze those specimens by using different technologies, so RNA-seq, immunohistochemistry, but also flow cytometry. And you see here, these are the results of the flow cytometry data. When we look at CD8 but also CD4 cells, we could see that at the tumor sites, we have a co-expression of different inhibitory receptors. This is in the primary metastatic tumor, but also when we look at the primary tumor, but also when we look at the liver metastasis from colorectal cancer, we could observe the same. So co-expression of inhibitory receptor, you see here, CD39 is always present as inhibitory receptor, combined with several others. And this was confirmed by immunohistochemistry. So we need somehow to do something, because we know that these T cells will not be able to work properly due to the presence of this inhibitory receptor expression. And in particular for colorectal cancer, we could observe how CD39 seemed to be a major player for the T cell functionality. So how can we reinvigorate the T cell responses? The first thing that we need to know is that, of course, the inhibition of inhibitory receptors, so blocking inhibitory receptors, will help in reinvigorating T cell responses by increasing proliferation and cytotoxicity of T cells. And this can be done by using immune checkpoint blockades. That is a therapy that is nowadays widely established and approved by FDA, and has already been tried in several combinations in clinical trials, also with CAR T cells. However, you also know that by using immune checkpoint blockade, you will unleash the complete T cell of your body against, so also the autoimmune T cells will be completely unleashed, and so you might have some side effects. And to avoid this, we decided to exploit again genome editing in the lab to destroy inhibitory receptor together with the endogenous TCR and to generate T cells that only express on the surface the tumor-specific T cell receptor and that are able to counteract the immunosuppressive tumor microenvironment. This approach of multiple engineering in T cells has already been published by Carl June group several years ago, nicely showing that these T cells were not counter-selected in patients, so indicating that despite they are multiple engineered, they are still able to persist in the patients. There was no immunogenicity observed in this clinical trial, and indicating also that multiplexing in genome engineering is safe and feasible. We set up then in the lab a procedure to perform this multiple disruption of genes. And we have now the disruption of several inhibitory receptors. You see here LAG3, TIM3, 2B4, CD39, TIGIT, and PD1. And nicely, despite the manipulation procedures, the cells were still able to expand and to proliferate in the same way compared to untransduced and manipulated cells. And they still maintain a memory stem T cell phenotype. That is a phenotype important because it has been shown to be endowed with multipotency and long-term persistence, not only in hematological malignancies, but also in yellow fever vaccination, in gene therapy, and in immunotherapy studies. We decided to test in vitro and then in vivo these engineered T cells. And so you see here that, for example, we performed CD39 knockout or TIGIT knockout and tested the ability of our T cells to kill primary and metastatic patient-derived organoids from colorectal cancer patients. And you see here that when we performed an IncuCyte assay co-culturing our T cells with organoids, we could observe a preferential killing, so an higher killing of organoids when they were co-cultured with our CD39 knockout T cells compared to the CD39 wild-type counterpart. You see also better here from this IncuCyte image that here, when we used the CD39 wild-type T cells, there were still some organoids present, while they started to be completely lost here when the organoids were co-cultured with CD39 knockout T cells. And also when we performed TIGIT knockout in T cells, we could see that there was a preferential killing of target cells when they were co-cultured with TIGIT knockout T cells compared to the TIGIT competent counterpart. And this was done by employing a different T cell receptor specific for different solid tumor antigens. However, I would like to focus on CD39 as an inhibitory receptor because it's the one that we investigated right now more. CD39 actually is a quite important molecule. It is involved in the functionality of T cells because it plays a role in the ATP adenosine pathway. And we know that when in the tumor microenvironment there is ATP, we have pro-inflammatory environment, while when we have adenosine, we have an anti-inflammatory environment. And CD39 is here catalyzing the hydrolysis of ATP to AMP, and AMP then will be hydrolyzed to adenosine by employing another receptor, CD73. So CD39 is really here in the middle, and we think that its ability to enhance the functionality of T cells is mainly due to the inability of CD39 knockout T cells to have AMP as a substrate for adenosine generation. And actually, this is something that came also from our data, because while we know that CD39 knockout T cells are able to increase the killing of target cells compared to the CD39 counterpart, we could nicely observe that when we added adenosine to the co-culture, we could completely lose this beneficial effect, indicating that it might be really the reason of the inhibition of adenosine accumulation in the microenvironment behind the functionality, increased functionality of these CD39 knockout T cells. We also decided to test combinatorial strategies because, as I mentioned before, on the tumor you don't have only the expression of one inhibitory receptor, but you have co-expression of several inhibitory receptors. And so we decided to try to combine CD39 knockout with also anti-PD1, so to block PD1 by using PD1 blockade. So we added in the co-culture nivolumab, and as you can see here, we already could see before the addition of nivolumab the beneficial effect of CD39 knockout T cells in killing target cells, organoids, but this activity could be greatly enhanced by the addition of nivolumab. So yes, combinatorial strategy, of course, they have some beneficial effect in killing the tumor cells, and we decided then to also test in vivo the efficacy of the inhibitor receptor disruption. And we did that with CD39 knockout T cells in 2 different models of colorectal cancer. In the first model, we had subcutaneous infusion of organoids in mice, and then mice were treated with T cells. And when we looked at the tumor growth, we could see that after organoids injection, so some days after organoids injection, we could already see a beneficial effect of CD39 knockout T cells in mice because we had a reduced tumor growth, but this was even nicely observed in a different tumor setting in which the organoids were infused in the liver of the mice in order to mimic liver metastasis from colorectal cancer, and we could see that upon T cell infusion we could observe a statistically significant control of tumor outgrowth in mice compared to the control mice, but also to the CD39 wild type treated mice. So this is true with CD39, but we also tested that with LAG3 engineered T cells in a different model. So we test them in a multiple myeloma model. In this case, we had mice that were infused with multiple myeloma cells and then treated with T cells. All mice eradicated the tumor. But then we performed the second tumor cell infusion, and what we could observe is that while mice treated with engineered cells, but that still express LAG3 on the surface, the majority of mice were able to control the tumor challenge, but some mice still developed the tumor, while all mice treated with LAG3 knockout T cells were completely able to control tumor regrowth upon tumor challenge. So with this, I would like to conclude, and what I wanted to point out today is that we now have all the different features of the pipeline set up to have a T cell therapy approach. We have now our library of T cell receptor that are specific for hematological but also solid tumors that we are currently validating. We have now the CRISPR-Cas or other nucleases, so I didn't mention, but we are working also with base editing to destroy the endogenous TCR repertoire, but also to counteract the immunosuppressive tumor microenvironment. And we can see that actually the TCR-edited IR-disrupted T cells have a functional advantage in eliminating target cells and may benefit from combinatorial strategy to enhance ACT efficacy. So my last slide is the most important, so the acknowledgement. I would like to thank all the people in the lab, especially Chiara that is supporting us working on all the different projects, all our collaboration partners inside and outside the Institute, the funding agencies, and last but not least, every one of you for your attention. Thank you. This is the group with a bias in gender.
Great talk. Thank you, Iliana. So, Maria.
Can you hear me? Yes, excellent, Iliana. I have 2 questions, actually. First, so the mechanism of the benefit of introducing the CAR in the tract locus was mostly seen with the CAR that has a lot of tonic signaling. So in the case of TCR, what is the mechanism? Because there is no tonic signaling, but is this the level of expression? Have you looked at that? Is that
yes, we did some study, and I think it's the level of expression because now it's uniformly expressed, and it might be because of how the TCR construct is made with the beta chain to a peptide alpha chain. So I think that when you insert it into the alpha locus under a specific promoter, because we observed a difference compared to the CAR story. With the CAR story, the beneficial effect was when you were using the endogenous promoter. For us, we could see a benefit when we were putting into the tract locus band under a specific promoter that was the F1 alpha.
Okay.
So it seems that you need a strong promoter to drive a high expression of the T cell receptor, but then, yes, I think that there is a problem of the expression, so the expression level makes the difference. Okay. So it's a more, a more uniform expression of the T cell receptor there, and I think that this helps the cells to work better. But it was shown also from Bush group that you have T cells that are working better if you do insertion of the T cell receptor in their proper position where they should be physiologically.
Okay. The second is that a question is that in the publication of Carl June, Ruth and NYE, so they show that the more you multiplex, so if you add more knockouts, then the yield is lower. Have you tested that? Do you see the same?
Actually, we are using a different approach from Carl June, so we do not have the multiple disruption, but with a different protocol that we are setting up with Intellia Therapeutics. So we have sequential disruption of the things. So we destroy the TCR chains and the IR, but in different moments in order to avoid also the translocation issues and the poor persistence. So yeah, I cannot disclose much, but we have a different procedure.
Thanks.
One last question because we are out of time.
Yes, thank you. Eliana, hi, great talk as always. I have a question. You know, I'm interested in the survival of T cells. So again, as a segue to what the comment was just made about that paper from Carl June, you mentioned these multiplexer cells survive, they stick around for a long time. They don't have a selective disadvantage. What I'm asking is, do they have a selective advantage? And the question is, are you foreseeing to use your approaches for a translational application only after lymphodepletion, or are there scenarios where you think these cells might have a selective advantage indeed and expand better?
So that's a great question. Actually, we are working in looking at also the gene expression profile of these multiple genetically modified T cells. And with the approach that we are setting up with Intellia Therapeutics, actually, we see that they persist longer, but as I mentioned, it's a different procedure for disruption of the genes. So it's not like the Carl June did, that he destroyed everything at once with increased translocation and all the other issues. It's a bit different. And so far we did not see, at least from the first results of gene expression, preferential or some differentially expressed genes that might indicate a preferential survival of those T cells, but we have just very preliminary data, so we are working more on that to better characterize those T cells. We know that they are not surviving less, we know that they are surviving compared to in the same level compared to untransduced T cells, so they have the same ratio of proliferation and survival. We do not see a lower yield also when we culture them for a long time or we put them in mice. So this we know. We are investigating better.
For a clinical application, you would see like using this first in patients that are lymphodepleted, right?
Yes, what we are doing, the first one, the WT1 TCR that is going around clinical trial for AML patients, this occurs in patients after lymphodepletion.
Thank you.
Yes.
Thank you very much.
Our last speaker is Professor Dimitris Mouliakakos. He is the director of the Hematology and Oncology Department at the Otto von Guericke University in Magdeburg, Germany. His center has actually expanded the applicability of CAR T cells from malignancies to autoimmune diseases. His talk today is about CAR T cells for systemic lupus erythematosus, and we are ready to listen to you.
Yeah, thank you very much for this kind introduction and invitation. Actually, this is my second time being in this meeting. The first time was 7 years ago, the first one in Thessaloniki, when I was still a resident. So time has passed, and it's really nice to see how the society is evolving at the meeting. So today I will not talk about fancy technologies like the previous talkers, so I feel like a dinosaur. I will talk about actually the second generation CARs and how you can repurpose them. And I will tell you actually a clinical story. So this is why we partly are here. We know immunotherapy has really heralded a new way to treat patients, especially the cell therapeutics. And this is something I will also not tell you anymore because you have heard it. But this is just to show that I will really talk about the second generation CAR T cells that found their way into the clinics as a first agent, and they are directed against malignancies that are B-cell derived. So their target is CD19, that is found on several B-cell-derived malignancies, and as the name also implies, on B cells that are non-malignant, which is important for our clinical story. And we all know, and we have seen it, this is a success story in terms of cancer, and the indications in cancer are increasing, and also the lines of treatment are earlier and earlier. But the question today is CAR T cells and beyond, and my question is probably CAR T cells in cancer and beyond. So what you will hear as a next part after our talks is actually the point of care production. And I think this is a very important thing. I will not talk a lot about this because this is the subject of the next section, the next talks. But I just want to mention that while I was in Erlangen, so before getting the position in Magdeburg, we spent around 10 years establishing the production of CAR T cells on site. And we had the license to use a construct, a CAR T cell that is directed against CD19 with a 4-1BB co-stimulatory domain, as you have heard. And this was actually done within a cooperation with the Hellenic biotech in order to use those CAR T cells within a clinical trial. But at the same time, we are allowed to use them in individual treatment attempts. And here you see an example where we used it in Burkitt lymphoma. So this is a very aggressive type of B-cell-derived malignancies. And this patient received a very strong chemotherapy for almost a whole year. But soon after stopping the chemotherapy, he relapsed. And you see the relapse was in the central nervous system. And normally there is not much you can do about it. And we tried our CAR T cells in this patient, and they worked very well, as you can see here. So this use of CAR T cells that are produced on site can really help the patients, especially in indications, malignant indications that are not covered by products that are approved. Otherwise, he would not stand a chance. So this, of course, was used in patients with cancer, in patients with follicular lymphoma, for example. Back then, there was no approved drug, and so on. And we have a very strong department for rheumatology, and scientifically we work very close together. And we also drink a lot of coffee together, or drank a lot of coffee together. And during one of our lunch breaks, while drinking coffee, we started discussing about lupus. And as a hemato-oncologist, I always thought, okay, cancer patients are really the ones that are the sickest. And soon after talking with Professor Schett, I realized, okay, this might not be always true. So this is a very severe disease. We have around 100 cases per 100,000 people. We have a relatively high number of patients in Europe, so this is a rather relevant patient group. The mortality is increased, the quality of life of those patients is decreased. It affects normally young women. Actually, everything can be affected, so all organ systems are affected, and there is no cure, so you have probably a lifelong treatment. And in the cases where you have a refractory disease, there's no actual standard of care. So when you look at the pathophysiology, you see that B cells, this B stands for B cells, are quite central in the pathophysiology. And as if you remember, B cells are actually one of our off targets when using CAR T cells. But then if you look also here carefully, you see that T cells might also be involved. So the TH stands for T helper cells that might promote the B cell to release an autoantibody that in most cases targets autologous double-strand DNA. It forms a complex. You have a complex antibody deposition into organs like the kidneys, where we have constant inflammation. This constant inflammation leads to tissue destruction and organ failure. So people, for example, require technical support for the kidney function. They got heart failure because they had heart fibrosis after the so-called Lippmann-Sachs endocarditis. So it's really, really a bad, or it can be a bad, very bad disorder. And of course, targeting B cells seems very obvious, and people have done it. And they have done it a lot. And for the first trials, we're actually using an antibody that we also know from cancer therapy is an anti-CD20 directed antibody. This is also important. It's called rituximab, and you see one of the biggest studies, the Explorer trial, which is also more than 10 years ago, and they treated their patients with rituximab versus placebo. And you see here this BLAC, they have a lot of very innovative names in rheumatology for describing disease activity. So one of the names is BILAG score. The BILAG score between the both groups, placebo and anti-CD20 antibody group, is really the same. And also the flare-free survival or rate is also the same. And this goes on. So data on using rituximab or B-cell depletion in lupus is not very striking, which of course leads to the important question, does it actually make sense to deplete B-cells in lupus or not? And based on this data, it might not make sense. So is it really worthwhile following this up? And at this point, you really need to look more in depth. I think this is also very important in general when you look at clinical data. And you need to ask yourself why it might not work. Is it really because the concept is wrong, or is it the way we do it that it might be wrong? And if you look at how an antibody works, you see that an antibody does not work by itself. So it needs some partners. So if an antibody binds to a B cell, for example, it needs macrophages to take up this B cell. We call this phagocytosis, that is antibody dependent, antigen dependent. If the macrophage or NK is activated, they need to release certain molecules that drive the destruction of the target cells, the cytotoxic effects. And then you also need complement, actually, that leads to destruction of the target, which is marked with an antibody, which is cell independent, so you don't need other effector cells. And I described to you that you have this constant inflammation, this complex that are created in lupus patients, and this constant inflammation actually leads to a vast depletion of complement factors. So if you look at the patient and you look at in the blood, you see complement depletion, which is actually a surrogate marker for disease activity. So they don't have complement. This might not work very well. Then there is data that describes that the macrophages are not performing very well. So phagocytosis is defective, and also their ability to secrete tumoricidal, let's say, molecules that might kill the B cell is also altered. So there is actually an incomplete clearance of B cell and B cell derived cells when you use those antibodies, and when you look in patients and also murine models. So it might be that actually all the cofactors that help an antibody being effective are not working in lupus that well, so this might be a reason that we don't see clinical efficacy the way we want. And then the other important question is regarding the target. Maybe the target is wrong. So I said that that B cells play a role, but B cells undergo a development, right? So they also age and develop during their lifespan, and they also change their phenotype. So if you look at CD20, you see that they express CD20 at a rather early time point, but lose it when they turn into plasmablasts or long-lived plasma cells. If you compare to CD19, you see that they lose it after the plasmablast stage, and there is, for example, CD38, which they keep as plasmablasts and long-lived plasma cells. So in case our target population, so the bad B cells, are rather plasmablasts or long-lived plasma cells, we would not target them using an anti-CD20 approach. And this is also important, keeping this CD38 approach in mind, because people actually used anti-CD38 antibodies, which I will show you later. So what we else see in lupus is that actually plasmablast frequencies increase, and you see a so-called plasmablast expansion, a rebound, when you treat those patients with anti-CD20 antibodies, like a homeostatic proliferation of plasmablasts or whatever. There's no explanation yet for this. So as I said, CD38 was used by colleagues at the Charité in Berlin. They published it in the New England Journal 2020, and it was used actually in patients with lupus, in single patients that were refractory to the standard treatment. And you see that daratumumab, which we also use in the treatment of cancer, and especially in myeloma patients, had an effect. So they looked at the kidney function as a surrogate for the disease activity, and kidney function improved. And here another interesting score, it's called SLEDI score. It's like the BILAG score and disease activity score, a combined one. You see that this goes down. However, this was not forever, and people were still on drugs, so they were not treatment-free. And as I said, in looking at all these things, CAR T cells might have certain advantages, even old CAR T cells from the second generation. They are not dependent on macrophages or complement. They are rather self-sustained. They do their work by themselves, first of all. And second of all, we know that they can deplete B cell at a very deep level. So we know here what happens when you look at the B cells in our patients. They go to 0, and they really take some time to come back. And they have a different target in our case, the CD19. So if we assume that it's the plasmablast, not the long-lived plasma cells, it might work. So if in this case I would have a BCMA targeting CAR T cell, I would probably, or CD38 targeting CAR T cell, I would probably have used that, just based on what I showed you and the expression on long-lived plasma cells, but it might have been good that we did not have such an CAR T cells back in time available in our lab. So this is just a biofantasy, right? CAR T cells might work in lupus, and there was actually a paper in 2019 in a murine lupus model where they actually used a CD19-directed CAR T cell, and it worked. So here you see the kidney function that actually improves, so less protein in the urine is good. So you see that the mice that receive CAR are doing better, and they also survive longer. And this was part of our talks, but then we had a patient. So they had an urgent case that they wanted to discuss with us. So this was only talking. And this young lady, she was almost 20 years old. She had lupus since she was 14. And it was very severe. So almost everything was involved that could be involved. Her kidney function was that bad that you could say, okay, in the next 2 to 3 years she will need support. She had her heart function was affected by the fibrosis, so she could not do sports, she could not go to school. So she was really very much affected, and she already received many agents and had already received many agents. So she was already beyond state of the art in terms of treatment. Also agents that are B-cell directed. So in this case, we really started talking about a real patient and a patient that needs treatment. Otherwise, we could not foresee how this patient would survive the next 10 to 20 years. And she was a very young lady, so we talked to her. We talked to her parents, told them, yeah, we have the possibility to produce this kind of cell, and we have no idea what will happen when we treat you, but based on what we assume, and based on the science that we have done and that other people have done, it might work. And she gave very fast a consent. She said, this sounds like only 10 days of hospital and then I can go back to school. We said, yeah, might be. And of course, we were very strict with our explanations, but she was very enthusiastic. So, and her parents also gave their consent, so we went on. So the first thing, of course, was to see if we can produce CAR T cells from a lupus patient, and it worked. So we could produce CAR T cells, but as you can appreciate here, we had a lot of CD4 T cells, and people that work a lot with CAR T cells, know that the CD4 T cells are probably the ones that make the side effects. So we see, for example, in CAR T cell productions where they take the CD4 and CD8 and separate them, put them together in a fixed ratio, less cytotoxic effects. So we have a lot of CD4 cells in the back, and I also showed you CD4 cells are actually a driver, or can be a driver of lupus. So this was not good. Then we picked a dosage of 1 million CAR T cells fresh per body weight, but this was only based on what we are using in our clinical trials against B-cell-derived malignancies. And we infused them. So no problems during infusion, and you can clearly see that CAR T cells are expanding in the patient's peripheral blood, and they reach the peak at day 9. This is something that we really see also in our in our malignant or patients with malignant disorders. And you see that there were some B cells left, despite all these B-cell directed therapies previously, and they were all gone after CAR T cell treatment. So this was good. The patient was doing very well, no toxicity, nothing. So we were very happy. So those first 10 days were more or less sleepless for us, because of course it's a very strong, of course, responsibility that you have in such a experimental treatment. And what you nicely can see here is the double-strand DNA antibodies. Yeah? Here, the red line marks that when we gave the CAR T cells, they went down to 0, very fast. Here you see what happens with the kidney function. Proteinuria went to normal values very fast. Here you see what happens to the complement factors. And as I told you, complement is a surrogate marker for disease activity. So complement normalized C3 and C4, which fits very well to what we have, of course, we are seeing clinically. And then another thing that we see in patients which we treat with CARs is actually hematotoxicity. This is something that we underestimated at the beginning. We were focusing on CRS, on ICANS, but actually a very large proportion of patients with CAR T cells have long-term hematotoxicity, which is really difficult to treat. So you might need stem cell boost, you might use TPO receptor agonists, and which we also were afraid of in this case, because we also know that lupus can affect the bone marrow function. What you can see here is that all cell lines actually regenerated very well. I mean, the erythropoiesis is not 100%, but she had a iron deficiency erythropoiesis before, which she maintained also afterwards, but this is not an issue. So all cells came back nicely. And you see here the SLEDAI score. I mean, dramatically, this clinical score decreased. So it went after day 44 to 0. And this is a patient now. So 2 years afterwards. So we might also have our patients that every year says 1 year after treatment, 2 years after CARs, hopefully 20 years after CARs, she will be treatment free. So she is under no medication, has no symptoms. She has having her normal life back. She's also actually in a dance club, and also so she's really doing very well. Everything is in physiological range. The CAR T cells are not detectable, and we can also discuss later probably if it's really necessary to have persistent CAR T cells in cases like autoimmune disorders in opposition to malignant disorders. B cells have recovered, which is very interesting, and I will also show you data on that. And she never required IgG substitution, so she maintained also her immunoglobulins level at a very stable very stable concentrations. And of course, this was one patient, which was like a miracle, and she's still treatment-free and cured. I don't know, it's probably very early, but she does well. So, of course, many other patients asked when this thing was published. And back then, when I was in Erlangen, we really started to use this license to treat the patients. Of course, you are limited to a certain number because afterwards you need a study, but the studies, of course, take time in order to be approved. And as you can see, we had in total 5 patients, and this just shows you that it worked. So we could generate CAR T cells in this Prodigy device by Miltenyi in all patients. This shows you the product composition. So in all cases, we had an increased ratio of CD4 to CD8 cells. And here you see the killing efficacy. So we did this killing assays with CD19 positive tumor target cells in order to really show that those cells can kill them. They all kill. So it's actually possible to generate, to produce CAR T cells from lupus patients, not in one, but all 5 that we tried to. And then we went on to treat those patients. And as you can see here, you see again this SLEDAI score. In all patients, within 3 months, you have a dramatic improvement of the clinical picture. The kidney function is going back to normal. The double-strand DNA antibodies are gone. And then when the B cells return, this is the interesting thing, the repertoire is different. So you don't have the same B cell receptor actually distribution that you had before. And I mean, what we called this, and we named it, was reset. So it's immunological reset. Somehow by probably also using the lymphodepletion together with this deep depletion of B cells, you have something like a reset, and you get your B cells back, but in a different way than they were before. And probably you don't get your pathological B cell clone back. And this is a very interesting observation, but it's, as I said, early. But as we can say, as of to date, we can say we have 100% efficacy in our patients that we treated. No one had a severe toxicity. All of them had stable vaccination titers, and this might be also interesting in terms of the target. Maybe it's good to not target long-lived plasma cells and only target the plasma blasts. So maybe it was good that we did not have a CD38 or BCMA CAR available. And this might also, of course, open the door to a new way we treat autoimmunopathies, right? Because this has been tried now in Erlangen in systemic sclerosis, so our rheumatology department is very active. This has been tried in things that I didn't know existed before, and synthetase syndrome, although I'm co-author now on this. but the disease was not really known to me. So there are many diseases that are B-cell driven that cause all severe autoimmunopathies, and that could probably be targeted and treated with our old-fashioned CD19 CARs, and probably with the newer CARs even in a better way. So what I think I've convinced you about is that, and probably everybody else that was talking today, is that CAR T cells are really well established in cancer. But in our case here, that it's possible to generate CAR T cells in patients with autoimmunopathies, in this case lupus, that treating those patients has no issues in terms of toxicity, and we can also discuss if we have some ideas on that. That plasmablast in terms of lupus might be the population to target, because there was a long discussion I found out. I didn't know, I'm not a rheumatologist, but if you talk to people from the German Rheumatology Institute in Berlin, there's a ongoing discussion, what is a key cell? And there are many people that actually say it's the long-lived plasma cells, and this might not be true, because we don't target them and the disease goes away. Then we see this B-cell recovery without a disease recurrence, which is also important, to have this that the B-cells don't come back, but we don't see lupus coming back, which is great. And we think this temporary, very deep B-cell depletion, but also this lymphodepleting regimen, and which affects the T-cells actually mostly, might also be the reason that we have some kind of immunological reset, how we call it. And in general, I think that B-cell derived autoimmunopathies might actually be the next big thing also for pharma to address. So at this point, I thank you. So those were the four people. So Professor Schett, who is still in Erlangen, Professor Magnussen, who is still in Erlangen. Back then, Gerd Krünke and me, we were attending. So now he is the director in the Charité in Berlin for rheumatology, and I'm next to him in Magdeburg, not very far away. So thank you very much for your attention, and I'm happy to take your questions.
Thank you, Professor Mouillakakos. That was actually very impressive, to me at least, I don't know.
Thank you.
Any questions from the audience? Okay, because I have a few too.
That was great, Dimitri. My question is, okay, I need to to remember it. Sorry.
Maybe you can go to the next question.
Shall I go first and then you can? Okay, I'm sorry.
Should I start?
Please, go ahead.
Okay, so thank you, very nice presentation. Actually, I have a question on the BCR repertoire, because you showed that there is an immunological reset. I was wondering whether you monitored the clones by high-throughput sequencing, and you could see if the pathological clone that was existing before, how it was moving, or if you could still track it, or if it's completely gone.
Yeah, it's a very good question, and I could answer it if I would know which of those ones is the pathological clone. So this is a problem, that it's really difficult to identify in the bulk analysis, or the single cell analysis that we have done the pathological clone. We are working on that, so with the biomathematicians, but this was not part of this analysis. This was more a rough analysis to show the repertoire is different. But of course, tracking the single clone that causes the disease would be very cool, yes.
But even if you do not know exactly the clone, if you could see one dominant, one high dominant before predominating, and you could see changes in the repertoire, that's
yeah, this is also, yeah, I know what you mean, but this is, it's not a quantitative thing. So it's not that we can, based on the quantity of clones, say this is a dominant clone, because it was very, scattered.
Okay.
So this is, I think we have to go more looking for the
specificity.
Yeah.
Okay, great.
Thank you. So I remember my question was if you expect during B-cell recovery a recurrence of lupus, similar to what we see with B-cell malignancies after antigen loss?
I mean, this is a very good question, and we don't know, because B-cells are now back, and I mean, of course, people also say there is also a genetic, component. It's not like that you get rid of the. Normally, it should not be that you get rid of the target population. Otherwise, you would not have, this genetic component in lupus. That would totally negate it. So I'm not saying that it's gone, it's cured, but if we manage to keep them 3-4 years without any treatment, those patients, and we have already the CAR T cells produced, why not re-expose them when they have a relapse? Because the treatment in lupus patients is not fun. So all these steroids that they receive, chloroquine, all those agents, I mean, they are affecting the quality of life. So I would not even be too bothered. I mean, of course, it would be cool to have the cure for lupus, but even if it's not the cure for lupus at the end, if it's just buying them 4 years of treatment freedom, sure, would be good. Yeah.
Okay, one last question.
Dimitris, I know you're not a rheumatologist, but do you think lupus is one disease, or this works in part of lupus patients and in other don't work? Do we know which patients it works?
Yeah, I mean,
now you have 5 to 5.
Yeah, we have actually now 7 of 7 in autoimmunopathies, looking at the other 2 ones. I mean, if it's a B cell, I mean, if it's a B cell driven autoimmunopathy, I think the chances are very high, but you have to try it. So in this systemic sclerosis patient it worked, in this antisynthetase syndrome patient it worked, and I don't see a reason why it should not work. However, of course, if at the end it's another patient that, or another disease form that more relies on long-lived plasma cells, right? Maybe the CD19 CAR is not the right one then. But as a principle, it should work.
Thank you very much. This was a great session, I think. Now it's time for coffee break, and we will all be back at 11:30 for the next CAR T-cell session. Thank you.
So I took his place, so happy to see everybody here. I'm going to chair this table together with Ioanna Sakelari, who is online. Ioanna, are you there? We cannot see her.
I'd like to start because I'm going to participate in another meeting too.
We cannot hear you. Can we please turn on the volume?
Okay, let's start.
Now we can see her, but please turn on the volume, please. Hi, Ioanna.
Yes.
Okay, now we can hear you. I'm together, I'm Alex. Evgenios is not here, and so we're going to chair together this table, okay?
Okay. I'll start because I have another
yes, please do that.
Another meeting. So,
but we will start with Anna Vardi, Ioanna, okay? Because Michael is not online, we will start with Anna Vardi.
Okay.
Thank you.
It's a great pleasure to participate in this fruitful congress. I feel sorry I'm not over there because I had been already invited for a lecture in another meeting, and we'll start this very important and interesting session with the topic CAR T-cell point of care, manufacturing, and real-world evidence in Europe. And the first speaker will be Anna Vardi, who is a consultant in hematology department in George Papanikolaou Hospital, and he will tell us about CAR T cells in the real world and the real world data. It's well known that the real world data show excellent results in terms of toxicity and efficacy. And in the Hellenic experience, reaching up to 100 cellular therapies by now, commercial cellular therapies for all indications, both in lymphomas and leukemias, show very nice results, and they are exceeding up to 100 approximately 100 cellular therapies applied mostly by George Papanikolaou Hospital and Evangelismos Hospital, from Patras, Attikon, and from the Children's Hospital, and from Crete. So they are exceeding up to 100, as I told you, and the results are very nice and very efficacious. There are also 6 or 7 cellular therapies applied for multiple myeloma in the context of clinical trials. So Anna will tell us about the real-world data, both, I think, world-to-world and the Hellenic ones.
Really my pleasure to be here today. I thank the organizing committee for providing this opportunity. We'll talk about real-world data regarding the use of CAR Ts in hematologic malignancies. And we'll start... Oh, not moving. Is there something wrong? There you go. Okay. We'll start with large B-cell lymphoma. We'll quickly go through the registrational trials that led to the formal approval of 3 anti-CD19 CAR Ts in large B-cell lymphoma. I don't know. Yeah. Yeah, okay. So these are the 3 registrational trials that led to the approval of the axi-cell, the tisa-cell, and lyso-cell, ZUMA-1, Juliet, and Transcend clinical trials, respectively. The overall response rate in these 3 trials ranged from 52% to 82%, and the CR rate ranged from 40% to 54%. The PFS was nearly 40% in all 3 trials and appeared to show a plateau, indicating that this proportion of patients may ultimately have been cured. Okay, I'll try to push the right button. So, The first real-world evidence about the use of these CAR-Ts came from a retrospective cohort analysis of 70 patients with large B-cell diagnosis who received either Tisa-cell or Axi-cell as inpatients in the period between January 2018 and November 2021. This study population was at higher risk as it would be anticipated for real-world patients compared to clinical trials. So it was of higher age than in the registrational trials. There was a higher proportion of patients with advanced refractory disease. For example, patients with primary refractory large B-cell lymphoma were 62% versus 26% in the ZUMA-1 trial, respectively. The patients in the real world were more highly treated. For example, 40% of them had received more than 4 prior lines of treatment versus 21% in the Juliet trial, respectively. And practically all of the real world patients required bridging therapy, and this is an indirect marker of higher tumor burden and a more progressive disease, thereby it is considered to be an adverse prognostic factor. Nevertheless, the overall response rates and the CR rates were similar to the registrational trials, and the PFS was also similar, nearly 40%. Regarding the toxicity, grade 3 or higher CRS and ICANS occurred less frequently in the real-world study compared to the clinical trials, and this possibly reflects the earlier identification and the earlier intervention for these specific toxicities. This study revealed that LDH, more than 4 prior treatment lines, and elevated CRP at baseline were independent adverse prognostic factors for PFS. Now moving on to BALL, the pivotal study in this case was Ileana, evaluating the use of Tisa-cell in children and young adults with relapsed refractory large BALL. The overall response rate in the Ileana trial at 3 months was 81%. The 1-year overall survival was 76%, and the 1-year EFS was 50%. Later on, we had the ZUMA-3 trial, which evaluated Brexucell for adults this time with relapsed refractory BALL. The response rates were also very good, 71% overall response rate and 56% CR rates. The 3-year overall survival was nearly 50%. There's not much information from the real world regarding the use of CAR-Ts in BALL. There is a study published in 2019 with data from the CIBMTR and the Cellular Therapy Registry that overall showed similar efficacy and safety compared to the Ileana trial. But what also was shown in this real-world study was that Tisa-cell is also active in CNS disease, and this is particularly important because these patients were actually excluded from the registrational trials. Moving on now to the Greek experience regarding the use of CAR-Ts in large B-cell lymphoma and BALL, I'm going to start by presenting data already announced in 2021 at the 32nd Hellenic Hematology Congress as a plenary presentation. At that time, the analysis involved patients who had received CAR-Ts in one of the then certified CAR-T cell therapy centers. These were the Papanikolaou Hospital in Thessaloniki, Evangelismos Hospital in Athens, and Hagia Sophia Pediatric Hospital also in Athens. So they evaluated the patients who received CAR-Ts in the period between January 2020 and July 2021. 52 patients were evaluated, 42 proceeded to leukapheresis. Successful manufacturing was done for 36, and at time of data cutoff, 28 patients were infused with a CAR-T cell. The median age of the patients was 54 years old for patients with large B-cell lymphoma and 21 years old for BALL. Most lymphoma patients suffered from DLBCL, 3 patients suffered from primary mediastinal, and 2 with transformed follicular. The patients that were involved in this study were highly pretreated. In the case of lymphoma patients, the median number of prior lines of treatment was 4, and 3 for patients with BALL. Regarding the use of axi-cell or T-cell, this was quite balanced for the lymphoma patients. Of course, for BALL, everybody received T-cell according to the formal approval of the drug. The time from apheresis to product receipt was 33 days for patients with large B-cell lymphoma and 39 days for patients with BALL. Now, the time from product receipt to infusion was somewhat shorter for patients with BALL, as it should, approximately 10 days, whereas it was a bit longer for patients with large B-cell lymphoma. For these 28 patients that were infused with CAR-Ts, 18 needed bridging therapy. 14 out of 21 patients with large B-cell lymphoma and 4 out of 7 patients with BALL. In terms of toxicity now, 75% of patients developed CRS, 21% of the patients developed CRS of grade 3. Regarding ICANS, 7% of patients developed grade 3 ICANS. 57% of patients required the use of tocilizumab, 43% of patients required the use of steroids. 32% of patients required high-dose corticosteroids, and these were actually the patients that required admission to an ICU. In any case, there was no mortality secondary to CRS or ICANS. Now, moving on to different aspects of toxicity that might not be as frequently discussed when we discuss about CAR-Ts, and these are the cytopenias and the hypoglobulinemia. 89% of patients developed neutropenia of grade 3 or 4. 36% of patients developed thrombocytopenia of grade 3 or 4, and nearly half of the patients developed hypoglobulinemia. The 3-month overall response rate was quite similar to the registrational trial, so 50% for large B-cell lymphoma and 71.5% for ALL patients. The survival rates were also similar, 37% 1-year PFS for large B-cell lymphoma and 67% 1-year EFS for BALL. A multiparametric analysis revealed that LDH at baseline was an independent adverse prognostic factor, both for overall survival and for PFS. And I'll shortly present some more updated data from a single center experience, our center in Papanikolaou. This involves patients who were infused in the period between November 2019 and October 2021. These are 31 patients. As we now speak, this number has risen up to 52, including both commercial and investigational products, such as anti-BCMA CAR-Ts in the case of multiple myeloma. Anyway, at that time of the analysis, 52% of our patients received a CAR-T cell for DLBCL, 10% suffered from PABCL, 22% of patients suffered from BALL, and we also administered Brexucel for mantle cell lymphoma, and at that time we were the single center doing that. CRS occurred in 84% of our patients. We had to administer tocilizumab in 58% of patients. Low-dose steroids were required in one patient with DLBCL, whereas high-dose steroids, along with anakinra and siltuximab, were administered only in 2 patients with mantle cell lymphoma who actually required ICU hospitalization. And ICANS grade 3 occurred in a single patient with mantle cell lymphoma, so we're talking about relatively minimal toxicity. Increased baseline LDH was associated with lower PFS, and increased CRP at day 14 was associated with poor overall survival. A multiparametric analysis identified the diagnosis of mantle cell lymphoma to be an adverse prognostic factor for overall survival, and we'll see why this might be. So what do we know about the use of Brexucel for mantle cell lymphoma? The pivotal study in this case was ZUMA-2, which evaluated Brexucel for patients with relapsed refractory mantle cell lymphoma who had received 1 to 5 prior therapies, and these therapies had to include a BTK inhibitor, that is either ibrutinib or acalabrutinib. The overall response rate at 3 years was exceptional, so 91% and a 68% CR rate. These results are pretty amazing, especially if you compare them to standard of care. The PFS was also exceptional. Nevertheless, we can't clearly see a plateau in this case. In terms of toxicity now, CRS occurred in the great majority of patients, 91%. 12% developed a CRS of grade 3 and 3% developed CRS grade 4, but there was no death secondary to CRS in the study. In terms of ICANS, 63% of patients developed some sort of neurotoxicity, 22% developed ICANS of grade 3, and 9% of patients developed ICANS of grade 4, but again, there was no grade 5 event. What do we know from the real-world studies? First of all, we have data from the post-authorization safety study of the FDA-approved Brexucel, utilizing the CIBMTR registry infrastructure. These patients were treated with FDA-approved Brexucel, and this study may have included patient and disease characteristics that are broader than the scope of the ZUMA-2 eligibility criteria, including also no prior BTK inhibitor therapy. So, shortly, out of 135 patients that were included in this real-world study, 59% would not be eligible for the ZUMA-2 trial. Still, the objective response was similar to the registrational trial, 84% overall response rate and 71% CR, and the progression-free and overall survival were also similar. In terms of toxicity now, CRS grade 3 or higher occurred to 9% of patients, ICANS of grade 3 or higher occurred to 29% of patients, 69% of patients required the use of tocilizumab, and 55% of patients required the use of steroids. In the end, 5% of patients succumbed due to an infection, and 2% of patients died of neurotoxicity. Later on, and more recently, we have real-world evidence of the use of Brexucel for relapsed refractory mantle cell lymphoma coming from Europe. This is a smaller study involving only 33 patients who were infused with Brexucel. Again, we're talking about a high-risk population compared with the ZUMA-2 trial, including a higher MIPI score, a worse performance status, and in some cases, previous allogeneic stem cell transplantation. Also, we have to note that the turnaround period from apheresis to CAR-T delivery was significantly longer than in the ZUMA-2 trial, 29 versus 16 days. Still, the PFS and OS were similar to the ZUMA-2. When it comes to adverse events, only 3% of patients developed CRS of grade 3 or higher, 36% of patients developed neurotoxicity of grade 3 or higher, and 27% of patients required admission to an ICU. The study reports of a treatment-related mortality of 15%, so 5 deaths were reported, 4 from infections, and 1 from clinical deterioration in the context of prolonged steroid therapy. I'm going to compare our experience for just 5 patients that have been treated so far in Papanikolaou for mantle cell lymphoma with Brexucel. It is a small number indeed, but for us it's sort of an alert because it contrasts our prior minimal toxicity experience that we had with large B-cell lymphoma and BALL, so I'm going to comment a bit on that. We have to say that our 5 patients had higher adverse prognostic factors in any case, high tumor burden. 2 of them had bulky disease, 2 of them had a TP53 mutation, all of them had bone marrow involvement, and 4 out of 5 had the blastoid variant of mantle cell lymphoma. They were very highly pretreated, so 4 out of 5 so the median number of prior therapies was 4 for our patients, and all of them had received at least 3 prior lines of treatment. 80% of them, so 4 out of 5, were refractory to the last prior therapy. The turnaround time from apheresis to delivery in our center was 24 days. And here is toxicity. All of our patients developed CRS. 2 out of 5 developed CRS of grade 3, and 2 out of 5 developed CRS of grade 4. In terms of ICANS, 1 patient developed ICANS of grade 3. We had a death in the ICU secondary to CRS, a patient who died at day 5. In total, we report 3 fatal events. This one that I already told you about, this patient who died of CRS grade 5 at the ICU on day 5. Another patient died of disease progression on day 20, and there was another patient who died of infection at day 90 while he was in PR. For the 2 patients that are currently alive, they are both in CMR. So we wondered, because these were the first 2 out of 31 patients who were treated with CAR-T in our center who actually required admission in the ICU, and it was the first fatal event that was directly associated with a CAR-T cell treatment. Why would that be? Could it be the CAR-T cell product? Could it be Brexucell? But why would it be Brexucell? It's not much different than Axicell, it's actually the same construct. The only difference is a step that has been added in the manufacturing process where B cells, circulating B cells, are being removed because the investigators thought that they may be involved in exhausting CAR-T cells ex vivo. Could it be disease biology? So could it be mantle itself? Could it be the tumor load? And admittedly, as I told you, our patients were very, very, very progressive. Could it be prior treatments, so prior ibrutinib treatment, for example, and we know that ibrutinib is indeed immunomodulating T cells, and it may act synergistically with CAR-Ts. It is actually being explored in CLL in order to increase efficacy. We don't know, but at least for the last question, we may have some data soon coming from the cohort 3 of the ZUMA-2 trial, which will involve the same type of patients, but with no prior treatment with a beta-K inhibitor. So to summarize, real-world evidence supports efficacy of anti-CD19 CAR-Ts, which is similar to what has been observed at the registrational trials, despite including population with more adverse risk factors. Toxicity is generally lower than in the pivotal studies, and this most possibly reflects accumulating experience and early intervention. The exception in this rule might be Brexucell in the case of mantle, and we need to advocate that particularly for this, we might need to be extra cautious and refer our patients to CAR-T early. So thank you for your attention.
Thank you very much, Sana, for your that you shared with us our experience and the Hellenic experience. I think that we are now in a new era, and the new era is that we do perform cellular therapies in earlier lines of disease, and we accumulate experience in second line in large B-cell lymphomas, and we have the experience of Brexucell in acute lymphoblastic leukemia in patients above 25. And we see that we don't have such large toxicities as we had already with Brexucell in mantle cell, and we see that our results, we hopefully, our results will be better with these new indications. Are there any questions or comments, Alexandros?
Yes, Anastasia?
Thank you, Anna, for providing us all this data from the real world data. So one question, did you measure the circulating CAR-Ts after the infusion, and did you correlate them with the outcome or even with the fatal events that you saw? I mean, did you see the expanding, a huge expansion of CAR-Ts which could correlate with the adverse events that you observed in the last trial, in the last administration?
Yes, thank you for the question. Indeed, we do perform CAR-T cell measurements by flow in our center. I have to say, although in the literature it has been reported that there is a trend of association between CAR-T cell expansion and toxicity as well as efficacy, we didn't see that in our data.
Thank you, Anna. Do you think that it might be the problem with this product is the ratio that you have, CD4-CD8 ratio? This is fixed. I don't know how fixed it is. And do
you
think
mean about
maybe,
Brexucell?
Yeah. And do you think maybe the CD4 portion is too high?
I honestly think it's the tumor burden because, as my director mentioned earlier, we now have experience from Brexucell in ALL, which underwent CAR-T therapy in an MRD negative status, and we had no toxicity at all. We also had a better outcome and a better toxicity profile in patients with mantle that didn't have so extensive tumor burden. So really, I think this is the main attributing cause to toxicity. It may be, let's say, a false effect due to the fact that when we received the indication, we started with patients that most urgently needed the treatment and had no other treatment option. So by default, our first patients would be those with exceptionally high tumor burden. So maybe what we saw in terms of toxicity was related to that.
To combine the endothelial injury of ibrutinib in such patients, because all the patients that had ibrutinib and the washout period of 3 or 5 days before conditioning, I think, they were under ibrutinib, it would be a crucial factor for them. We'll see by accumulation of our data. We don't have so many data on mantle cell lymphoma by now.
Indeed, it remains to be seen. Maybe the cohort 3 of the ZUMA-2 trial will also provide data regarding that.
And in our center, we are investigating the whole thing by our research. Okay, are there any other questions?
Okay, I have a comment before we go to the next speaker. It's, as a clinician, I find, and I'm making an appel, I find unacceptable from one company, the Kite, that he doesn't give us the certificate of analysis what we're giving. It's a living drug, and we don't know what we're giving. But if you ask from other companies, they give it. So we know what we're giving. That's why maybe thank you, Anna. The future will be, we go to the next speaker. Thank you so much. Because we are clinicians, but we have a lot of experience with also producing cellular therapies or giving cellular therapies. And maybe the future, and this is the theme of this table, is the point of care, that we're not only clinicians, but we'll also produce the cellular products that we're giving, which now are called drugs. And for that, we heard yesterday also we need facilities and all these things. And I'm very happy to introduce our next speaker, Professor Michael Schmidt. Everybody who is in Europe and all over the world, and especially in Germany, knows the amazing work they've done in Heidelberg. And he tells us about the program in Germany of point-of-care CAR T-cell manufacturing. Professor Schmidt, Michael, very good friend of mine. Thank you for being with us today and give you the...
Yeah, thank you very much for your kind introduction. I hope you can hear me. Is that correct?
Yes, thank you so much. Everything okay?
I would need to share my slides with you, but this is obviously not possible here. So I asked the administrator to give me the right to share my screen with you. Okay. Do you see my slide now?
Not yet. Please, can you fix this? Now it's okay. Thank you.
I can see it.
Yes.
Now it's okay? Yeah? Okay. So first of all, I would like to thank the organizers for inviting me to give a presentation here at your exciting 6th Congress of the Hellenic Gene Therapy Society. And I have to apologize that due to sudden private reasons, I could not come to Athens, what I really regret. And I hope I can come another time to meet, especially Anastasia Papadopoulos and Evangelia Yanaki, who very kindly invited me. So with this slide, I want to share our experience here in Heidelberg for the last 5 years of CAR T-cell therapy. You see the number of patients treated climbing up to more than 160, and the black line indicates the total number, and you have a number of other lines with other colors encoding for Yescarta products, Kymriah, Tecartus, Abecma, Breyanzi, so all what we gave, but I would like to guide you to this blue line and the red line, which indicate that our homegrown point of care HCK1 product is making a big contribution to the total program. So you see the product we use right from the beginning, Yescarta and our own production are not so far away from each other. And that indicates that by point of care treatment, you can improve the number of patients you can treat at your center. So our Heidelberg HT is what you have on the number plate, and trial with 3rd generation CD19 targeting CARs is a trial which is completely academic. Very kindly, we could obtain the vector, which is here depicted with the CD28 plus 41BB as co-stimulatory molecules from Professor Malcolm Brenner at the Baylor College of Medicine, Houston, Texas, USA. And we do the leukapheresis here under the auspices of my wife, Anita Schmidt, who's a professor for transfusion medicine. Then you see the technicians working here in a very handy fashion on the cell production in our GMP core facility, and Peter Traeger here with some fellows at the ICU. We're doing cram rounds for patients treated here. Peter is our director of the stem cell program, including the CAR T-cell therapy. So the entities we are targeting are acute lymphoblastic leukemia in adults. We have not opened pediatric part of our study. Chronic lymphatic leukemia is a very strong point. I'll show you some slides in a couple of minutes, but also other non-Hodgkin's lymphoma, like diffuse large B-cell lymphoma, follicular lymphoma, mantle cell lymphoma were included. The patients received a standard lymphodepletion therapy with fludarabine and cyclophosphamide over 3 days, and we initiated the study in autumn in 2018. And you see here our stratum 1, which is underlaid by blue. This is all the ALL patients. So if you see for prior treatments, there are numerous treatments, and almost all patients underwent allogeneic stem cell transplantation before they were treated with CAR T cells. Also, very frequently, they obtain either monoclonal antibodies like blinatumomab or inotuzumab against CD19, CD22, and TKIs. For the stratum 2, which includes a number of patients with non-Hodgkin's lymphoma, The pre-treatment of allogeneic stem cell transplantation up to 2 times is a little bit more scarce, but you see here the standard treatments which are obtained, and you see that they are heavily pre-treated patients. And if you come to a brief summary, we could achieve an overall response of 84% with CR here, and with MRD plus CR in another 30%, and we had 8% stable disease, 8% progression as a best response for the non-Hodgkin's lymphoma, including CLL. You see an overall response of 50%, 44 complete remission, 6% partial remission, 28 as stable disease, and 22 progressive disease. Here I come with some PET CT scans and MRIs. This is a young lady who was referred to us after a relapse from ALL after 2 allo transplantations, blinatumomab, and a number of TKIs. You see here lesions which completely disappeared. Also lesions on her legs and feet disappeared over the time of treatment, and a similarly impressive response was seen into MCL, mantle cell lymphoma patients here in a patient after R-CHOP, RDHAP, autotransplantation, iprotinib, rituximab plus Revlimid. You see vast cervical lymph node packages which completely disappeared after the treatment on day 13. And this man with a retro, a bulbar tumor, was completely leading to a vision loss of vision at his left eye, completely recovered very quickly after transplantation and fully after 3 months. So that was a very big success. He's now driving several hours his car to the grandchildren and is reading different newspapers every day. Also, he received 2 allogeneic and 1 autologous stem cell transplantation for his mantle cell lymphoma before he was dosed in with our product. So what about overall survival and progression survival in ALL patients, the red curves, and NHL, including CLL patients, the blue curves? You see after day 90, when we have end of study, we have almost 90% patients surviving, and ALL patients 75% surviving. Progression survival is a little less. This is about 72% for the ALL cohort and 68 for the NHL CLL cohort. But very favorable results in this highly pre-treated cohort. Then Luisa Schubert, who was very initiative in that study, was calculating here for the same thing, the Kaplan-Meier curves for survival, overall survival, progression-free survival for almost 3 years. Then you see that the overall survival for NHL patients was 60% and 40% for ALL patients, that we had a 45% survival after 3 years for NHL CLL patients. As for freedom for progression after 3 years, and 8 ALL patients come to a level of 24 patients still being progression-free after 3 years. This is enormous, because you would think usually by day 200 all patients would not only have progressed without treatment, but also died from the underlying disease. So when you compare this with what has published as a 5-year follow-up within the ZUMA 1 trial, you see that it's very nice what we can achieve. This is overall survival here, 60% for NHL, and it's 50% here for the patients in the SUMA-1 trial. As for the progression-free survival, we can reach 40% with our product, and this here is 35. So dose matters also in CAR T-cell business. You have here a spider graph, which is time plotted against the copy number per microgram DNA in the mononuclear cells of the peripheral blood. And the dose levels with 1 million, 5 million, 10 million, 50 million have a clear dose-dependent persistence of the CAR T cells in the peripheral blood. So you see green, the higher dosage levels are also the curves which are still up at day 90, while the other curves rather go quicker down. So green is the number, the color of hope. So Luisa and Felix Correll plotted this again, dose level and survival, and progression-free survival, overall survival, progression-free survival. And you see for both the ALL lines in red and the NHL lines in blue, that you have a clear dose dependency in response. The real take-home message is that we didn't see ICANS in all patients. This is just a list of the ALL patients, but also in NHL patients, no ICANS, no neurotoxicity at all, and CRS was scarce, and if it occurred at a low level, grade 1 or 2. So let's jump from ALL to CLL. We included patients with early relapse within 2 years after end of chemoimmunotherapy or chemoimmunotherapy refractoriness plus failure or intolerance to both Bruton tyrosine kinase or BCL2 inhibitors, or if relapse occurred after allogeneic stem cell transplantation, all patients were ineligible for or refractory to standard interventions like DLIs, antibodies, or chemoimmunotherapy. And Patrick Dirks from our group, he summarized that and presented that recently at the EBMT CAR meeting. You have here heavily pretreated patients up to 10 pretreatments. Almost all patients had a P53 abnormality, which means they were at high risk. And you see here with increasing dose level, we didn't see a response with dose level 1 or 2, but dose level 5 resulted in a very fine number of complete remissions with MRD negativity, and that persisted here up to 650 days. So that we think that, especially for CLL patients where we have no authorized CAR T-cell product in the market yet, we can really add with our own homegrown CAR T-cell product something substantial to treatment of these patients. Of course, we wanted to know what is behind that, how can we distinguish responders from non-responders. So we took blood and analyzed together with the group around Simon Haas at the Berlin Institute of Health, especially Cheyenne Yusufian and Dominic Fonfisch, in a high-resolution immunophenotyping by flow cytometry, the different immunocells, and put a focus particularly on CD8 and CD4 positive T cells. And we saw that the pattern for CD8 cells in non-responders, so yellow is a high frequency, is completely distinct from the pattern in responders. So while in responders you find functional effector states, in non-responder you rather see non-activated T cells or dysfunctional effector states of T cells. So as we recently filed a patent application, I can all today share with you the full signature of these cells. You see here the responders are lacking CD39. This is how they are characterized, and the same is true here for the responders. Again, the density, the high frequency is in CD39 effector memory like T cells for the CD4 compartment on the right, and also on the left in the responders for CD8 plus T cells. CD39 is a molecule which is responsible for expulsion of T cells. So for response and endurance of response, it's very important that you have in your product and later on in the peripheral blood of the patients these very vivid and active T cells. So this is Simon Haas again. In a very interesting vector plot, he could show that in an ALL patient who had a very long response, the pattern of these responders in terms of NK cells, in terms of CD4, CD8 cells, gamma delta T cells, is very, this is the lilac ones, the violet ones, the bubbles here, is very resembling what we see in burgundy red healthy donors, while non-responders have a completely different pattern in this vector graph where you have monocytes, non-classical monocytes, intermediate monocytes who do not contribute to the T cell response against leukemia. Let me conclude. CAR T cells manufacturing at site, point of care, was feasible for all included patients. This is a very important message, and we are very proud of this because we see in the interaction with Big Pharma that they have a lot of out of specification events, up to 30%, and we have here a success of 100%. That certainly reflects this manufacturing by hand. It's like making pottery or like making nice suits on a 1:1 ratio by hand manufacture versus industrial production on the assembly line. So I could demonstrate the excellent safety profile of third generation CAR T cells. Its absence of neurotoxicity, its only low cytokine release syndrome, and the responses were at least as good as we see with the commercial products on the market, if not superior. And clearly, we saw a dose-dependent response to our treatment. So we are preparing additional, in amendment, additional 20 CLL patients and also patients with primary central nervous system lymphoma, and we hope that we can open that in October this year. So, of course, making things by hand is good, but you might also want to produce more products within a time unit. That's why we proceeded and purchased two 3D Max Prodigies from Miltini company here by Cologne in Germany. We established for another CAR against lymphoma and myeloma, BCMA, in a cooperation with colleagues from the Max Dalbrück Center, a BCM CAR production. We were inspected by the Paul-Ehrlich Institute and obtained a manufacturing license in September last year. We will soon open the Carlotta trial with Martin Bernke from NCT Dresden in the summer this year for both myeloma and lymphoma patients. And as I already alluded to in my intro, I'm very indebted to Anastasia and Evangelia, who wanted me to bring this time to Athens, but I will come certainly later this year. And we have already established a cooperation with their Gene and Cell Therapy Center for the production of CD19 third generation CARs in Thessaloniki. And Evangelia has also a very strong background. She was several years with Malcolm Brenner. So I think we are on a safe ground here. I would like to thank all the people here from the Cell Therapy Center. There are 6 professors. I mentioned Peter Traeger, Casimir Tito, Udo Hegenbart, and Stefan Schöland with a strong background in amyloidosis, Alex Radikovic and Thomas Luft for MDS, the coordinators, the case managers, the nurses, the doctors on the ward, the stem cell team here, making the Lucas product for further production. This is my GMP research team with Luisa and with Felix. I mentioned already Luisa and Felix. I showed you Anita, who's our transfusion specialist, head of production, and Angela Hügelhofen, who is our head of quality control. These are the nurses of our apheresis team. I thank you very much for your kind attention. I would also cordially invite you for our 7th cell therapy symposium. You see, we are one number ahead of you. 5 years of CAR T-cell therapy in Heidelberg at the beginning of November. Here, come to our lovely city with the old castle, the old bridge, and the Holy Ghost Church here, and enjoy with us. Thank you very much.
Very nice. Yes. Thank you very much. You are very welcome even in George Papanikolaou Hospital, because we are grateful to your help in manufacturing all these cells, and we see that you have excellent results even in CLL, that it's very pioneering, I think. I don't know if it's the quality of your cells that
we smile with them very nicely. Yes.
Yes. We are working very hard to launching a clinical trial to manufacture our institute cells in George Papanikolaou Hospital, and we'll see that we'll have very good results as of your own experience. Are there any comments or questions?
Thank you, Anna. Kat's here because I see here the audience.
Questions. Which is the best quality of the cells that give us the best results? Can you give us an answer, or we are trying to these best results? Do you think that
there is a clinical answer to this and a laboratory answer to this? Yes. We all know from our own and the commercial products that, of course, you should treat patients as early as possible and not in the 11th line.
It's the selections of patients.
Yeah, so the selection of patients, and that's what we actually see. We started with a indication for Jessicata, for example, and Kymriah, 3rd line for diffuse large B-cell lymphoma, and now it's advancing to the 2nd line, and the same will be effective for myeloma and BCMA, targeting CAR T cells. Moreover, there are some do's and don'ts. There are good drugs and bad drugs before you bring the patient to leukapheresis. So you should avoid, for example, bendamustine. Yeah,
yes, it's our clinical practice for that.
Yeah,
but I think that doesn't answer all the questions we have. But I think that the quality of the product would be an answer.
Well, that's what I said. There is, I mean, it's like cooking. Yeah, if you go to the market this morning and you buy a nice fish or you buy a nice meat, and you go to somebody who sells you only mediocre quality of fish and meat, then you can be a world-renowned cook, but at the end, you will not make a delicious meal. So try to get the best T cells you can get out of the patient, and as all of you know, there are tremendous efforts to go away from autologous to allogeneic, to point of the care T cells or even other immune cells like natural killer cells, to transduce them with the CARs and employ these products. So this is a clinical thing. The other thing is, for example, that as I showed you, you have some markers which are of positive prognostic value and others of negative prognostic value. So you should make efforts in your culture to improve, for example, the cytokine mix, or to do also a second genetic maneuver besides transduction with CAR, that you, for example, knock out nauseous genes like this CD39, which is just making the T cells weak and sleepy.
Okay, because of the time, we have some questions here from the audience. Professor Miyakakos.
Yeah, this is Miyakakos from Magdeburg, so we know each other. Too bad that you're not in Greece today. I have a question actually regarding CD39. I mean, CD39 works together with 73, right, to make ATP to ADP to then to cyclic AMP. Did you see whether 73 is co-expressed on those cells, and do you think it makes sense to go for, you know, an adenosine driven approach to block? I mean, there are actually antibodies that block because it's also expressed on cancer cells, for example 73, to really interfere with this ATP ADP AMP cascade to improve CAR efficacy.
You can, thank you for that interesting question. You can see here on the poster behind me Otto Meyerhoff's name. So here on the campus 100 years ago, Meyerhoff and co-workers discovered ATP, so our focus is on that. And we are in the middle of analyzing the CD39 interactions and pathways in CAR T cell products and also in the blood, peripheral and bone marrow blood of our patients. So these are ongoing research experiments, and hopefully in the next meeting I can give you a good answer.
Yeah, thank you.
Okay, thank you, Professor Smith. Thank you for expanding your program also in Greece. It's excellent. Before we go to the next speaker, I have one question. So you make this point of care production, we have now a product. How are you planning to bring it to the market authorization as a product which will then be not point of care, or as a process point of care in the hospitals? I'm wondering.
Is that still a question to me or already to
It's actually a question to you, because you have a point of care, very nice product. And if I wanted tomorrow to have it in my hospital, are you how you bring it to the market as a market authorization? Will you have it as a product to give it and will be decentralized, or you will produce it in your facility, or you want that all hospitals have their point of care production of the product that you created, these excellent results that you tell me? So I'm trying to see the future, where it goes.
Thank you, Yanis, for this very important question. So we had a long discussion, first in our clinic and later on with the regulatory body, the FDA equivalent, the Paul Ehrlich Institute. You can go two ways. One way Manel will describe in his talk is to really go to the EMA like a big pharma and ask for marketing authorization. This is not our way we will pursue, because we think that this is from a German perspective too costly. We would have to provide, according to what me was told from the Committee of Advanced Therapies, the CAT at EMA, you have to provide evidence that you can deliver your product to 27 countries in Europe. And we can simply not do it because we don't have the financial and logistic power for that. It's not our purpose. We want to give best care to patients who come to our institution. And we think we have the best product, which is now available as for CD19 CARs, but we would not extend that. So what we just filed yesterday is an application to the Paul Ehrlich Institute for a hospital exemption, according to German drug law, Drug Act, paragraph 4b. So that is what we are saying. They will exempt us from our obligation to bring to go to a market authorization, but they will give us a hospital exemption to produce the car here, and we have a positive signal in a written form, even from the health insurance, which is covering most of the people here in Germany. So that's what we call an AOK, the general health insurance for employees here in Germany.
So that means if a hospital wants to join your program, we have to make collaboration, tech transfer, process development on site in the hospital exemption on site, with a goal to give it to the patients, but I know first how it will be funded, because if it's hospital exemption, it must be funded somewhere. It's not funded from the insurances. So there are still some issues there. It's very nice, the academic and the tech transfer and all these things, but we bring it to the patients, but at the end, I don't know how we can do that. Let's see what happens.
Yeah, we can discuss next time. I showed you it was 5 years of a wait, and since 3 years we are negotiating with the healthcare payers. They are here in Germany ready to do that, to give us money for the product on the basis of a hospital exemption.
Okay.
So you need some investment for the first years, and after that you will have some remuneration. But it will help you for your entire GMP core facility to develop new products, fancy products, which are designed from your own spirit, from your own period, people on the side point of care.
Okay, I hope next year we hear about the HD CAR2 product and CAR3 product in this meeting. And thank you so much. So we have to continue, and indeed, one great success story was from Spain, as already Professor Schmidt said, where they started the point of care and they went the way that Professor Schmidt said through the EMA and the marketing authorization. The case of CAR-RE001 development clinical data will present it from Manuel Juan. He is online. Thank you so much for being with us today.
Thank you. So thank you very much for the presentation. I think you are able to hear me and everything is working. So thank you, organizer, for inviting me, and I will try to explain our experience that started some time ago. I'm Manuel Juan, I'm the head of immunology department, and now I'm responsible of the production. And this is my disclosure that can be summarized in the fact that even I don't have personal profit for these developments and my work in CAR T cells. Obviously, I have a bias when I talk about this proposal because I'm very involved. I think the concept that we have for the CAR T cells, also for in general for the immunotherapy, is the fact that the immunotherapy is just another kind of treatment that are based in two elements: the production of a product, the clinical care of the patient. I think both are very important, and the control of this data that can be summarized here like a pharmacology of this data. It's necessary to develop good clinical practice and good manufacturing practice in two different situations. One is the option to use products for industry, but also to have infrastructure to make the possibility to obtain or to have this treatment. So basic research and clinical research are very important. When we start our small history of CAR-T development, our aim was just to treat our patients, to give the best treatment as possible for our patient. And by this reason, after trying to look the first people involved in this CAR-T treatment, we decided to develop our own CAR-T cell based in an antibody that was developed in our lab, in immunology lab, more than 30 years ago. It's a CAR-19 because we started developing a CAR-19, and after having the possibility to be in Pennsylvania, in the University of Pennsylvania with Dr. Kaljun, we obtained several research projects, and these grants allows us to develop the preclinical steps of our CAR-19. It's a second-generation CAR product for having the first data that were developed mainly for two postdoctoral students, Dr. Boronat and Dr. Castellan, both with a very brilliant career in the first steps of postdoctoral training, but they decided that even probably our development will be not so important for research to obtain big and great paper. They decided to make the effort to try to make this for the patient. So anyway, this data allows us to make a point of care proposal based in a clinical trial. We will have the first economical support by a grant of the Spanish government. And this clinical trial was not enough with this support, and we obtained the support of a crowdfunding project that allows us to propose a point of care development also based in this machine of Miltenyi Biotech. And we define the clinical trial steps, and we start to treat patients now 6 years ago in this clinical trial that was approved by the Spanish Drug Agency. At this clinical trial and the work of more than 200 different people, professionals in the hospital, allows us to arrive to have some results that was presented to our Spanish Drug Agency, and we obtained the the approval for the use of this drug in acute lymphocytic leukemia, lymphoblastic leukemia in adult patients. Now, over 25 years, it was not a covered proposal. Even the clinical trial include along this time other diseases, CD19 positive. In any way, by this system, by this situation, in some way we arrive to fulfill, to arrive to have success in our objective, because under hospital extension we can have the reimbursement by the public health system, and this allows us to treat our patients as we proposed at the beginning. But it was clear for us in 2021 when it was approved that this was not enough, because there are other patients and other people in the world that could be benefit of this treatment. And this is the reason why we decide to find national and international partnership to extend the use of our product. Our hospital is just in the middle of the city of Barcelona, in the crowded place in the center of the city, next to the university, the School of Medicine, and next to an important research institute that make this very important. But it's also interesting that we have a very near collaboration with the Hospital Sant Joan de Déu, that is the pediatric hospital. And in the last month, we arrived to have an agreement with a non-profit company that it's based in the northeast part of the city, that allows us to produce the lentivirus that we use for this development. So our hospital is this structure with this U shape. In the middle is the School of Medicine. It's where we produce the cells, And we have here, very near, another building where is the Research Institute, and in the middle is the University of Barcelona, where we produce at the beginning the lentivirus for the treatment. So, making different collaboration between our hospital, pediatric hospital, the Research Institute, and this technological structure, institution like that, we arrive to make a structure that allows us to make an agreement or to develop next to our public health system in Catalonia, CatSalut, also in Spain, through the research network like TERAP and Certera, trying to find also national and international partnership. We want to arrive to share and to have the opportunity to use our product in a public point of view. This public distribution, probably it's quite clear in Spain, but not so clear outside Spain. And this is the reason why we are making some collaboration with pharma companies in different situations where it's not clear that this public development was possible for the product. So our hospital is here, as we see, and this is the U shape in red of our hospital, and in one of the corners we have the clean rooms where we produce the cells, and in the School of Medicine along the last 5 years we have been producing also the lentivirus and the GMP condition. But the production of the lentivirus and the GMP condition, it was also approved by the Spanish Drug Agency, allows us to treat between 15 and 20 patients each round of production, that it's for 20 patients, for 20 bags, so it's not a lot. This is the reason why we look for a bigger place and to try to make an escalation that we are doing in other things. The clean room for the cells are in the hospital, and we have several rooms, and the big one is this one where we have here the different bioreactors that we use, not only the Prodigy for Miltenyi, but also now we have 2 cocoons, and we have 2 additional Prodigy in other small clean room that is focused in the development of products for HIV patients or infected patients that allows us to make more control at this production. In any way, this is our installation, this is our clean room where we produce our car. Next to this hospital for adults, we have the pediatric hospital where we expect to have additional clean room for production not only of immunotherapy of CAR T cells and even also TILs, but also we want to arrive to make gene therapy in this structure that it's almost concluded. We expect to have the delivery of the installation of the facility at the end of June and start here our work. And the other place that I want to present is where we produce now the lentivirus. It's in this part of the northeast of Barcelona. We have 2 different clean rooms that has been qualified also by the Spanish Drug Agency, and this is the place we are doing this. In this, with all this infrastructure, we have been able to develop the first product, the CAR-19, that is here, that it's called Barnin-Captegene autoleucel or Barnin cell, that is based in our antibody, and after develop this in a Prodigy, we obtained the RE1 product that we call this. We have several results already published. This is the publication when you can look for it, but making a short summary of the data, you can see this table when we have the main data of the first patients treated, where you can find data about progression-free survival or overall survival for pediatric kids, where we don't have the approval now in this situation because the hospital exemption is also possible for products and indications that are not covered by the commercial one. But we have a very good results, and we are trying to improve this data, trying to arrive to centralized authorization, and later you can see here the data for adults that allows us to have the hospital exemption. Making a summary of the different treatment in these adult patients, we can say that more or less the data are very similar to the one published in the literature. Even for extramedullary disease that it's not covered, we have very good data also of efficacy. You can see here several patients with extramedullary disease, and also in CLL with our product that allows us to make a collaboration with people in Netherlands trying to develop a product specific for CLL, where the data are very promising for all the patients. In fact, one of the first patients treated with our CAR-19 was treated now in 2017. So now more than 6 years of survival of these patients. And you can see here how this works. This is the Spanish document where allows us to have a use authorization for the use of the product. And this is a formal way where under hospital exemption rule, this specific approval for our hospital, we are treating now our patients. It's the first ATMP, is the first product to be authorized through the hospital exemption for cancer in Europe. There are other products authorized for other purpose, but cancer was the first one in 2021. It was authorized for, it was the first authorization for adult ALL and the first public CAR product to be authorized. And this is a small timeline when you can see for the beginning to now where we arrive to have this approval for the hospital exemption rule. And now we are involved in a prime designation trying to obtain the centralized authorization because even because here the question, the previous question to Professor Smith, we think that it's possible to develop or to have a centralized authorization based in point of care development. And this is the reason because we are absolutely convinced that it's possible to have the product in point of care and have a centralized authorization. Why we are trying to obtain this prime designation. In any way, this prime designation was also the first for an academic product. Several other proposals for academic tried to be approved, but before this time, in June 21, there was no one approved, but later we obtained this in December of 21, this prime designation that now is under development with the European Drug Agency to try to obtain this centralized authorization based in the concept of point of care. Next to the RE1, we have also developed a BCMA like the group of Heidelberg. Also, several years ago, we start. This is the second product. We also have a normalized name. This Tesnifel is based in another antibody. Sorry, here you can see CD19. It's not CD19. It's BCMA, anti-BCMA. And this third generation lentivirus allows to produce this RE1 humanized product. And the data was developed not only by our center, because it is the first attempt to produce in 2 different centers in Spain, in Barcelona, and also in Clínica Universitaria de Navarra, in Pamplona. And we produced this for, at the beginning, 6 centers, now 2 additional ones. And we have already presented the data of 60 patients and trying to obtain the hospital exemption for all these centers. And we want to know this, if this is possible, but at least it's not impossible for the rules that we have in Spain of this. And later, if we have this approval, probably we expect to have the approval along this month or maybe after the summer, because it's times for the drug agency in Spain. We want also to try to follow the pathway to arrive to have the centralized authorization. The results of the first 30 patients, we have additional 30 patients that will be published next week in Lancet Oncology. That allows us to say that our current BCMA permit to arrive to have a 100% overall response now in 60 patients, and in general, the best response arrive at day 100 with 92% of MRD negative situation. But we think it's a good product, and it's important to have in mind that in a difference of other countries like Germany, in Spain, although the two products for CAR-BCMA are authorized by the EMA, are not available for the patients in Spain and other countries in Europe. And this is the reason why we think we need to have this authorization for 3 patients in Spain, because they are not an option for treatment for these multiple myeloma patients. These are the different graphs of overall survival, progression-free survival, of the first 30 patients, and that are also growing. There are other projects in consortium with different hospitals in Spain. We are trying to obtain the authorization, even I don't know why, how we will be arrived for pediatric indication, because also we are convinced that the product is very good. We have a collaboration with India where through the foreign offices, they arrived to ask us an agreement with India for our product based in a company, because it was not possible to make this in the public system of the India. So we have this agreement. We have also a company for CLL with CellPoint, now Galapagos. We have a very interesting agreement on developing of our RA1 in lymphoma that is making a comparison with our varicella and axicell, and we are in this situation. There are people treated in all these proposals. In Monil, India, more than 20 people have been already treated. CellPoint has also started the clinical trial in several patients has been treated, and Novo also have been treated. And we have an agreement also with a public company in Spain that is called Garaire in Galicia, in a region in the west part of Spain. And we are developing a dual CAR based in the combination of CAR-19 and BCMA that we call RE3. This RE3 is just the co-transduction of both lentivirus. And we have several different proposals, not only 19, BCMA, we have CD7, another, and the pipeline, even in solid tumors, that we expect to start this year for R2. Probably next year for IL-13 RI2, and also even for rejection, we have a very good results for this CAR. I think it's very important the fact that in Spain we have this proposal for the government that it's called Plan de Abordaje. It's a way to arrive to introduce the advanced therapies in our national system of health, and it's based in the concept that both commercial and academic products should be acceptable for treat our patients. Even there is several proposals where specifically advanced therapy is presented. This is in Spanish because there are Spanish documents. So our proposal is to have a network, not only in Spain, but also in Europe with different centers trying to make possible this point of care treatment based in whatever was the process, having in mind that if we change the process, the product is different. And in summary, we can say that 240 production has been already done in our center. In fact, 246 now. And the number of failure is very similar to the published. We are in the middle to other clinical trials. And we are almost convinced that this scheme where all the development is based in the academy for CAR T cells and later pharma is the way to distribute the product or to commercialize or to make available the product can be changed. And we think it's possible to develop and to make available also the product in the academy. This is the conventional scheme of how the products are arrive to market authorization. In general, academy start the concept and the clinical trials even, and later the pharma conclude all the processes. With the hospital exemption, we are able to make all these things just in the academy, but the use is limited. So we think that it's possible to arrive to market authorization, making the first step with hospital exemption approval by the local agency in the academy, and later to ask this prime designation or whatever was the system that was proposed by the EMA to arrive to a final use and market use, and at least in European countries. It's possible that in other countries, in other world countries, we will need pharma partnership because it's not possible also. But we are almost convinced that this scheme is possible, start in the academy and to finish in the academy, especially for some products where will never be developed by the industry, because the industry need to have a number of patients enough to be treated to have a clear option. And CAR, it's a future treatment based in the specificity, and this specificity reduce the number of patients. So specifically or especially pediatric patients can be very difficult that the industry, the companies develop products for this small number of patients. So in conclusion, we think the academic CAR is possible, requires a change of mentality, requires help for experts in regulation, and requires also some stamina, because big pharma doesn't happy with this situation, but they dislike the hospitalization clause, but there is there, and it's a possibility, and we think this is possible if we are flexible in the approval. So in our proposal, national and international partnership and collaboration, it's important. The last slide is just to say that in our case, more than 200 professionals, especially oncology, immunologists, people in research, of course, but also very important pharmacovigilance. For pharmacology is very important, but there are too many other departments, and being also very important hospital direction of the centers. And you can see here a picture with people in hematology, Julio Delgado for adult patients, next to Dr. Alvaro Susana Rivas for pediatric patients, people for hematology, people in production in immunology, and very important for, in our case, the support of the direction of the hospital. This is the medical director of the hospital that make us possible to develop all this next to a lot of different people that are not in the picture. So thank you very much for your time, and thank you for the possibility to present our data. Thank you.
Thank you so much for this excellent journey that you showed us, and you paved the way for this point of care. I have two questions, if nobody from this audience. First, my question first, then Dr. Yanaki, is I didn't understand at the end. Now you have a product, you go through EMA, you started as point of care hospital exemption. At the end, this product will be centralized produced and you sell it as a drug, or you want it at the end through the EMA to have the process tech transfer?
We want to validate the processing of point of care production. It's clear that because at the beginning we have been discussing about the possibility to produce the lentivirus in different places, but it's not possible. Even I don't understand why the lentivirus is an important concept for the point of view. For me, it's just an starting material. Anyway, the lentivirus should be centralized, produced in centralized way, but our proposal is to produce the product under agreement with all the centers, under the control of the data by our hospital, who will be the one that will be in contact with the EMA, but produced by the centers that want to be our partnership in this. We will never make a centralized production because we are not a company. We are a hospital, sorry, because I think it's very important to puntualize this. We don't want to go to any place to make this to others. Have in mind that we make all these things for treat our patient. Later is the fact that other people ask us if it's possible to use our product in the different places. And this is the reason why we are moving to this proposal in the EMA. But the proposal is basically to have authorization for production in several places and use in several places, but not a centralized production.
And if it's possible, another question.
Sorry, just one thing. It could be possible also because it's possible to produce for some places where it's not possible to produce the product, that they ask the product to us and we can send this product to these places. But this is not our aim. This will be the exception for our point of view. Sorry.
And to this point, I saw that for your process, you are using some closed system like Prodigy. Now you use also Cocoon. I mean, in this process, the tech transfer, how important is to use this closed system? Why don't use open systems? And if it's possible, if you validate again with open system, because now you are also a little bit with companies very tight and let's say.
I think I didn't explain well because when we change the procedure of production, just to changing the bioreactor or changing in an open system, finally the products are not the same. So it's very important that for each product we have one way to produce in all the places where the product will be used. So the tech transfer will be to follow exactly the same procedure that we have in the different places, and to have an agreement where the center should compromise to make the things at the same way that is done now in Barcelona and also in Clínica Universitaria de Navarra. It will be necessary to make cross-validation between the centers for the same starting material and to demonstrate that it's possible. So it's very important, finally, if this can be an option, to make in the same way, under the same rules, under the same protocols, the product. And finally, we will be, as a hospital, if we have the approval, we will be the responsibles of the product. But we need to have good collaborators. This is the reason why we don't want to sell the product, the concepts or the materials or all these things. We want to have collaborators, people who want to be part of our proposal.
Dr. Giannaki from George Papanikolaou Hospital.
That was great, Dr. Juan. I would like to congratulate you as your approach represents a model in Europe for many academic centers that look forward to in-house production of CAR T cells, including ours, as you probably heard from Dr. Smith earlier. Although it was a bit scary, I mean, this long path to materialize the in-house production, you showed us that it took you 30 years, almost, yeah.
No, it was the production of the antibody, but we start the development of the CAR in 2013, so now in 10 years. But
yeah, okay.
The clinical trial arrived in the last 6 years.
Yeah. What I would like to ask, maybe it's a weird question, I don't know, but we are concerned on this towards the same development from our side. How did you ensure funding for all this approach? And actually you presented a huge amount of cell products that you generated already, and there are too many to come, as I saw. Thank you.
The cost of the production is covered now under the hospital exemption rules or by the payment of compassionate use in some patients. So when we start the clinical trial, we should have the money for develop the clinical trial, and we obtain this money first with a small grant. Now we have bigger grants in the Spanish government, but we are moving around 1 million euros. That is it's peanuts in comparison with the grants or the budgets of the commercial clinical trial. So with these 1-2 million euros, we are able to develop the 60, 30, 40 patients treatment. and later present this data to the drug agency. For us, it's very important to have the hospital extension approval, because after that, this can be added to the list of products that are reimbursed by the health system. So we start with grants, with small quantity of grants, because this is not a company. And we are right to have data, and later we try to make the hospital exemption rule to have some way to have additional cover of the product, and later when we arrive to the final step, that will be the authorization for the use, or market authorization for the EMA, we need to find a way to cover all the expenses of the production. Now our product, it's evaluated as 89,000 euros in Spain, but in other countries the cost can be higher and maybe lower, because we are not a no-profit. We are a no-profit element, and we are looking to cover the cost of the production next some small quantity, having in mind that some of the products fail in the production, or we need to make additional things that is just covered by this. But now we have the price in Spain for our product. It's well defined by our public health system. It's 89,000 euros.
So, thank you very much. I think we'll end here this exciting roundtable. We show the way where we're going, where the most of us hematologists, and we are now going to produce also ourselves. We need a lot of patience, but I end with what you said at the end. We need also a lot of stamina to conquer the big pharmas. I think this is another thing that's new for us, but I think at the end we'll find a way. Thank you so much, and every speaker and everybody here.
Did you see the pictures?
Okay. Hi. Okay. Welcome to this exciting session on new tools and developments. It doesn't show in their current affiliation. You stay downstairs. It doesn't show in their current affiliation, but it almost turned into a TJET session. This one, as it turns out. So it's my great pleasure to introduce friend and peer Anarita Miccio. She performed her PhD at the San Raffaele Biomedical Science Park, performing research work on gene addition for beta thalassemia with Giuliana Ferrari. She then performed her postdoc at the Children's Hospital of Philadelphia before work as a researcher at the University of Modena and Reggio Emilia in Modena. She's now a group leader for the gene therapy group at the Imagine Institute of Genetic Diseases, and we look forward to her talk on base editing. Okay.
Thank you, Captain, for the nice introduction, and I would like to thank Evangelia for the invitation to this very beautiful meeting, I have to say. And so basically my lab is located at Imagine Institute in Paris, which is in the bigger Necker campus. And one of our main goals is to develop, to use editing and novel editing tools to develop therapeutic strategies for beta hemoglobinopathies. And so, as you might know, beta hemoglobinopathies are diseases caused by mutation affecting the production of the beta globin chain of the adult hemoglobin tetramer. And as you might also know, the beta-like globin gene are sequentially expressed during development, and soon after birth, the fetal gamma globin gene, so gamma 1 or gamma 2, or HBG1 and HBG2, are silenced, while the beta globin gene, the adult beta globin gene, is mainly expressed. So sickle cell disease is caused by a single point mutation that determines the substitution of valine for glutamic acid in position 6 of the beta globin chain. And this leads to polymerization of the sickle hemoglobin, which in turn causes formation of sickle-shaped red blood cells. And these cells tend to obstruct small vessels, leading to organ damage. And in addition, they have a short half-life, and so the clinical phenotype is also associated with hemolytic anemia. While on the contrary, beta thalassemias can be caused by more than 400 different mutations that causes a reduced or absent synthesis of beta globin. So we have, as you have heard yesterday, the beta 0 mutation or beta plus mutation. So we talk about beta plus mutation when we still have some residual expression of the beta globin, while beta 0, in the case of beta 0 mutation, the gene is completely inactivated. And so this reduced or absent synthesis of beta globin leads to the precipitation of the uncoupled alpha globin chains, and this in turn causes ineffective erythropoiesis with apoptosis of erythroid precursors, but also oxidative stress and death of red blood cells. And so the clinical phenotype is mainly characterized by anemia. And so the typical treatment are transfusion of red blood cells from healthy donor or pharmacological treatments, which however are lifelong therapies and are not equally effective in all the patients. And so the only definitive therapy is the transplantation of allogeneic hematopoietic stem cells that then in the body give rise to normal red blood cells. But as you know, this is available only to a small fraction of the patient, so the lucky ones that have a compatible donor. And so here it comes gene therapy, which is based on the autologous transplantation of genetically modified HSCs, hematopoietic stem cells. And in the last 10 years, many genome editing strategies have been developed for beta hemoglobinopathies, mostly using the CRISPR-Cas9 nuclease system, but also using novel editing systems such as the base editors. And in this case, the goal was either to correct the disease-causing mutation or to reactivate the expression of the fetal hemoglobin, as I will explain to you in a few slides. And therefore, the CRISPR-Cas9 nuclease system has been extensively used to develop therapeutic strategy for beta hemoglobinopathies. And as you know, the CRISPR-Cas9 nucleases generate DNA double-strand breaks in a specific region of the genome that can be repaired thanks to a donor template and the homologous direct repair pathway, and the donor template carry the desired edits. And so in this case, this pathway can be exploited for gene correction. And alternatively, the double-strand break could be repaired through the non-homologous engineering pathway that then ultimately lead to small insertion and deletion. So in this case, this can be exploited to inactivate genes or cis-regulatory regions. And as you will see later, both these strategies have been actually used to develop therapeutic approaches for beta hemoglobinopathies. And so the genome editing and CRISPR-Cas9 nucleases has been used to directly correct the endogenous gene, basically by generating a double-strand break in the mutant genes that is then repaired thanks to a donor template carrying the wild-type beta-globin sequence. And there are 2 clinical trials that have started or are about to start using this strategy. However, the HDR pathway that is required in the gene correction approach is less efficient than the non-homologous enjoining pathway in hematopoietic stem cells, which are the target cell population in our gene therapy approaches. And in addition, this approach requires the delivery of a donor template, which could be complex but also toxic. And we can also have double-strand break-induced toxicity. Indeed, the nucleases generate a double-strand break in the genome that can cause a DNA damage response leading to apoptosis. And in addition, it has been shown that they can also create a large genomic rearrangement, such as deletion and translocation. And therefore, the idea in the field is to develop a double-strand break-free or almost free strategy for beta hemoglobinopathies, such as for many other diseases. So either using base editors that can make C to T or A to G base conversion, or prime editing, a less developed technology that, however, can induce all base conversion. And finally, epigenome editors that can activate or inactivate genes as they contain an effector domain that can deposit or remove marks of active or inactive transcription, such as histone modification and DNA methylation. And so my talk will be mainly focused on base editing, a technology that has been developed by Professor David Liu at the Broad Institute. And in particular, base editor can make this pinpoint mutation in the DNA without generating double-strand breaks, as they contain an inactivated Cas9 nucleases fused to a deaminase, that is the enzyme that can make the C to T or the A to G base conversion in the case of the cytosine or adenine base editors. And therefore, base editing has been already explored to correct the sickle cell disease causing mutation and also some beta thalassemia causing mutation, as they do not induce double-strand break. You don't need to deliver a donor template, and we and others have shown that it can be highly efficient in hematopoietic stem cells. And so, unfortunately, there is no base editor that can perfectly revert the sickle cell disease causing mutation. However, we can use adenine base editor to convert the mutant A into a G and generate the Makassar hemoglobin variants that carry an alanine instead of glutamic acid in position 6 of the beta globin chain. However, this hemoglobin has the same properties of the normal hemoglobin. And indeed, we know that hemoglobin polymerization occurs because the valine in position 6 of the sickle beta globin chain interacts with an hydrophobic pocket of an adjacent hemoglobin tetramer. And so we predicted that the alanine in position 6 of the Makassar beta globin is not bulky enough to fit in this pocket. And actually, it was demonstrated back in the 90s in the lab of Stuart Edelson and Claude Poyart, actually in Paris, that indeed, at least in vitro, hemoglobin Makassar cannot polymerize. And so generating the Makassar mutation was not really an easy task and required a lot of protein engineering and guided RNA optimization, but at the end we managed to achieve up to 90% of base conversion in an erythroid cell line carrying biallelic sickle cell disease mutation. And this strategy has been successfully used by the group of Mitch Weiss and David Liu in human sickle cell disease hematopoietic stem progenitor cells, where they achieved high frequency of gene correction, and HPLC analysis showed expression of the beta globin Makassar, this one in blue, and accordingly, sickling assays show a significant reduction in the frequency of sickling cells. And so in our lab, we focus on the most prevalent beta thalassemia causing mutation, and we started with a beta plus mutation, the IVS1-110, that is a G2A mutation. And as you can imagine, it can be reverted using adenine base editor that indeed can make A to G mutation. And so this mutation maps to the first intron of the beta globin gene and create a splicing acceptor site, leading to partial intron retention in most of the transcript and the usage of a premature stop codon. And so this at the end causes a strong beta globin downregulation and a severe clinical phenotype. And therefore, Julia, at the time PhD in my lab, she treated hematopoietic stem progenitor cells from 2 homozygous beta thalassemia patients and a compound heterozygote for this mutation and another beta thalassemic causing mutation. And she achieved up to around 80% of genome editing efficiency with no indels, which are typically generated after double-strand breaks, thus confirming the double-strand break-free nature of base editors. And then the cells were differentiated towards the erythroid lineage together with the control healthy donor cells. And as you can see, RT-PCR showed restoration of the correct splicing with a complete, almost complete absence of the mutant mRNAs, which are characterized by a lower mobility because of the partial intron retention. And then HPLC analysis showed expression of the wild-type beta globin in red after gene correction, and the ratio alpha non-alpha was similar to the one observed in healthy donor samples in the homozygous samples, and was substantially ameliorated in the heterozygous patient where we corrected only one of the 2 alleles. And flow cytometry showed rescue of apoptotic precursors from apoptosis and levels of reactive oxygen species that are typically high in beta thalassemic culture were significantly reduced. And finally, we observed an increased enucleation rate, reaching a frequency that are similar to those observed in healthy donor samples, and quantitative phase microscopy showed amelioration of the parameters that are typically altered in beta thalassemic cells, such as the perimeter, the surface, the volume, and the dry mass, which is basically a good proxy of the hemoglobin concentration in red blood cells. And so we then wanted to demonstrate the efficacy of this strategy in long-term repopulating hematopoietic stem progenitor cells, and therefore we transplanted control and edited cells into immunodeficient mice, so human cells in immunodeficient mice. And as you can see, the frequency of human cells, so the chimerism was similar between control and treated groups, as well as the proportion of the different lineages, thus indicating that base editing does not impact the engraftment or the multilineage differentiation of hematopoietic stem cells. And interestingly, the genome editing efficiency was maintained in all the hematopoietic organs, averaging around 75%. And in particular, we sorted and we selected the GPA-positive erythroid cells from the bone marrow, and the HPLC analysis on these cells showed restoration of beta globin expression with a normal alpha non-alpha ratio, comparable to those observed in healthy donor samples, thus demonstrating basically correction of the beta thalassemic phenotype in vivo. And so similarly, Julia treated hematopoietic stem cells from homozygous beta 0 thalassemic patient, achieving similarly good efficiency of gene correction, restoration of beta globin expression with the normal or nearly normal ratio alpha non-alpha, and the rescue of the cell phenotype, for example, a reduced frequency of apoptotic cells. And then finally, actually correcting a beta thalassemic mutation, we were also able to correct sickle cell disease. And this is because in the compound heterozygote for the sickle cell disease mutation and a beta thalassemic mutation, they basically are sickle cell disease patients because the only globin that is expressed is the sickle hemoglobin. And so we, upon gene correction of the thalassemic mutation, we basically reproduce the genotype of a sickle cell disease asymptomatic carriers that indeed carries a sickle cell disease gene and a normal beta globin gene. And this led to correction of the sickling phenotype in vitro. And as I mentioned to you before, and as you have seen yesterday, there are also many editing approaches that aim at reactivating the levels of fetal hemoglobin that is usually silenced soon after birth.
And this is because the clinical severity of both beta thalassemia and sickle cell disease is mitigated by the persistent synthesis of fetal hemoglobin in adult life, which is typically associated with genetic variants known as hereditary persistent of fetal hemoglobin or HPFH mutations. And in particular, we know that gamma globin improved the globin chain imbalance in beta thalassemia, basically by compensating for beta globin deficiency, while in sickle cell disease, once incorporated into the hemoglobin tetramer, it prevents hemoglobin polymerization and therefore red blood cell sickling. And in particular, we focus on HPFH mutations that are mapping to the two identical gamma globin promoters. And these are mainly point mutation, and the other generate binding site for transcriptional activators that then bind to the gamma globin promoter and activate gene expression, or they disrupt the binding site for transcriptional repressor that then are no longer recruited to the gamma globin promoter to repress gene transcription. And so overall, the overall output are a high level of fetal gamma globin expression. And so in our first study, we used the CRISPR-Cas9 nuclease system basically to mimic this HPFH mutation. So the idea was to evict from the gamma globin promoter LRF, which is a transcriptional repressor, basically by deleting its binding site using CRISPR-Cas9 nucleases and taking advantage of the non-homologous enjoining pathway that is highly efficient in hematopoietic cells. And so this strategy does not require the delivery of a donor template, and similar strategies based on non-homologous enjoining are currently being tested in clinics. But of course, we are still generating double-strand break into the cells. And therefore, also here, we decided to use base editor for the reason that I explained to you before, and also because they can be used to precisely reproduce not only HPFH mutation that destroyed the binding site for transcriptional repressor, but also to precisely reproduce mutation that generate activator binding site. And in particular, cytidine base editor can make C2T mutation, and therefore they can be used to generate these 4 HPFH mutations that are known to diminish the binding of the LRF transcriptional repressor to the -200 region of the gamma globin promoter. And in the very same region, we can use the adenine base editor that can do A to G conversion to generate the so-called British -98 mutation that basically recruit the KLF1 transcriptional activators. And therefore, Panayotis in my lab, when he was in my lab, he basically used base editors to disrupt the LRF binding site or to generate the KLF1 binding site in the gamma globin promoter, achieving base editing efficiency that range between 40 and 50%, with no or very little indels compared to our previously developed Cas9 nuclease-based approach, the one that was basically deleting the LRF binding site. So HPLC analysis showed fetal hemoglobin expression in the base-edited samples, which was even higher compared to the Cas9 nuclease-treated samples when we generated the KLF1 binding site. And even though the editing efficiency was significantly lower for base editing compared to Cas9 nuclease-treated cells. In all the cases, in any case, fetal hemoglobin expression was sufficient to revert the sickling phenotype. And we also tested this strategy in beta thalassemic cells, where similarly we observed increase in fetal gamma globin expression and rescue of the thalassemic cell phenotype. For example, a reduced frequency of apoptotic cells and improved enucleation. And as you can see, the effect was more evident when we create the KLF1 activator binding site. And so in this study, we also perform a detailed analysis and a safety assessment of this strategy. And in particular, the p53-induced DNA damage response that is typically observed in Cas9-treated HSPCs was absent in base-edited samples treated with ABE and modest in CB-treated samples. As evaluated by measuring p21, which is a downstream effector of p53.
And we also analyzed the transcriptome of edited versus untreated samples. And in particular, RNA-seq analysis showed fewer transcriptomic changes in base-edited cells compared to the same samples treated with Cas9 nucleases. It was interestingly and reassuring to notice that when we use adenine base editor, the only 3 changes that we observed were the expected one. So a upregulation of fetal gamma globin expression paralleled by a downregulation of the adult beta-like globin gene. And then we also, in this case, we wanted to test the efficacy of this approach in vivo in long-term repopulating hematopoietic stem cells. And similar to our previous experiment, we transplanted sickle cell disease or healthy donor HSPC, reaching similar level of chimerism and proportion of the different lineages in all control and treated group. And the genome editing efficiency in the bone marrow of the treated mice was, however, reduced compared to our input samples, although we were able to achieve up to 30% of genome editing when we generated the KLF1 binding site. And so, as we have a 4 gamma globulin promoter, a clonal analysis showed that this, corresponding to around 70% of edited hematopoietic stem cells, harboring at least 1 edited promoter out of 4. And this led to fetal gamma globin reactivation, also in ex vivo derived erythroid cells. And so now we are trying to improve this base editing efficiency. In particular, we are testing by using HDAC inhibitors, arenase inhibitor, or even apoptosis inhibitors, or by modifying the electroporation process and the culture time. And finally, for the adenine base editor, we got very good result by basically using the new hyperactive adenine base editors enzyme. And so I hope you are convinced that we can use base editing for developing gene correction approaches that, however, are definitely mutation specific, but also to reactivate fetal gamma globin, which has the advantage of representing a universal strategy for both sickle cell disease and beta thalassemia, and in particular, independently of the specific disease-causing mutation. However, as you have seen yesterday, I want just to point out that both lentiviral but also Cas9 nuclease-based strategy are really promising tools and are already in clinics with early promising clinical result. And so far, no safety issue related to DNA damage has been reported. However, we think that also base editing is very promising, and in terms of safety, but also in terms of efficacy. And indeed, the first clinical trial has been initiated in November 2022 by Beam Therapeutics, who recruited the first sickle cell disease patient. And so finally, in my last 3 minutes, I would just like to mention something that we will be then discussing in the following talk. So I briefly mentioned prime editing and epigenome editing technologies that could be used also for developing beta strategies for beta hemoglobinopathies. And in particular, as prime editing can induce all base conversion, it has already been shown, and you will hear later more about that, to precisely correct the sickle cell disease mutation and to have a wild-type hemoglobin. And so in our lab, as we are using prime editing, because as prime editing can theoretically allow the introduction of multiple mutation, we decided to introduce multiple HPFH mutation in the gamma globin promoter that together can further boost fetal hemoglobin expression. And in particular, we decided to combine amongst the different combinations, we also decided to combine the introduction of 2 HPFH mutations that generate binding site for the KLF1 and the TAL1 transcriptional activators. As we demonstrated that the presence of the 2 binding sites leads to higher fetal hemoglobin level compared to the introduction of the single activator binding site. And we are now in the process of optimizing the frequency of concomitant introduction of both the binding site in K562 cells in cell line, and we will hopefully move soon to primary cells. And finally, we can also use epigenome editors, for example, to reactivate fetal gamma globin. And one idea could be, for example, to reactivate the gamma globin promoter by depositing active histone mark or by removing repressive histone marks. And finally, again, as you will hear tonight, the idea for the future, actually in a couple of talks, the idea for the future is now to develop actually an in vivo therapy for beta hemoglobinopathies, which could be based on the mobilization of hematopoietic stem cells into the blood where they might be more accessible, followed by the intravenous injection of, for example, viral vector or nanoparticle that specifically target HSCs. And this will definitely reduce the cost and the complexity of the gene therapy procedure and maybe even improve the safety, and therefore allowing the widespread use of this gene therapy approach for beta hemoglobinopathies and also other diseases. And finally, I would like to thank all the members of my lab and our funding agency and national and international collaborators and the EuroBloodNet European Reference Network, and of course all the clinicians, the hospital, and the patient, and you for your attention. I will be happy to take questions. Thank you.
Thank you for this wonderful talk and for bringing us back to time. If you have a question, please go to the microphone. So it can also be heard online. Nicoletta?
Just two very short questions. One is, with the base editors, do we still need, for your approach, to have a biallelic correction or introduction of mutation, or can you see a result with just modifying one of the two?
There is really a linear correlation. What I would say, I would like to have 3 or 4 for the moment. That's why the idea of prime editing would be that even with one edited promoter, if he's doing a lot of fetal hemoglobin silla, we'll have a selection of these cells.
And the second question regards the prime editors mostly. So we know that when we're using genome editing to target the HPG promoters, many times we see this deletion of the intermediate region. So is this something that we might see, or are you seeing something like that with the prime editors as well?
So with base editing, we occasionally see that, and also with prime editing, but at much lower frequency compared to the Cas9. I have to say that in our experience, we see it when we look at repetitive regions that are, when we target a region that is duplicated, like the HBG region. We don't see it when we have 2 different guided RNA. And when we have 2 different regions, but it happens typically for the gamma globin and for the alpha globin genes that we are also targeting in another project. So I think it's still the frequency, we still have some deletion, but when we are targeting 2 repeated regions.
Thank you so much.
Anarita, you have a question. I could have asked later, but I want to ask it publicly. It's about the like sort of a negative selection that you see in vivo, that you start with input material edited cells around 60%, if I recall well, then it goes down to 30%. Can you comment on what is the mechanism that is going on there?
I think it's different there are some common mechanisms between CBE and ABE and some particular mechanisms. I think the CBE, these are not the new latest generation CBE that are made using the TADE from the ABE, so they are really the ancient CBE. And I think that over there we still don't know why, but still we have P53 induction, and so there is still the cells are not doing well with the cytidine base editor. There is something wrong with that. But in general, I think that also when we were using CRISPR-Cas9 nucleases, I think we are electroporating the cells, delivering material. So in all our experience, if we don't have an input, a drug product, let's say, with 80% of edited cells, you still have always in vivo the selection of non-edited cells. So in the Vertex trial, typically they have good results because they really set up a good drug product, while Sangamo, for example, starting from 50% is not enough.
And may I suggest also there might be also the additional mechanism of the drift in hematopoiesis in this particular mice and system, whereby you are editing certain progenitors that normally will stick around in a human for longer, but in these mice are simply negatively selected because eventually you end up having a completely different hematopoiesis.
Yes.
So there's also that mechanism.
Yes, and also we were thinking about collaborating with Pietro Genovese or maybe with you, but we're doing a clonal tracking also of the cells to try also to understand a bit, but it's not so easy with the base editing, let's say.
Thank you.
So just a brief question from me as well. Okay, just because we're also interested in it. So how do you see company interest in moving therapies forward that are for specific mutations beyond sickle cell disease, where hundreds of thousands that might benefit? 5S1 110 for CD39. Maybe you've been in touch and have put your ear on the ground and seen if there would be interest in translating this.
I think there is a general problem of company being interested in genome editing approaches where you don't have your technology. Like for me, we are going to the clinics with base editing. I'm very happy for that, but we are going by, we will not probably go with a company because it's very hard when you don't have the technology that a company invests on that, knowing that you have to pay the license to be therapeutic for the base editing. Saying that, then we still can do something, but still definitely for mutation-specific approaches is much more difficult. But I think for at least for thalassemia, it should be tried because still it's true that they are mutation-specific, but there are a lot of patients. I don't know the patient in France and in Italy that for which we got the cells. There are really a lot of patients that could be treated. So it's not, CGD and immunodeficiency where each patient has one mutation. So that I hope this will change, but at the end it's we are still pursuing the academic level way to go.
Thank you.
Anna-Rita, can I have a small question? You mentioned data with the sickle cell disease and thalassemia compound heterozygotes. In your future, how do you anticipate to follow that, or you can reactivate the gamma globin? Because in our series recently, we have shown that the compound heterozygotes respond better than the homozygotes sickle cell just by increasing the F. Hemoglobin F, simultaneously lowering the hemoglobin S.
Yes.
What
so
is your
basically,
thoughts?
those are in general also for in our lentiviral vector-based trial, sickle beta 0 patients are much easier to correct because for different reasons, but still they have a lower concentration of hemoglobin, so still definitely they are much easier to correct. So in this case, what we are doing is now doing experiment in parallel with our best strategy to reactivate fetal hemoglobin and the gene correction strategy, because in this case, you're right, it might be sufficient doing 20% of fetal hemoglobin.
Yeah. And also you have to take into account that these people are undergoing the hydroxyurea treatments, and most of them are going.
Yes,
so
it's difficult not to find patients that are not undergoing hydroxyurea.
That's right.
Thank you very much. Okay.
Thank you.
So we move on to Dr. Angelo Lombardo. He received his PhD in cellular and molecular biology from UniSR and the Open University in the UK. He's assistant professor at the Vita Salute San Raffaele University and group leader at the San Raffaele Telethon Institute for Gene Therapy in Milan. His wit as sharp as his tongue, he is a pioneer in the field of gene editing in clinically relevant cell types, and he's an inventor on several patent applications and has received several distinctions for his work. His team was the first to develop a hit-and-run approach for targeted epigenetic silencing, showing durable and highly efficient epigenetic silencing in multiple cell types, and he's going to tell us more on his developments there in his talk on epigenetic editing. The floor is yours.
Thank you very much, Carsten. Thank you very much also for the organizer for the kind invitation. It's a big pleasure to be here. I'm really enjoying the breadth and the high quality of this meeting thus far. So, as mentioned, I'll talk about the new kid in the block. So, I will discuss about gene silencing and specifically on epigenetic editing.
The talk is not yet on the screen.
I cannot see my slides. It's up. Thank you very much. These are my disclosure. So, I've subdivided my presentation in three parts. As I said, it's probably a technology that might be new for you. So, a very short and brief introduction of epigenetic editing, epigenetics and epigenetic editing, and then I will discuss two cases on which you can apply. We are applying actually epigenetic editing to treat diseases. So you're probably familiar with this concept. So epigenetics is a broad term actually, which refers actually to changes in gene function, and gene function means gene expression, that do not involve alteration in the DNA sequence that have been coined by Weddington many years ago, in the last century. And there are maybe two big categories of epigenetic modifications thus far known. One is an epigenetic modification that tackle actually the histone. The histones are basically core component of our chromatin, and these histone made the nucleosomes, and through these histones they contain tails, and these tails can be actually modified post-transcriptionally. And there are actually 10 different post-transcriptional modifications that can be deposed to histones. These are very important because these modifications can actually, as I said, alter the 3D structure and thus also expression of a given gene. Another kind of modification is DNA methylation. Often DNA methylation occurs at CPG sites, the nucleotides, and when DNA methylation is deposed, it's often associated to gene repression. David Alice coined the terms of writers, erasers, and readers. So there are a plethora of proteins in our genome that can actually depose these different epigenetic modifications at the histone tail or onto the DNA. Some can erase actually these modifications, and others can read combinatorially this modification, as I said, to lead to gene activation, gene on, or gene repression. And here's a very important and pivotal biological process. You may know that if you consider that basically we all derive from a single cell, the zygote, but then at the end of the day in our body we do have more than 200 different cell types. Depends on the which kind of single cell analysis has been performed. So more than 200 different cell types, all of them, most of them with the very same genetic code. But as you can see from this picture, with substantial different phenotypes, and hence also transcriptional programs. So what are epigenetic editors? So they basically stems from the gene editing platform, the gene editing technology. So these are chimeric proteins which are composed of two different domains. One, of course, is a programmable DNA binding domain. There are many different types, and you will see later we will use basically all of them, from Zinfinger protein, transcription effector like activators, proteins, so the CRISPR-Cas9 system. And these DNA binding domains are fused to epigenetic effector domains, as MSA, as I said before, can reprogram the epigenetic status of your target locus. For the sake of this presentation, I will focus mostly on gene repression, but as said also by Anarita, epigenetic editing is a broad term that allows you also to activate gene expression. And when deals with gene repression, probably one of the most famous epigenetic effector domain used in literature is this CRAB domain. I will discuss very briefly about this CRAB domain that have been again fused to different DNA binding domain to program transcriptional gene silencing. Just to give you an example of how this platform operates, so you may think of having a gene which is active. These green lollipops basically represents histone tails, which are leading to transcription, so positive marks on the chromatin. And you may think of expressing this epi editor, the CRAB domain fused to the DNA binding domain, to target your gene of interest, as shown in this cartoon. As you see here, the color of the lollipops changes as well as the histone marks deposed on this site. And this is very powerful and lead to transcriptional repression. One of the biggest problems of this effector domain, which as I said is very powerful, is that the epigenetic modification deposed by this transcription, the epigenetic domain, are labile, in the sense that when the editor is diluted out from cells, if transiently expressed, the gene turns back to activation. So you can really program transient wave and efficient wave of gene repression. This is an example. So here is beta-2M, you are familiar with this gene, and you can program an epigenetic editor, known also as CRISPR-I, so the CRISPR-Cas fused to the CRAB domain, to the promoter of this gene. And here you can see in this graph, these are percent of silent cells, a transient expression of this editor is sufficient to impose significant repression of beta-2M, but when the repressor is gone in the cells, you have gene reactivation. So you can program transient gene repression, unless you are not expressing stably this type of epigenetic repressors. But we know, we'll discuss it later, that may be problematic and risky. So a few years ago, actually, we thought of improving this platform by taking advantage of a beautiful process which occurred early during development. And this process actually is involving the permanent silencing of endogenous retroamylins which are present in our genome. And this process is activated by the family of protein known as Crab-Zinzinger protein. So these proteins contain a Zinzinger protein that is able to recognize sequences specific of the retroviruses, and the Crab domain can recruit, as seen here, a plethora of other epigenetic effectors, endogenous epigenetic effectors, which, as shown here, modify the histone tails towards a repressive state. It is exactly what the Crab domain was doing in the previous slide. So the gene is off. That's not sufficient, though, early during development. You need to depose a second epigenetic mark, and the second epigenetic mark is DNA methylation. So this complex then recruits another family of protein, the DNA methyltransferases 3A and 3B. And the effector domain, which is exclusively present during embryogenesis in embryo, the DNA methyltransferase 3L, which deposes then DNA methylation. So the sum of these modification is then permanent or stable. So once deposed early during life, these sum of epigenetic modifications remain as such for the entire life of an individual. And it is very powerful, as I said, because again, our genome contains 500,000 of these elements. And early during development, you don't want to express, of course, these elements. So the Crab-Zinzinger proteins, together the DNA methyltransferase, are taking care of them, deposing DNA methylation and gene repression. And as you can see here from this graph, the representation is then stable throughout the entire life of the individual. So we took advantage of these beautiful biological processes by making cocktails of epigenetic editors. You still see the Crab domain here. On top of that, we added methyltransferase 3A and 3L. These are the two effectors which actually depose DNA methylation, thus locking the initial repressive states deposed by Crab domain. And here, coming back to this scheme, the gene is on, you program these epi editors to target your gene of interest, the gene is off, but then when the editors are diluted out, because again it's a transient expression of that, the gene remains off in a stable fashion. And this is an example. Again, beta-2M, targeting beta-2M with 3 independent epigenetic editors, the Crab, 3A and 3L alone, so it's not a cocktail, these are individual. Only the Crab is able to depose robust repression, but then is reabsorbed. Some effect from the 3A alone, the methyltransferase 3A alone, but then importantly, when you combine them, you have this nice synergistic effect, and you see that most of the cells become negative. So what we show is that by repurposing this beautiful process, we can achieve efficient and durable silencing in cell line. It is a hit-and-run platform, as I said. These proteins are transiently expressed into cells. Then the cells memorize the epigenetic modifications imposed on the initial cocktail and propagated them throughout cell division. It works as in a combinatorial fashion, so you need to have the 3 effectors together most of the time. It is inherited by DNA methylation. I will not discuss all the other details, but you can revert that by pharmacological treatment, by acting on the epigenetic modification deposed by the methyltransferase 3A, or by targeted reactivation. As I said, I will not discuss about that. You can make epigenetic activators with the very same DNA binding domain, but with protein which they strip out, let's say, or favor DNA demethylation. An advantage of this platform is that there is no risk of induction of DNA double-strand breaks. As said, so it is very powerful and effective in cell line. What about primary cells and eventually in vivo? Is it platform working too? So these are the 2 examples. The first one is very rapid polyfunctional engineering of T cells with the aim again of silencing multiple genes in these cell types without causing eventually DNA double-strand breaks. I will not go too much into the details of that because it's been nicely described this morning in this morning session. So engineered T cells containing either an engineered TCR or CAR CAR are very powerful tool to treat cancers, though we all know that there are significant roadblocks that blocks the efficacy of the treatments. There is probably a consensus now in the field that using orthogonal editing platform may benefit this treatment. So what I mean with orthogonal editing platform, that in a very same treatment, you can, for instance, insert an exogenous sequence, being that a TCR or a CAR, for instance, knock out another gene, or express, activate another one. So approaches in which you can, again, in a single step, modify heavily your T cell of interest. And we discussed this morning about the fact that probably the most advanced technology in this field is the CRISPR-Cas9 that have been already used in the clinic. While the clinical benefit of that is not been yet well defined, what was clear in the specific case is that multiple breaks that occur in a cell can cause reciprocal chromosomal translocation. And the field now is quite obsessed about the potential adverse effects related to DNA double-strand breaks, which may be, as said, reciprocal translocation if you're doing multiplexing, or even on-site genomic rearrangements, which appears more and more often present when you're using a CRISPR-Cas9 technology. So one possibility to solve it is to actually use epigenetic. As I said, we are using a DNA binding domain, which I actually forgot to mention in the case of the CRISPR-Cas9, is catalytically disabled. In the case of the CRISPR-Cas9, we are using catalytically inactive Cas9. So we need a DNA binding domain. We don't need the catalytic activity of this protein, of course. And in this specific example, so we are targeting two genes, beta-2M and TET2. The first one may be relevant for the generation of universally transplantable, the first step of the universally transplantable cell. The second one can be relevant for the generation of cells that persist longer in patients. And if you can follow me in this analysis, so the first one is a flow cytometry for beta-2M. So beta-2M is a highly expressed gene. You see here, these are the few remaining, let's say, cells which are beta-2M positive. When you treat these cells with AP editors, you see that the appearance of these nice beta-2M negative population. So it's pretty effective in silencing beta-2M. Then if you look at the transcript level, you see that in these cells that have been treated with AP editors recognizing both sites, both beta-2M but also TET2, are significantly silenced in these cells. In this simple analysis, a PCR analysis to look for reciprocal translocation between beta-2M and TET2, you may notice that in the case of the ETR-treated cells, there are no appearance of these bands, which corresponds to reciprocal translocation between these two genes, imposed in this case by the Cas9. So a single treatment may be sufficient to achieve high efficiency of silencing without a chromosomal translocation. And the very same holds true when you try to further increase multiplexing, here targeting 3 genes simultaneously. If you see at the gene expression analysis, all of them have been performed, say, long terms after, midterm after the treatment. Both 3 genes are highly silenced in these cells. I will not show you the data, but again, if you look at reciprocal translocation, there are no reciprocal translocation between these 3 genes with the ETRs, so with the AP editors, but there are with the Cas9. Now, as I said, orthogonal editing is something that you want to do in T cell. You can do that by using a slight modified version of these epigenetic editors. Epigenetic editors, for instance, that can be coupled with catalytically active or nicases, for instance, Cas9. In this specific case, by playing with the length, these are for the aficionados, by playing with the length of the guide RNA, you can simultaneously program epigenetic editing on one site, for instance, beta-2M, and gene editing on another site. And this gene editing can come with a single nick that may, for instance, stimulate insertion, targeted insertion of an exogenous cassette. And in this specific case, we are targeting the track locus. So these are as I said, a single molecule. Always these molecules express as a messenger RNA in the T cells. And again, here you can see the level of beta 2M upon this procedure. There is a nice fraction of cells which lose beta 2M because of epigenetic silencing. At the same time, there is a nice fraction of cells which acquire expression of the NY-ISO, which is encoded from this donor template that is used by HDR to be targeted into the TRAC locus. And here you can see the percentage of the double positive cells, which are both beta 2M negative and NY-ISO positive. So as I said, in a single molecule, you may have multiple functionalities, epigenetic editing or gene editing. Once again, if you now use this platform in which you are targeting exclusively one gene, the TRAC-C in this specific case, no matter if it is with a Cas9 epi editor or an Nickase epi editor, again, you don't have reciprocal translocation between TRAC and beta 2M because beta 2M have been epigenetically silenced. Well, to conclude this, epi-silencing in T cells is efficient and durable, and you can silence multiple genes in these cell types without inducing chromosomal translocation. And this new advance in the platform allows to achieve a polyfunctional editing, meaning that you can couple epigenetic silencing with gene targeting, again, if you are targeting one gene in the sense of the gene targeting without any kind of chromosomal translocation. So let me finish my talk by briefly discussing our experience in using this platform in vivo. Here, as a target gene, we use PCSK9. You may be probably familiar with this gene. This gene is mostly expressed in liver hepatocytes. And the role of this gene actually is to control the level of LDL receptor on the plasma membrane of hepatocytes. So PCSK9 licensed, let's say, the LDL receptor for degradation. And there are interesting cases in human beings in which loss of function mutation of PCSK9 basically protect from coronary heart disease. Why? Because in the absence of PCSK9, the LDL receptor is more abundant in the plasma membrane hepatocytes, so more able to capture LDL and cholesterol, so to reduce cholesterol in the blood. The opposite, if there are gain-of-function mutations, there are gain-of-function mutations which actually increases the degradation of LDL receptor, so led to an increased circulating cholesterol. That's an interesting target that has been actually tackled by many approaches, many companies, monoclonal antibodies, RNAi, and now also gene and base editing. The former two are actually already in the clinics. It's a nice example because it's a single gene expressed in hepatocytes, and there are now, as you can see, you will see in a while, nice and effective ways to deliver eventually messenger RNA in the liver to test the efficacy of epigenetic editing. So our experience very briefly in this field, you always start by selecting effective and specific ETRs. These ETRs are epigenetic editors able to silence your gene of interest. And we do this nomination always in cell line that we engineer to report for the expression of our gene of interest. And in this specific case, probably for the first time, actually, we compare different DNA binding architectures. And specifically, I said dead Cas9 to contain the 3 affected domain, tail to contain the 3 affected domain, of zinc finger proteins to contain the 3 affected domains. Well, to our surprise, actually, when we did this comparison, the best performing among these 3 DNA binding domains were the zinc finger proteins. Maybe not all of you may be familiar with zinc finger proteins, but they've probably been one of the first DNA binding domain developed, even also for gene editing. And they were performing very well. You see here in green a control, which is Cas9 cleavage, while in other cases these are curves corresponding to different doses of RNA corresponding to the different type of DNA binding domains. Most of them reaches very high efficient level of silencing of PCSK9. You see here 80–90%. And again, the tail were, the zinc finger were probably the most effective one, even at low doses. Once you have a very effective platform, you need also to show that this is also specific. Well, now here we have to deal with epigenetics, not anymore with the eventual genetic alterations. So we are looking at two parameters here. When we're looking at specificity of our platform, RNA-seq, so transcriptome, to look if there are genes which are differentially expressed, because again, epigenetic controls gene expression. And second is whole genome DNA methylation, to look if there are regions which have been methylated throughout the genome unintendedly. And here we compare, again, different conditions, cleavage with Cas9, AP editors based on zinc finger protein or catalytically inactive Cas9. And these are Vulcano plot referring to RNA-seq. You see that in all of them, there is the appearance of PCSK9 as expected as one of the top downregulated genes. You see here that there are no other genes downregulated in the case of Cas9 cleavage. There are no other genes in the case of dead Cas9 ETR, so epigenetic editors. There are actually few genes, very few genes, in the case of epigenetic editor based on the zinc finger protein. Overall, we think that the platform is fairly specific. If you look now at the surrounding genes, because one of the mechanisms by which these epigenetic modifications are deposed implies also eventual spreading of this epigenetic modification at nearby sequence. If you look at the expression of genes nearby the PCSK9, this is a quite big window, should be probably 100 kilobases, and there are all the genes in this window. Well, none of them, with the exception, of course, of PCSK9, are perturbed or deregulated, indicating that silencing is pretty tight and specific on your interne site. And indeed, if you look at the DNA methylation, this is an example. These are controls. As expected, the PCSK9 promoter is lowly methylated. You can see also here at zoom. When you are using the epigenetic editors, you see an increase in DNA methylation as expected, but that was confined to your target site of interest. So overall, we think that zinc finger are the best performing one, and there is limited, if any, transcriptional epigenetic perturbation outside of PCSK9. Now we do have, I'm not showing the data, we do have improved zinc finger-based epigenetic effectors, which actually are not imposing any kind of transcriptional perturbation. So very briefly, now we do have to deliver these guys into the liver of mice. I will not discuss about payload optimization and selection of lipid nanoparticles, but again, we use lipid nanoparticles for the task for quite obvious reason. Well, first of all, they are perfect in the sense of delivering messenger RNA in the liver. They do not are associated with stable expression of the epigenetic repressors. You don't want that, might be very risky in sense of target activity and potential immune reactions. And this is what we found by injecting these particles with epigenetic editors targeting PCSK9, Cas9 cleavage, and a bunch of controls. So this is the circulating level of PCSK9. Upon cleavage, Cas9, you see 50% reduction, stable until the last time point analyzed, 330 and 30 days. And a very similar profile of PCSK9 reduction can be seen upon expression of epigenetic editor. So again, a single transient expression in the very first few moments after delivery is sufficient to depose silencing for a long period of time. Is durable. Is durable not only in homeostatic condition, but also when we then force liver regeneration. I will go very fast on this. Liver regeneration upon partial hepatectomy tends to increases proliferation of hepatocytes. And even upon partial hepatectomy, so upon proliferation of hepatocytes, epigenetic silencing remains stable in time. And that was accompanied by, indeed, deposition of stable DNA methylation at the PCSK9 promoter. Let me conclude that a single pass in ETR expression can lead to efficient and durable gene silencing in vivo. We monitor these mice up to 330 and 30 days, and the silencing efficiency is around 50%. As I mentioned, we do have now improved AP editors which are able to impose higher AP silencing with no off-target activity, at least in vivo thus far. The platform is compatible with non-viral gene delivery approaches. We have seen that the design architecture of the platform can influence efficacy and specificity of the treatment. We still have to fully characterize the mechanism of action of this platform. And overall, I think this is a valid and potentially safer alternative to other editing approaches for in vivo gene silencing. So let me finish off by acknowledging the people, the great people that are working on these projects. Martino Alfredo Capelluti, Valeria and Sara, they are responsible for the PCSK9 in vivo project. Deborah, together with Tanya and Alice, they left the lab on the T-cell epigenome editing project. The funding agency, and thank you also for your attention. I'm open to take questions. Thank you very much.
Okay, go ahead.
Thank you very much for this fantastic talk. I have a quick one, and apologies if I missed it from your slides. So how many of these epi editors are you adding per target? So are you only targeting a single site per target? Are you adding multiple?
Yes, it depends on the application. In the case of, for instance, it depends on the application and the gene. Sometimes you have to hit many times the very same target with the epigenetic editor. That's the case, at least for now, for some of the genes that I showed to you in T cells. Some of others can be also silenced with a single target. Let's say. I said it depends. It's gene dependent. Let's say most of the time. Yes.
Fantastic. Thank you.
Great work. Thank you for your presentation. Comparing the two technologies, so the cleavage with Cas9 and the methylation with your zinc finger module, so which one would be your preferred one, and for what reason? And yeah.
Of course. So in the case of Cas9, it's already in the clinic as mentioned many times before, but I'm noticing more and more problems in this specific technology. It's probably more versatile at the end of the day, because with Cas9 you can inactivate or insert genes if you want. Cas9, you mean CRISPR-Cas9, not discussing about base editing and other platform like this. On the other side, our technology is a DNA break-free technology, that's for sure. We do have indications that it's better tolerated than a Cas9 cleavage, especially if you're doing multiplexing, for instance. On the other side, our technology is a technology which depends on DNA methylation. I haven't mentioned that, but in the initial slides, we often tend to target CpG islands because we need a certain density of CpG to depose sufficient CpG methylation to be propagated in culture. So each of the two has their own pros and cons, let's say. For gene silencing, if there is a nice CpG, I will go for epigenetic editing.
Thank you.
You're welcome.
Follow-up question on the methylation. So on one of your final slides, there was a tendency of demethylation. At least it appeared to me that some of the methylation marks got lost at a later time point. I assume that was the reason also that you did this regeneration experiment with the liver.
That was not an analysis on DNA methylation. Unfortunately, was rushing too much. That was the level of PCSK circulating PCSK9. They were though they were not statistically different from controls. Upon partial hepatectomy, that's what you mentioned. Instead, DNA methylation. If you look at the specimen from liver before and after partial hepatectomy, they were comparable between the two. So DNA methylation was more constant than the levels of PCSK9 in the blood.
So you think that the daughter cells remain the pattern?
In the daughter cells, in one of my last slides, remains the pattern of DNA methylation. Yes, after proliferation of the hepatocytes. Yes.
One last question from Nicoletta.
Thank you so much. It was a fantastic talk. I have 1,000 questions, but I'm only going to ask one. So basically a comment, more or less, not a question per se. How variable is the epigenetic silencing, especially the heritable epigenetic silencing, compared to targeting different genes in different tissues? And is there a guideline kind of to know what kind of epi editors to use for each specific region?
Right. So, as we stated in the paper 2016, probably the most important things, actually, the only genes that we investigated thus far are genes with CpG island. I know that there are, Jonathan Weissman published a paper last year or something like that describing the fact that also genes which, without annotated CpG island can be silenced. Annotation on annotation depends. So probably the gene that he was referring to were genes with a certain number of CpG islands. As said, DNA methylation is the only epigenetic marker that can be inherited throughout cell division. So in my opinion, it's very important that the genes you are targeting contains a CpG island or a certain set of CpG that can then impose a sufficient repression of the gene that can be propagated throughout cell division. As I said, also this might be a limitation of the technology because, as you may know, not all our genes contain a CpG island. A significant fraction of them, probably 70% of them, they do contain CpG island or some sort of CpG island at the end of the day. So that's the only probably requirement thus far. And again, you need to have very potent epigenetic editors. Those that we are developing now, that we have developed now, are really based on the very same effector domain, but for instance, containing a single DNA binding domain. Instead of being a cocktail of 3 independent effectors, they are containing a single DNA binding domain with the three epigenetic effectors fused together. That's the talk we have now.
Thank you. Thank you very much.
Thank you, Angelo.
You're welcome.
Now we'll move to the third speaker. I have a great pleasure to introduce a young investigator, a very promising one, who just recently joined as an assistant professor at the Department of Biology, Genetics and Developmental Biology at the University of Thessaloniki, following a very productive career at the University of Washington, Seattle, and working on developing novel global gene vectors. She's going to present us her current research on this issue. Nicoletta.
Thank you very much for the introduction. Thank you, everyone, for being here, and it is my pleasure to present you our later work regarding our effort to create a new or next generation of gene therapy vectors for beta hemoglobinopathies. And trying to find this. Yep. I think that this audience more or less will agree that gene therapy is could represent a revolution in health sciences. So we know that there is so far a very limited number of therapeutic products that have reached the market, but we do know also that the number of ongoing clinical trials regarding gene therapy and of IND applications is gradually increasing over time. And actually, it is estimated that by 2030, the number of therapeutic products reaching the market will be almost 10 times as many as we have now. So in order to support these efforts, we can ensure that the new generation of gene therapy vectors will be characterized by both a high efficiency and a high safety profile. And this can be accomplished when we're talking about high efficiency, first of all, by creating vectors that have a high transgene expression that can also be linked to a high therapeutic effect, and at the same time try to reduce the cost of vector production and also increase applicability and availability of these approaches. Now, in terms of safety, there are many things that we can do. First of all, we can try, of course, to employ insulators for a subset of the vectors that we're using to avoid silencing of our transgene and also interaction with the neighboring gene, especially when we're talking about integrating viral vectors. And we can also try to create vectors that are very cell type specific. And another prerequisite nowadays is that the vector has to be efficient, but also in very low vector copy numbers or low viral load in general. So on this slide, you can see a few of the commercially available viral vectors, and the design of these vectors is pretty simple, I could say. There is a transgene, and usually we have a constitutive or very strong promoter driving the expression of the transgene. It can be an RSV or CMV promoter, and these vectors are definitely efficient. However, a similarly constitutive promoter lately, the MNTU3 in a viral vector for cerebral adrenal leukodystrophy was recently linked to, I think, 3 cases of MDS in patients recruited in a phase 3 clinical trial. So constitutive expression, high expression is good, is efficient, but can be unsafe. Another thing that I wanted to mention is that transgene overexpression is not always beneficial. So recently we saw several preclinical studies regarding gene therapy approaches for Friedreich's ataxia, where they used an AAV vector for in vivo gene therapy. And these vectors were also overexpressing the therapeutic gene, the FXN gene. And what they realized was that this overexpression of the therapeutic gene actually led to a mitochondrial dysfunction. And the phenotype that they got at the end was very similar to actually the phenotype of the disease itself. So it's very important to also try and fine-tune the expression of a transgene, and also, and especially for this case, the results were variable compared to different tissues. Again, I would like to emphasize that sometimes we need to really focus on cell type specific viral vectors. So a very characteristic case of transgene expression driven by cell type specific regulatory region is that of the lentiglobin vector or vectors in general. And here we have, of course, a modified beta globin gene that is our therapeutic transgene, and this is linked with the beta globin promoter, so we have an erythroid specific expression. And in order to achieve very high levels of transgene expression, this vector also incorporates a 3' UTR beta globin enhancer, and also a truncated version of the beta globin LCR. And this region spans about a 3 kb size. So these vectors, and we heard yesterday, and we know that they have been both efficient and safe in the clinic. However, there is the issue of the size. So this large, especially these large LCR cassettes, leads to a few limitations regarding cell vector production. So we have lower viral titers that subsequently might lead to an increased cost of production. And another thing is that these vectors are kind of more difficult to transduce the target cell type that we're after, the hematopoietic stem cells. So a few years back, we questioned ourselves, can we further improve these lentiviral vectors for beta hemoglobinopathies? Can we find other erythroid-specific, again, enhancers, but smaller and still potent enough to successfully replace the micro-LCR? How can we look for enhancers? First of all, over the last decade or two, there have been numerous efforts to decode and identify enhancer elements in a number of tissues and cell types and developmental stages. The majority of these studies relied and continue to rely upon some common enhancer features. And the first, and I think undisputed feature, is chromatin accessibility or DNase I hypersensitivity. We do know that all enhancers are DHSs, but definitely all DHSs are not enhancers. Therefore, there are more features that have been used to identify or describe these regions, and these can be, These can be, of course, post-translational histone modifications or transcription factor binding, especially cell type transcription factor binding. However, these characteristics are more or less descriptive and they're not predictive. So it has been shown that not all enhancers need to bear all these characteristics. Fortunately for us, over the last decade or so, there is a way to functionally interrogate and dissect these regions. And the way to do that is called a massively parallel reporter assays. So what happens here? If we have a library of candidate enhancers, then we can clone this library usually into a plasmid, into the 3' end of a plasmid, or we can couple each candidate enhancer with the barcode and clone it on the 5' end on the plasmid. And the metric that we get at the end is RNA abundance normalized by DNA presence. And what was usually done until now is the creation of a plasmid library bearing these candidate enhancers and transfection of those libraries in cell lines, usually cancer cell lines that are easily manipulating and easily accessible. However, we now know that the best way to really identify an enhancer sequence is to put it in a genomic context. So I'm not going to stay here for too long, but there have been similar assays based on lentiviral integration of these cassettes to identify these enhancer sequences. So we know that there are ways to functionally find enhancers. In a recent study, we were able to precisely map the regulatory landscape of adult erythropoiesis. You will hear more about this tomorrow from Dr. Georgolopoulos. I just want briefly to say that what we did was to perform a dense DNA-1-seq over a course of 12 days of ex vivo erythroid differentiation. And what we observed was that we could find regions gaining DNA's accessibility, as you can see here, and these were highly correlated with activating genes. We therefore theorized that we could find erythroid-specific enhancers within these opening DHSs. So our next step was to compile a list of all these opening DHSs, and that gave us a list of about 5,000 DHSs that gain accessibility during erythropoiesis. In most cases, we're talking about intronic and intergenic regions, and most importantly, these regions have minimal to no accessibility in other hematopoietic lineages. We then dissected each one of these regions into overlapping tiles of about 200 base pairs each, so now we have a library of 15,000 sequences. And our next step was to incorporate this library into a lentiviral vector. And this viral vector has two features that I want just to emphasize. Instead of using a general minimal promoter, we used a short beta-globin promoter. And the other feature that we wanted to introduce was an insulator. We basically used a recently discovered dual function insulator. So this is an insulator that was identified in the lab of Dr. Evangelia Giannaki, and we knew beforehand that it was an enhancer blocker sequence, but then we also realized from very long-term strenuous in vivo experiments that it can also protect against gene silencing over time. So we kind of created this closed system, or as close as possible, for our assay. Then instead of measuring like RNA to DNA, we used a modified lenti-MPRA FAXed assay. So what we did was to transduce this 15,000 lentiviral enhancer library into HUDEP2 cells. This is a cell line that is very similar to erythroid precursors. And we did that with an MY of less than 1 to ensure that not more than one of our sequences is represented per cell. And then we sorted the cells into 3 bins: the low GFP expression cells, medium, and high GFP positive population. And we thought that If a sequence is really an enhancer, it would be found more frequently in the GFP high population. At the end of this assay, all these 15,000 sequences were ranked based on their effect on GFP expression. And I'm not going to get into much detail because we started with 15,000 sequences, and then we started to narrow down, and we selected about 40 of them, and we individually tested them in the same cell line, the HUDEP cell line. And what we observed here is that we can get a broad range of enhancer activity. So by using it, if you want to really fine-tune a gene, you can use a stronger or less strong enhancer to achieve this. Something quite important that we also noticed is that comparing the results from the HUDEP2 cell line to CD34-derived erythroid cells, we got a pretty good correlation. However, when we got back to a cell line that is very commonly used for all these genomic assays, the K562 cells, we didn't see much in terms of correlation in our results. So now we have enhancers, they have a broad range of activity, but are they really erythroid specific? To answer this question, we performed 2 sets of experiments. In one of them, we transduced CD34 positive cells with each of these 40 enhancers, and then starting from the same pool of cells, we differentiate them towards the erythroid, megakaryocytic, and granulocytic monocytic lineage. And as you can see here, we only got a good expression, a good GFP expression in the erythroid ex vivo differentiation. We didn't see much in the megakaryocytes or granulocytes. And to confirm this, we actually went one step ahead, and we also performed in vivo experiments. We transduced, again, CD34 positive cells with a pooled mini or micro library of these 40 enhancers. And post-sacrifice, you can see that we got more than 80% of GFP expression in the erythroid lineage and a minimal expression in other lineages apart from the myeloerythroid progenitors. One thing that came out of our CD34 trials was that these enhancers have a variable activity. So the stronger enhancer at the beginning or in the early stages of erythroid differentiation is not the same as the strongest enhancer at the end of the erythroid differentiation. And what was pretty interesting is that this ectopic, basically, enhancer activity was almost perfectly correlated with the in situ DNA accessibility, and not only for the strongest early and late enhancer, but in general for all the sequences that we tested. Another thing that was kind of interest to us after we focused on these very strong early and these very strong late enhancer was that they indeed lacked the stereotypical enhancer characteristics. You can see here that we did not observe any H3K27 acetylation or K9 trimethylation. To identify exactly what these sequences regulate, we performed knockout or deletion experiments in the same cell line. We deleted these 2 regions. This is the early enhancer, and we found out that actually it regulates PVD1, and the late enhancer regulates the PPAR alpha expression. So now we have not 1, but 2 enhancers that we wanted to test in a therapeutic vector, and this is exactly what we did. So we used the vector. Our transgene was an shRNA for BCL11A, and this vector also has a GFP cassette just for normalization tracking purposes. So here we cloned our 2 novel, very small enhancers, 300 base pairs each, and compared them with the entire micro-LCR, so the difference in size is almost tenfold. The first thing that we noticed was that we could indeed get a much, much higher titer with the new enhancers, kind of expected. We have a shorter cassette, we will get a higher titer. And what was very nice to see is that despite the difference in size, the HbF reactivation capacity of these vectors was pretty much identical in all three cases. Then we performed in vivo experiments, xenotransplantation experiments in NBSGW mice. We used healthy donor CD34 positive cells. We focused only on one of the two vectors that I described, since we already kind of knew that they're pretty similar. So what you can see here is that we have exactly the same engraftment in all lineages, which kind of means that if our PVD1 enhancer had a leakage and was not really erythroid specific, we would be able to see an effect, especially in the B cells and generally in cell repopulation of the bone marrow. And another thing that we observed was, again, expected. We observed GFP expression only in the erythroid compartment from the human engrafted cells, almost nothing in the other cell lineages. And what was very nice to see is that with the new vector, we got an almost twofold increase of HbF positive cells in the engrafted human erythroid cells. We then wanted to test these vectors in thalassaemic CD34 positive cells. We used cells from overall 5 donors, I believe. The experimental procedure is pretty standard: pre-stimulation of the cells, transduction, and then ex vivo differentiation for 18 days. So what you can see here is that the first thing that we observed was a proliferative advantage, especially in the case of the new enhancer. We got a statistically significant higher number of cells in different time points throughout the erythroid differentiation. And I'm sorry, one thing that I forgot to add here is that for these experiments, we went with the same MOI for both vectors. And moving on to HbF expression, again, you can see, of course, we have an increase of HbF using both vectors, but the increase is more pronounced using the PVD1 vector, especially in enucleated cells. Our results from flow cytometry were confirmed by HPLC, and something that made us very happy is to see that using especially the new vector, we were able almost to balance the beta-like globin to alpha globin ratio. So it was, if you can see here, is almost 1. And of course, this HbF increase also had an effect in the fitness or kind of phenotype of the cells. You can see here that we have a very significant reduction in the oxidative stress. The ROS levels were pretty significantly reduced, and also we got a much better enucleation of the cells transduced with a vector. So our efforts don't stop quite here. I would actually like to invite you all in the oral presentation sessions for today. There are 2 more projects related to the one that I just presented. The first one will be presented by a PhD student, and it regards her effort to combine these different vectors and see if we can achieve or create a synthetic kind of super enhancer. I don't know if everyone likes this terminology. And we do have some very promising results so far. And another talk today will be from a master's student from our lab. Alexandra is working with something kind of different. So now we have a small transgene. We also have small regulatory sequences. And we thought that there is space for us to add something more. So we decided to add a selection cassette that can actually allow the cells to expand both ex vivo, but most importantly, in vivo. So to conclude with, I hope that I convinced you that we can mine the genome for lineage-specific strong enhancers, if we need strong enhancers. We used this information to create
new shRNA vectors for beta hemoglobinopathies that had increased viral titers. They had a similar safety profile to the microLCR control vector. They were able to increase HbF both in vivo and in thalassaemic cells. And with that, I would like to thank all my collaborators, Kyriaki and Alexandra, of course, for their very, very hard work. Pavel, Sova, and Grigoris, I could not have done any of this without them. And of course, our supporters, Thalia Papagiannopoulou, John Stamatomagionopoulos, Jeffrey Erstra, and Evangelia Yanaki. But most importantly, I would like to thank and dedicate this work and this effort to George Stamatomagionopoulos. He has been the mastermind behind this work. And I'm very glad that it's finally completing. And with that, I would like to thank you all, and I'm ready for any questions.
Thank you very much, Ms. Nicoletta, for this excellent presentation and the nice data, which are very promising. I just have a quick question regarding the temporal behavior of these enhancers. Is there any concern that this might be active during the early hemopoietic development, as like the LCRs have been detected recently by Sutherland's group that have transactivated neighboring genes? Is there any concern about that?
That's a great question. So we don't believe that even the earliest of the enhancers that we have found can be active in more primitive cells, in stem cells or early progenitor cells, because we did all our assays in already established or committed erythroid cells. Of course, we could do more and more
in vivo.
More in vivo experiments, but I think that the in vivo experiments that we did, especially by using that, and that was kind of the reason why we went with the shRNA for BCL11A, to actually prove that possibly there is no activation in other hematopoietic lineages.
Okay, thank you. Any question?
Okay, then maybe I'll ask. Well, go, Anarita, go. Ladies first. Go.
Very nice data, really. Still like a lot lentiviral vector. I was wondering, you said that you use the same Y, but is the vector copy number the same in between when you compare, the classical lentiviral vector with all the locus control region and the short one?
The vector copy number is not the same, actually, no.
So how much, if you normalize per vector copy number, do you still have a beneficial effect, or?
I think it was about we found like a twofold increase or something like that. It was not like a hugely different integration of vectors, but yeah, the difference was mainly because of the different integration, I guess, and not as much because of the compact enhancers being stronger than the micro LCR. This is not possible.
But at least you will have a better titer vector.
Yeah, so we have a better titer, and even when we normalize for that by using the same MOI, we still get a better integration. So the cells are more easily transducible, I guess.
Vector, yes. Thank you. Nice work.
Thank you.
So then, with the smaller elements, and if you're not reaching, still not reaching the wild-type levels of LCR-based enhancement of expression, would it make sense to combine several of these enhancer elements, maybe in a beta-globin vector, for example, to maybe get higher per integration beta-globin expression?
So far, it's a very nice question. So far, we are trying to combine at least two of them, so there is no reason to create an equally large, like 3 kb enhancer by just using different genomic sequences. I think the micro-LCR will always be the best. But we do have some nice data by combining these two enhancers. We still have just a 600 base pair enhancer that can be trimmed down actually quite a lot. And it seems that it can be used. We have not tried it with a beta globin vector, but we did try it with a gamma globin vector. And we do some very, very good results with that. And you're welcome to wait for Kyriaki to present her data, because she's going to talk about that.
Not least, presentation of the session by, and I have the pleasure pleasure to present Professor Dr. Luca Biasco at the University College of London and the Executive Director of the Hematopoietic Stem Cell and Gene Therapy, Sana Biotechnology in Cambridge. Dr. Biasco also is known for his elegant and energetic presentations, so we look forward to that.
No pressure. Thank you. I know I'm the last person standing between you and the coffee break, so I do my best to keep you awake. Today I'm going to talk about our work on designing and devising system for in vivo delivery to hematopoietic stem and progenitor cells. And thank you, the organizers, for the kind invitation to this fantastic venue and to this very interesting meeting. So as you know, human hepatopoiesis is organized hierarchically with hematopoietic stem and progenitor cells, HSPCs, that are localized at the top of the hierarchy. They are in the bone marrow, in an adult individual, and they are able to generate a plethora of mature blood cells with different morphology and function. In particular, the CD34 compartment that defines HSPCs is heterogeneous. You have hematopoietic stem cells and multipotent progenitors at the top of the hierarchy. They have the ability to generate all blood cells, and they are usually endowed with a long-term survival ability. While you have more short-term unilineage or oligolineage committed progenitors, megakaryoid, erythroid, myeloid, and lymphoid, that have a more limited potential. Now, you also know that a series of genetic mutations have been identified that affect the ability of HSPC to differentiate properly in different lineages, with resulting defects in red blood cells, neutrophils, monocytes, and lymphocytes. So naturally, the ability to intervene with a therapeutic payload in this population would unlock the possibility to correct a plethora of genetic disorders. So it's very appealing, and this is the concept at the basis of the ex vivo hematopoietic stem cell gene therapy, which you heard a lot from the previous speakers, which is based on autologous transplantation of CD34 cells that are isolated from a patient. They are genetically engineered with a viral or non-viral vector system, and then they are briefly expanded in the presence of cytokines, usually, and then they are infused back into the patients, and the patients usually needs to receive a conditioning to make space in the bone marrow for the engineered cells. So this therapy, this technology works in a lot of contexts, and has been proven clinically in a series of genetic diseases. But there are some limitations, and primarily they are due to the fact that this is a very complex protocol, and as a result is a very complex drug product. Is a composition of a cell product and a vector product in one. So to produce this drug, to deliver this drug is complicated, and this limits the access to patients, and of course ends up in being an extremely expensive therapy. So naturally, the best solution would be to find a system where we can deliver a genetic payload directly in the bloodstream, and count on the fact that with a delivery platform, this could reach the hematopoietic stem cells directly in the bone marrow of the individual. So this would be a much easier manufacturing-wise. It would not require any conditioning regimen because you would meet the cells in their natural niche, and it would improve dramatically access to patients. So in this talk, I'm going to talk about two main enveloped delivery platforms that you can use, that we're working on at Sana, to deliver genetic payload to hematopoietic stem cells in vivo. One that is a lentiviral-based platform that contains a viral genetic material and permanently integrating payload. Another one that is a viral-like particle that is still an envelope particle but contains a cargo that could be the fusion of a Cas9 or and a single RNA that essentially provides for the means of editing, and this would lack the viral genetic material
differently from a lentiviral vector. But regardless of the platform that you use, when you are developing an in vivo delivery system, you have to meet certain characteristics, particularly for in the in vivo delivery to HSPCs. Number one, these genetic payloads need to be able to deliver, to be delivered in HSPCs when they are in steady state, without any cytokine stimulation, because you don't have the luxury of activating these cells when you are targeting them in vivo. Number two, you have to reach these HSPCs in their natural niches, which are primarily in the bone marrow, but also in the circulation. And number three, potentially you have to develop a system that avoid major cellular sinks and other blood cells and deliver specifically to hematopoietic stem cells, primarily so that you have highest bioavailability on the target. And when you have to develop this preclinically, your options are limited. You can take HSPCs from a healthy individual or from a patient, and you can test these cells either by in vitro assays, where you would test the ability to transduce or to engineer these cells when they are in steady state condition, or in vivo in xenotransplantation animal model, where you would transplant these cells and then you would go in with the vector and see whether you're able to eat these cells when they are engrafted in the mouse. Why am I not mentioning here syngeneic mouse? Why we are working only in humanized mouse models? The primary reason is that the mouse is not permissive to all envelopes. So certain vectors are not designed to eat mouse cells. So you just don't have that option. The other issue is that when you are targeting your delivery system for a specific human receptor, this might not be expressed in the mouse, or might not be expressed in the mouse in the same cells where it's expressed in humans. So these are the options we're left with. And I'm going to talk about both systems, and in particular also about the potentials and limitations of using humanized mouse models for delivery in vivo. Now, I started this program 1 year ago, and we started with a very simple constant, like single antiviral vector encoded for a GFP, and we tested two main envelopes, a BAVTR baboon-derived envelope and the VSVG envelope, which is the one that most commonly is used in a clinical ex vivo application. And we designed a system where we would test this vector on 34 cells, freshly isolated or freshly thawed, without any cytokine stimulation. So you would take these cells, you would let them rest for 1 day in the media, and then you would expose them the next day for 24 hours to these viral vectors without any cytokine. Then you would wash the vector out, and you would add cytokines just to keep the cells alive for 7 days, until which time you can then measure transduction and by flow or by VCM. So with this system, you can already highlight differences. Not all envelopes come with the same potential to eat these cells. We know that VSVG doesn't get into these cells very well. VSVG use LDLR as entry route, and this receptor is overexpressed only when 34 cells are activated. So we also tested several formulations of our BAVTR-LV, and we constantly see that we can get very good transduction up to 100% in different MOIs. We went a step further, and we FACS sorted the individual subtypes. I mentioned that 34 cells are heterogeneous, so we FACS sorted HSCs, MPPs, etc., and we exposed them to these vectors in resting condition. And you can see that BAVTR-LV is able to transduce all subtypes almost equally at MOIs that are close to an in vivo scenario. Again, also when you're doing this test in vitro, you have to think about what will be a potential application in patients. If you go in with the MOI 1000, okay, you can make things happen, but then eventually you cannot translate this data in vivo in humans. So this was a very good basis to start testing this vector in vivo. So naturally, the classical in vivo scenario is the following, where you would infuse CD34 cells in an empty we use MBSGW mice because you don't need to condition them, and they have the KIT mutation, they have a better myelin engraftment. You add 34 cells, you follow these mice for around 12 weeks, and then you go in with your vector intravenously. And then 12 days later, we would take down the mice and check the bone marrow and peripheral blood. Now, this study that we conducted very early was around September, October of the past year. We clearly showed that BAVTR is able to get, this was one of the initial formulation we had, was able to get into around 2% of total human cells and 2% of HSPCs in these mice. And clearly you can see the difference with the VSVG, where essentially you can barely detect transduction in this system. Now, this is all good and well. I mean, it's simple enough. But the problem of this experimental model is that the 34 cells that you infuse change dramatically in nature. So if you wait 12 weeks, eventually you start from a pie chart like this, where the majority of the 34 cells are myeloid progenitors, and you have a big chunk here of HSCs and MPPs that essentially you lose progressively as you wait, as these cells are staying for a long time in the mouse. And the majority of the 34 cells are actually pre-BNK. So when you measure 34 transduction in these mice, you're measuring it in a completely different population. So for this reason, we developed two alternative experimental models to add on top of the long-term humanized mouse. One is the co-infusion model, where you infuse the 34 cells, and then essentially right away you infuse your virus. So this model is to model access to HSPC in the peripheral blood. So essentially the virus is chasing the cells in the circulation, and you have the best chance to eat all the progenitors because this is the fresh infused material, so its composition is exactly the one that we have in humans. And then we developed a second model, which is the day 7 short-term humanization model, where you infuse the CD34 cells, then you wait for just 7 days, and then you're going in with your viral vector. You can also mobilize these mice during these 7 days. As you can see, the composition changes, the 34 cell changes over the 7 days, but not so dramatic as I showed in the long-term humanized model. The other thing that I'm going to show you is that all the human cells are in the bone marrow by day 7. So this isolates the question, can I get to these cells when they are only in the bone marrow and they are not circulating? So we started with from a co-infusion model, and clearly the numbers that we obtained by infusing BAVTR-LV right away in 34 cells were much higher right away as compared to the long-term humanized mouse, where we get up to 30% total 45 cells and around 12% of phenotypically defined HSCs. This is if we take down the mice at day 12, but what happens if we follow these mice for a longer time? So this is kind of an ex vivo scenario where you infuse the cells that are transduced, but you don't have the manipulation. The cells are freshly infused and the vector has to transduce them. And nonetheless, you have, it's very interesting because in the first 6 weeks, you have a transduction level that is around 20%, which really resemble what we observe in the bone marrow at day 12 takedown. But then after 6 weeks, you have a drop in GFP positive cells and then a stabilization around 5–7% of transduction. So when you take down the mice at week 16, this is the transduction you're able to measure. It's around 3%, which is different from what you measure at the beginning. And it's very interesting to notice that even though we had 12% of phenotypically defined HSCs, they should stay there, they should stick around, but eventually, clearly at the end, you barely see HSC actually. We have to merge them with MPP, and the transduction at the end is 2%. So this is telling you actually that this mouse model is rather skewed, and a lot of the progenitors that would normally survive in humans, they are not very well represented here at week 16. Now, let's move on instead on the day 7 short-term humanization model. We made this very interesting, I think, at least in my view, it's a very interesting observation. So if you infuse the 34 cells, then 2 days later you start a mobilization regimen. So these cells went to the bone marrow and then you start to mobilize them. regardless of whether you mobilize them or not, you don't see egress of these cells in any compartment, total 45, linear genetic 34, not even in the HSC. So none of these cells egress to the peripheral blood. So mobilization increases their number, but only inside the bone marrow. So these cells either are more, or are also more, we think are also more accessible, just because you are mobilizing them from the endosteal niche to the perivascular niche. And so you can get to these cells through the vasculature. We think you have a better chance, a better access. But this model does not allow for access to the peripheral blood. So it's a very complicated model, and it's difficult for the vector to find these cells. Also, these cells are very few in the bone marrow. And nonetheless, these are more recent data where we optimize our BAVTR LV platform. And essentially, now we can get up to 6% and 5% of basal access of human 45 and HSPCs when they are only located in the bone marrow. And here, okay, we have pretty variable results. We have now more recent data that are confirming this, where essentially you have a higher access to HSCs upon mobilization, even though these cells never went to the peripheral blood, just by the fact that they are more accessible inside the bone marrow. So these are numbers we can start work with. So we decided to move on and develop a VLP-based platform, which contains a Cas9 fused with a GAG and a B2M single guide RNA. And we made it with the envelope that was a BAVTR envelope because of what I just showed you on the LV counterpart. So we started over. Does this platform edits resting steady-state HSPCs? And the answer is yes, very well. We can get up to, here we can measure editing in two ways. The pink is editing by flow, and the dark blue is editing by molecular analysis, by sequencing. And every time the sequencing gives us higher results, and the reason is that by flow, you don't pick up all the monoallelic edits, and also the fact that the readout depends on the turnover of B2M. There are certain cells, like HSCs, are more resting, they have a lower turnover, and so if you don't wait long enough, you cannot see the effect of the functional effect of your editing as well as in the other cell types. Now, again, we did also editing on individual subtypes, and you see that overall HSCs, they are a little bit more resistant to edit, but we have very, very good numbers in terms of editing ability of steady-state HSCs, even at very low MOI that, again, are compatible with an in vivo use. And then we started testing these VLPs in vivo in the 3 different models: long-term humanization, day 0 co-infusion, and day 7 short-term. And these results were pretty much on par with what I showed you with the LV counterpart. Particularly now, I would like to focus on the day 7 short-term data. Remember these numbers. We got in this early version around 2% editing of 45 cells and 0.7% of lineage negative 34. Again, this is without any post-selection anything. Okay, these are good enough starting point, but we clearly want more for an in vivo application. Okay, so we started working on optimizing these VLPs, and we had recently what I think was a major breakthrough, which we conducted another study on short-term humanization. This time we are immobilized with the MD3100. And this was, again, the early version of the VLP that I showed you before, very reproducible results. This time was 1.3% editing on 45 and 0.5% of 34, pretty much the same what I just showed you. With the optimized VLP, we now get to 20% in vivo editing of human 45 with just a single dose and 16.2% of lineage negative CD34 cells. Now, these are numbers we can start work with in terms of a clinical translation. But we decided to go a little bit further, and we also perform additional experiments where we decided, what if we add more than one dose? And we wanted to see whether adding 3 consecutive doses, and we combined that with MD3100 mobilization, if this would improve the editing. And indeed, this was the case. With 3 doses, we can now get up to 47% editing of human 45, which correspond to more than 30% editing to the lineage negative 34 compartment in vivo. And you can see that you can really pick up the edited cell clearly by flow. Now, the 3 doses also improve access to HSCs and MPPs. Here is like the data broke down by subpopulations. In some cases, like pre-BNK, you will get a very efficient editing above 50%, but you can clearly edit myeloid cells, erythroid progenitors, and also the primitive subfraction. Why we think that 3 doses are better than 1? Besides the additional amount of VLP that we infuse, we think that because we do this concomitantly with the 3 doses of MD3100, we have data showing that you are expanding HSCs in the bone marrow with this mobilization regimen, so you're making them more accessible. So it's a combination of VLP dose and accessibility. Now, potency is there. The last piece is specificity. Can we make this vector specific to target HSPCs? At Sana, we are developing a fusogen platform that is based on paramyxoviruses system. Paramyxoviruses, they have G and the fusion proteins that are separated, so the recognition, the vector attachment to the cells and the fusion is on 2 separate molecules, which is not the case for other viruses like VSVG. And essentially you can attach a binding site, a binder to the G protein, which you can make it so that it recognizes a specific receptor on the cell. When this binds to the receptor, it triggers a conformational change in the fusion monomer, which promotes the fusion of the viral and the cellular membrane. So the result is that in theory, you can deliver a genetic payload only when the binder recognizes a specific receptor. One of the receptors we are working on is CD133. And the reason is that this is very nicely, specifically expressed by CD34 positive cells and not CD34 negative cells. So this is very appealing in that sense. Starting over, can we get into resting 34 cells with this envelope now? And the latest version tells us, yes, we can get there, and now we're getting results that are starting to get on par with BAVTR and very different from a VSVG vector. So the potency is almost there. How about the specificity in vitro? We are testing different panels. I'll just show you a brief comparison, a quick comparison of 34 cells either stimulated or not, and T cells stimulated or not. If you use a BAVTR or a VSVG, you can get into both cell types, but if you use 133, you are clearly specifically eating the 34 cells and almost not at all the T cells. We're also doing spiking experiments, and it works very well in that context too. So here you start to avoid major cellular sinks in the peripheral blood. What if we test this vector in vivo? So in the day 7 humanization model, the story is complicated. This is what you infuse. 97% of the cells are 133 positive, of the 34 cells. But if you wait 7 days, already a lot of these cells become negative. And the MFI of these cells, the 133, is also lower than the original material. So the density of the receptor is not the same as the one you would have in a human. And if you wait for 12 days, time of the takedown, you also, again, lost half of the 133 positive cells. So it's a moving target in this particular scenario. So nonetheless, we tested 2 groups, BAVTR, a broad fusogen, and CD133. At day 12, they all have the same composition. If you gate on total human cells, more or less 18–20% of them are 133 positive. But if you gate on GFP positive cells, then you start to see some enrichment, whereby the CD133, you have 36% of the GFP positive cells that are 133 positive, while only 12% of the BAVTR cells are 133 positive. Now consider that from the moment of injection to the day 12, you have lost 133 expression, so probably the specific targeting is even higher, but a lot of the cells now have become 133 negative by the time of analysis. As a result, you have an enrichment for lineage negative 34 cells and for 133 cells in the 133 vector, while this is not the case with BAVTR with a broad fusogen. Now, the last piece of data is, and this is my last data slide, is about another fusogen that we are developing. This is for 117. And we can use, in this case, the long-term humanized mice because they carry a very stable population of 117 high cells in the bone marrow. The point is that this population is a tiny fraction of the human cells, is 0.3% of the total human cells, so is the needle in the haystack, and is primarily and only in the bone marrow. So the vector has to make all the way from the peripheral blood, finding these few cells in the bone marrow and transduce them. So we tested again this 117 fusogen versus a BAVTR, and the results were amazing. When I saw these results, I said, okay, this platform works. Where essentially if you gate on GFP positive cells, you clearly see that the majority of these cells are 117 positive, while instead if you gate on GFP positive cells using a BAVTR vector, you pretty much have the same representation of 117 positive cells as you have in the total 45. So as a result, the transduction levels was very low in the rest of the human cells, but it was like we eat about 7% of the target population, which corresponds to a 175-fold enrichment for your target cell population. And this was not shown at all as an enrichment. Sorry, this is I don't know what happens to the slide. This is a conversion, MAC-PC conversion. This is total human cells, and these are 117 positive cells. So clearly you don't have enrichment for the 117 positive cells in the BAVTR, and you're clearly enriching for these cells with the 117 specific fusogen. So in conclusion, I showed you that we are working on the three main pillars of an in vivo delivery. So we are developing systems that are able to get into these cells in their steady state. We are developing and we are getting good results now of editing in vivo HSPCs in different compartments using humanized mouse models in different versions. And now we are also achieving the results that we are starting to avoid cellular sinks and getting more specifically into HSPCs. And with that, I'd like to thank my team at Sana, particularly Sean, Anjali, Vlad, Casey, the rest of the leadership team, Jagesh, Doug, Ed, and particularly these guys here. These guys are phenomenal. These are vector production team, Kai Tudor, Kelsey, they optimized the BAVTR, Matteo, Stoppato, Alex, Justin, they worked on the 133 project, Fumi Adevale for the Fluor Core facility, and all of you for your attention. Thank you.
Thank you very much, Luca, for this exciting data. Thank you very much. I have a quick question regarding, was there any reason why you opted for the BioVar TR instead of BioVar-less? Because looking at your slides, we had the same about the results using the BioVar-less with the MOI 50 was about 65% transduction,
yes,
which is similar.
We are working on different platforms. We're also testing that. In our hands, in the particular condition we use in vitro, we never tested in vivo, I gotta say. We didn't see such a difference with BAVTR. It's also a matter of means of production. Depending on your producer cell line and the way you produce the vector, certain envelopes in your hands, in your lab, might just work better. In our hands, with our production system, BAVTR is still a little bit ahead of BAVR-less.
How was the title?
Well, we get, I can tell you, well, I don't know if I can tell that. What I can tell is we get around E9 in primary cell lines. And what I can tell is the results that we obtain in vivo, this 6% that we get in the 7-day model, they are obtained with infusing around 6 E8 TUs per mouse, which correspond around E10 physical particles per mouse. And again, this is a question, if I can take a chance, to put things in perspective. When you use an LMP and you are getting like 70% transduction of 34 cells with LMPs, usually in those systems, by a back calculation, they are infusing around E16 particles, physical particles per mouse. We get results now around 40% with 6 log less. vector. This has a certain relevance in terms of like translatability of the approach. Although, of course, our vectors are more difficult to produce.
Okay, thank you.
That was a very good question. So your in vivo data are scary good. I mean, that's really. Once you activate one microphone, the other one gets deactivated. So your in vivo data are really scary good. I mean, that's such high efficiency after the triple dose, and for CD117, which is a very low percentage cell population without mobilization, as I understand it. So two questions. So one, the triple dose after mobilization and so on. So how does the cost compare? What is the amount of material you have to put in there if you want to translate this? So does it become very expensive to do this? And the other question is, given your result for non-mobilized efficiency, if you do a triple dose there, would that be an approach to do in vivo editing without mobilization?
Okay, these are very good questions. So first of all, when we did three doses, it's essentially 3 times what I just said. It's 3 doses of the same 6E8 per mouse. So you have to do the math in terms of the vector that you would need. But yeah, it's still a relatively big dose, but it still logs away from what you would dose in terms of LMPs. In terms of mobilization, the best results we always obtain with mobilization, even when the cells are technically not mobilized, they don't go into peripheral blood, just because you make them more accessible within the bone marrow. So what we think is that right now we're thinking for a potential translational application to still use mobilization, because of course it gives a slightly better chance to eat the cells in the peripheral blood, but also that you make the cells more accessible in the bone marrow, within the bone marrow. So I think the best data we obtain are also always with mobilization.
Okay, may I? That was great. My question actually is kind of overlapping what Karsten has asked. So it comes to mobilization. First, is there a specific reason that you opted for plerixafor alone instead of the combination, so you get rid of the GCSF? And the other is that, you are using 3 rounds of mobilization and 3 infusions, right? And mobilization is with plerixafor, and people have shown that repeated plerixafor mobilization may result actually in less CD34 positive cell yield. How do you explain the increase you have in your readout, the outcome readout? Yeah.
Okay.
Okay. Yeah.
So, first question, I have 3 answers. One is practical, one is biological, and the other one is translational. Practical. This helped our in vivo team because they were dosing GCSF during weekends. And MD31, when we moved to MD31, they were so pleased. They said, finally we can get our weekends back. Second, biological. GCSF mobilize and activate a lot of myeloid progenitors that then will exhaust. So you think you're eating a lot of cells, but then eventually you're eating a lot of short-term progenitors. You have this nice initial boost, but it doesn't translate necessarily to a long-term, eating long-term HSCs. Translationally, plerixafor alone is more suitable for application like sickle cell disease. So we think that for these 3 reasons, it was more reasonable to use MD3100 alone. The second question was about, don't tell me you forgot.
I forgot also.
Repeated doses of plerixafor, right?
3 doses.
Wow, talking about short-term memory. Yes. Actually, our data are showing that at least in this humanized model, you have a substantial expansion of HSCs after 3 doses. We measure absolute counts by counting beats. in the bone marrow, and we did the comparison with or without G-CSF, with 1 dose or 3 doses of plerixafor alone. And actually, the true lineage negative, 34 positive, 38 negative, 90 positive cells, so the very HSC niche cells are actually more. Then, of course, if you are adding G-CSF, you're driving differentiation. That's why you lose 34 cells. So plerixafor has the advantage also to preserve a little bit the self-renewal of the long-term HSCs. So we think, we are confident that this might work.
My only, let's say, objection is that plerixafor usually works by detaching CD34 positive cells from the bone marrow to the periphery, and does not have a great result on self-renewing. And is actually the combination that allows stem cells not only to renew but also to mobilize, egress the bone marrow.
Yeah, phenotypically we see the opposite, but it's a point well taken. We are actually now conducting a long-term study where we are not sucking the mice just 12 days after dosing, but we are keeping the mice alive so we can directly address your point.
Okay.
But it's well taken.
Thank you.
Okay.
Another one question, sorry.
Oh.
Did you observe any immune response for the in vivo?
These are humanized mouse models. That's a limitation of the model. You cannot measure that, of course. We think that eventually the best suited scenario for that is going into NHP, which we hope is going to happen around next year, to address that question.
Okay, thank you. Thank you very much. We'll come to the end of the session. I'd like to thank all the speakers for their excellent presentation and the audience for the participation. Please be back in 15 minutes after the break for the plenary session.
So, ideally at 4:40, huh?
the contrary, on IPS is nicer because you can create, you can do base editing, and you can transpect them again theoretically. While the results are gone after a while. So, but you have most importantly is the notation that you need to check. But otherwise, we set up the protocol also in T cells, and it's working very well because you use the T cells of the patient to screen for guided RNA because we don't have the long mutation, and it works very well in T cells, in T cells also. Because of the version printed in Julien Zuber.
πίσω, αλλά όχι. Λοιπόν, σας καλωσορίζουμε στην κεντρική συνεδρίαση της εταιρείας μας, η οποία θα παρουσιαστούν οι καλύτερες εργασίες, οι οποίες έλαβαν τη μεγαλύτερη βαθμολογία μαζί με τις δύο βραβευμένες εργασίες του συνεδρίου. Εδώ έχουμε μια ιδιομορφία. Η διάρκεια των ομιλιών θα είναι 7 λεπτά, με 2 λεπτά για ερωτήσεις. Και η παρουσίαση μπορεί να είναι είτε ελληνικά είτε αγγλικά, αλλά με αγγλικά τις διαφάνειες. Κύριε Ρουπελάκη.
Καλημέρα και από μένα. Σας καλωσορίζουμε στη συνεδρία των προφορικών ανακοινώσεων. Θα ξεκινήσουμε με την πρώτη παρουσίαση. Ο τίτλος είναι Diverse Immunogenetic Profile of Pathogen-Specific Cells in the Context of Adaptive Immunotherapy από την ομάδα της κυρίας Γιαννάκη και παρουσιάζει η κυρία Πανταζή.
Πρέπει να πατήσω κάτι. Καλησπέρα σας. Εγώ σήμερα θα σας παρουσιάσω αποτελέσματα αλληλούχησης του ταύ κυταρικού υποδοχέα παθογονοειδικών ταύ κυταρικών προϊόντων ανοσοθεραπείας. Στο πλαίσιο της αλλογενούς μεταμόσχευσης αιμοποιητικών κυτάρων, η οποία αποτελεί τη μοναδική μέχρι σήμερα ριζική θεραπεία της λευχαιμίας, οι σημαντικές αιτίες αποτυχίας είναι 3. Οι ευκαιριακές λοιμώξεις, η υποτροπή της λευχαιμίας και η νόσος μοσχεύματος κατά του ξενιστή. Οι κυταρικές θεραπείες με μη τροποποιημένα ταύ λυμφοκύτταρα που αναγνωρίζουν εξειδικευμένα αντιγόνα παθογόνων ή σχετιζόμενα με τη λευχαιμία αντιγόνα έχουν αναδειχθεί τα τελευταία χρόνια ως πολλά υποσχόμενες εναλλακτικές, εξατομικευμένες και φυσικές μορφές θεραπείας των ευκαιριακών λοιμώξεων και της υποτροπής λευχαιμίας αντίστοιχα. Ενώ παράλληλα δεν παρουσιάζουν αλλοδραστικότητα. Για την παραγωγή των παθογονοειδικών ταύ λυμφοκυττάρων, αρχικά απομονώνουμε μονοπύρηνα κύτταρα από την περιφέρεια του δότη του μοσχεύματος, τα οποία διαγείρουμε με τα αντιγόνα στόχους και εκτίσουμε τα ειδικά κύτταρα in vitro για 10 μέρες. Το προϊόν εχείεται στον ασθενή όταν εμφανίσει λίμωξη με το αντίστοιχο παθογόνο. Τα παθογονοειδικά ταύ λυμφοκύτταρα αναγνωρίζουν εξειδικευμένα τα αντιγόνα μέσω του ενδογενούς ταύ κυταρικού υποδοχέα τους, ο οποίος αποτελεί ένα διαμεμβρανικό υποδοχέα, είναι ετεροδιμερές και αποτελείται ως επιτοπλίστων από 2 αλυσίδες, μία α και μία β, οι οποίες ενώνονται και δημιουργούν 2 τμήματα, το σταθερό τμήμα και τη σταθερή περιοχή και τη μεταβλητή περιοχή, στην οποία υπάρχει και το σημείο πρόσδεσης των αντιγόνων, όπου παρουσιάζονται από τα μόρια του μείζωνος συμπλέγματος ιστοσυμβατότητας. Για την παραγωγή των ταύ κυταρικών υποδοχέων γίνεται ανασυνδυασμός. Συγκεκριμένα, για την παραγωγή της μεταβλητής περιοχής της β' αλυσίδας γίνεται VDJ ανασυνδυασμός. Συνδέεται ένα V γονίδιο με ένα D και ένα J, και αυτά συνδέονται με ένα από τα 2 σε γονίδια που αντιστοιχούν στη σταθερή περιοχή της αλυσίδας. Ενώ για την για την α αλυσίδα, για τη μεταβλητή περιοχή γίνεται ένας VJ ανασυνδυασμός, ανασυνδυάζεται ένα V με ένα J γονίδιο και σε αυτή την περίπτωση έχουμε μόνο ένα γονίδιο σταθερής περιοχής. Και στις 2 αλυσίδες, στη μεταβλητή περιοχή, έχουμε 3 υπερμεταβλητές υποπεριοχές, με την CDR3 να είναι η κύρια για την αναγνώριση, για τον καθορισμό της συμπληρωματικότητας. Η ετερογένεια και η ποικιλομορφία των ταύ κυταρικών υποδοχέων είναι αποτέλεσμα αυτών των ανασυνδυασμών και ενισχύεται από μικρές προσθήκες ή ελλείψεις νουκλεοτιδίων στα σημεία ένωσης των γονιδίων. Γεγονός που επιτρέπει στο νοσοποιητικό σύστημα να αναγνωρίζει πληθώρα αντιγόνων. Στόχος μας είναι ο χαρακτηρισμός του ρεπερτορίου των γονιδιακών αναδιατάξεων της β' αλυσίδας του ταυκυταρικού υποδοχέα σε παθογονοειδικά ταυκυταρικά προϊόντα που παράγουμε από υγιείς δότες σε τρέχουσες κλινικές μελέτες για την αντιμετώπιση των λοιμόξεων μετά από αλλογενή μεταμόσχευση. Τα κυταρικά αυτά προϊόντα είναι τα 3 VSTs που έχουν ειδικότητα σε 3 ιούς, τον κυταρομεγαλό, τον Epstein-Barr και τον BK-ιό, και τα 5 STs, τα οποία στοχεύουν επίσης τον άδενο-ιό και τον Myceta aspergillus fumigatus. Παράλληλα, αναλύσαμε και μονοπύρηνα κύτταρα από την περιφέρεια των ασθενών πριν και μετά την έγχυση των παθογονοειδικών κυττάρων. Επειδή τα κυταρικά προϊόντα που παράγουμε είναι πολυδύναμα, θέλαμε να αναλύσουμε όχι μόνο το ολικό προϊόν, αλλά και τους επιμέρους υποπληθυσμούς ειδικών κυττάρων. Για το σκοπό αυτό, πραγματοποιήσαμε ανοσομαγνητικό διαχωρισμό για να απομονώσουμε τα κύτταρα που εκρίνουν ιντερφερόνι Γ μετά από διέγερση με τα αντίστοιχα πεπτίδια στόχους. Το ίδιο κάναμε και για τα 5 STs. Για την ανοσογεννητική ανάλυση του ρεπερτορίου του ταυκυταρικού υποδοχέα, αρχικά πραγματοποιήσαμε απομόνωση RNA, σύνθεση cDNA και ενίσχυση με multiplex PCR των γονιδίων της β' αλυσίδας του ταυκυταρικού υποδοχέα. Κατασκευάσαμε βιβλιοθήκες κατάλληλες για αλληλούχηση νέας γενιάς με ειδικό πρωτόκολλο και τέλος πραγματοποιήσαμε βιοπληροφορική ανάλυση με επικυρωμένα εργαλεία, ορίζοντας ως κλονότυπο αλληλουχίες με το ίδιο V γονίδιο και επανομοιότυπη CDR3 αμινοξική αλληλουχία. Τα 3 και 5 STs εμφάνισαν ένα πολυκλονικό ρεπερτόριο ταυκυταρικών υποδοχέων με τη μέση τιμή των διαφορετικών κλονοτύπων αναδείγμα να είναι περίπου στις 22.000 και τη συχνότητα του κυρίαρχου κλονοτύπου, δηλαδή αυτού με τη μεγαλύτερη συχνότητα, να μην υπερβαίνει το 20% σε κανένα από τα κυταρικά προϊόντα. Για να χαρακτηρίσουμε έναν κλονότυπο ειδικό, θα έπρεπε να εμφανίζεται σε ένα μόνο εμπλουτισμένο υποπληθυσμό, με πάνω από 10 διαβάσματα και υψηλότερη συχνότητα σε σχέση με το υλικό προϊόν. Οι συνολικοί αριθμοί των κλονοτύπων που χαρακτηρίσαμε ως ειδικούς φαίνονται στο διάγραμμα και αν λάβουμε υπόψη το μικρό αριθμό δειγμάτων που αναλύσαμε, ο αριθμός τους είναι αρκετά μεγάλος. Αξιοσημείωτα, οι ειδικοί κλονότυποι ανιχνεύτηκαν σε PBMCs ασθενών έως και 15 εβδομάδες μετά την έγχυση των 3 VSTs, των οποίων η παρουσία μάλιστα συσχετίστηκε αρνητικά με το οικό φορτίο και θετικά με τον αριθμό των κυκλοφορούντων παθογονοειδικών ταφ κυττάρων. Όπως χαρακτηριστικά βλέπουμε στον ασθενή 1, στον BK, το φορτίο του ιού, που φαίνεται με την κόκκινη γραμμή, μειώνεται όταν αυξάνεται η συχνότητα των κλονοτύπων που χαρακτηρίσαμε ως BK ειδική, που βλέπουμε με την μοβ σκίαση. Με την ίδια τάση αυξάνονται και τα κυκλοφορούντα BK ειδικά κύτταρα που βλέπουμε με τη μοβ γραμμή. Συμπερασματικά, τα 3 VSTs και 5 STs χαρακτηρίζονται από ένα πολυκλονικό ρεπερτόριο ταυκυταρικών υποδοχέων με υψηλή ποικιλομορφία και παρέχουμε μία καινοτόμο μέθοδο ιχνηλάτησής τους in vivo. Επίσης, παρέχουμε ένα πάνελ παθογονοειδικών ταυκυταρικών υποδοχέων που μελλοντικά μπορούν να χρησιμοποιηθούν τόσο για τον προσδιορισμό του νοσολογικού κινδύνου για ανάπτυξη λοιμόξεων, όσο και για την επιλογή βέλτιστων ταυκυταρικών υποδοχέων για υιοθετούμενη ανοσοθεραπεία και κατάλληλων υποψηφίων για ανοσοθεραπεία στο πλαίσιο αλλογενούς μεταμόσχευσης. Ευχαριστώ πολύ.
Ευχαριστούμε πολύ την κυρία Πανταζή. Ερωτήσεις από το ακροατήριο? Ευχαριστούμε πολύ. Αν δεν έχουμε ερωτήσεις, να προχωρήσουμε στην επόμενη ομιλία. Από την ίδια ομάδα της κυρίας Γιαννάκη, ο τίτλος είναι «Safety and Efficacy of Pentavalent Specific T-cells for the Treatment of Opportunistic Infections Post-Haploidentical Transplantation Παρουσιάζει η κυρία Μπούσιου.
We will present our results of the study examining the safety and efficacy of penta-STs for the treatment of opportunistic infections. As we know, opportunistic infections present a major cause of morbidity and mortality after allogeneic transplantation, and conventional therapies are limited. They have major toxicities and potentially resistance development. So adoptive transfer of pathogen-specific T cells has recently emerged as an appealing alternative strategy. The objective of our study is to evaluate the safety and efficacy of donor-derived specific T cells targeting CMV, EBV, BK virus, adenovirus, and Aspergillus fumigatus. For that reason, we draw a small amount of blood from our donor who has to be seropositive for at least one of these pathogens. Mononuclear cells are incubated with peptides specific for the antigens, and 10 days later, after proper stimulation, we get the expanded cell product. The infusion is made at a dose of 20 million cells per square meter in haplotransplanted patients previously treated with high-dose post-transplant cyclophosphamide, who have an infection from one of the targeted pathogens. In order to be included in the study, patients must receive no or small doses of corticosteroids at the time of infusion. To the results of our study, up to date we have created 11 products in our GMP unit. Final products were expanding approximately 5.4 times fold over baseline, reaching an average of 162 million cells, as we can see in the chart. They are enriched in CD3 cells containing both CD4 and CD8 and expressing mainly central and effector memory markers. Regarding specificity, all products were specific for the antigen the donor was seropositive to. We have only 2 products that were not specific for CMV because the donor was seronegative, and the same for 2 products regarding Aspergillus fumigatus. Infusions. Infusions were made to 6 patients at a median time of 90 days post-transplant. We had 4 patients receiving low dose of corticosteroids, 3 for GVHD and 1 for cyclosporin toxicity. We can see in the table the 6 patients. 4 of them had only 1 infection, CMV in the first patient, EBV in 2 of them, 1 of them having also been diagnosed with EBV PTLD. 1 patient had BK virus cystitis. 1 patient had simultaneously 2 pathogen infection, and 1 had a triple infection, both from CMV, EBV, and BK. We can see here the clinical response of 1 patient who had an infection from CMV. We can see that the CMV load is rapidly declining as the CMV specific cell rise. And 20 weeks later, we can see at the end of the chart that the CMV load starts to arise, but CMV cells also expand, so the patient didn't have the CMV reactivation at last. Same image here for EBV and also for BK. Regarding response, penta-ST infusion was safe. We had no infusion-related toxicity or GVHD occurrence attributed to the cells. The best response was achieved at 3.5 weeks post-infusion and correlated with the expansion of circulating penta-STs. Patients also received drug therapy according to guidelines. And we can see in the table that all of the patients achieved complete response to the infusion, except for the 3rd patient who had an EBV infection with CNS involvement, who had lesions in the MRI after the infusion of the product. These cells can offer also long-term prophylaxis against the infections. With a median follow-up of 5.5 months, we had no reactivation from the targeted pathogens. We had only 2 reactivations in patients for viruses other than they had received the cell for. We had 1 patient reactivating CMV, but the product he had received was from a seronegative donor, so it wasn't specific. And we had an EBV reactivation in a patient with poor in vivo expansion of the EBV T cells, who later received the second infusion. And we can see here the results for a patient who initially received the product for EBV infection. 10 weeks later, he has a CMV reactivation in the blood, but the load rapidly declined as the CMV-specific cells expanded. This patient had resolution of the reactivation without receiving drug therapy for the CMV reactivation. As a conclusion, we can say that pathogen-specific T-cell therapy is feasible and safe, especially for patients at high risk for opportunistic infections after allogeneic transplantation, potentially minimizing the need for drug therapy and preventing reactivations. Thank you very much.
Ευχαριστούμε πολύ για την πολύ ενδιαφέρουσα παρουσίαση και που ήσασταν και πολύ ακριβείς στο χρόνο σας. Ερωτήσεις από το ακροατήριο?
Αυτό που είχατε πει, 162 εκατομμύρια, είπατε εκεί τώρα. Ναι. Μπορεί να φτάσει και περισσότερο με τις διαργασίες που μπορεί να κάνετε.
Να σας πω την αλήθεια, δεν είμαι ο κατάλληλος άνθρωπος να σας το απαντήσω, γιατί δεν είμαι στο εργαστήριο. Δεν ξέρω αν μπορεί να φτάσει παραπάνω.
Ωραία. Ναι. Πώς?
Όχι, γιατί.
Ναι. Και ο ανασωφενότυπος αυτός είναι σταθερός.
Ναι, είναι ποσοστή αναλογία στο προϊόν.
Okay. Yeah. Karsten. Oh, yes.
Okay, yes, we can, but...
Okay.
Σας ευχαριστούμε πολύ.
Ευχαριστώ και εγώ.
Let's move to the next talk. Ας προχωρήσουμε στην επόμενη ομιλία. Genetic manipulations of Lima-1 expression delineate its role as a molecular therapeutic target for cervical carcinoma από την ομάδα της κυρίας Παπά και του κυρίου Ανάγνου παρουσιάζει η κυρία Δρακοπούλου.
Καλησπέρα σας. Good afternoon. So I'm going to keep the English version. So I'm going to talk about cervical cancer. So cervical cancer is the most common and lethal type of cancer in women of reproductive age. Previous studies by our team have highlighted the differential expression of various proteins between normal ATK1T keratinocytes and cancer cell lines such as SIHA, HeLa, and C33A. Among those proteins, Lima-1 or eplin was found to be significantly increased in normal keratinocytes compared to the C33A cancer cell line. What is Lima-1? Lima-1 is a cytoskeleton protein whose expression is often reduced or lost in various cancer types such as prostate cancer or breast cancer. It is encoded by Lima-1 gene found on chromosome 12, and it exists in two isoforms, a smaller one, eplin alpha, and a bigger one, eplin B. Studies in cell lines that normally express either of these isoforms showed that eplin-1 is mainly located in the periphery of the cell. And when it comes to our cell lines, the normal ATK1T and C33A, you can see that there is the Lima-1 the expression of Lima-1 is higher in the normal ATK1T cell line, and specifically, eplin alpha isoform is the expression of alpha isoform is greater in ATK1T cells, both compared to the B isoform, but also compared to the expression of those isoforms in the cancer cell line. So what was the aim of this work? The aim of this work was to further investigate the role of Lima-1 in cervical cancer, mainly by three approaches. First, by knocking down eplin in the normal keratinocytes, HCK1T cells. Secondly, by overexpressing LIMA1 in the cancer cell line C33A. And third, by performing an immunohistochemical analysis of 89 samples that cover the whole spectrum of cervical carcinogenesis. So, in order to knock down LIMA1 in HCK1T cell line, we employed shRNA technology, so we produced SH LIMA1 lentiviral vector together with the control no target vector and proceeded in HCK1T transduction MY25. Following selection with pyrimycin, we went on performing different assays such as proliferation, migration assay, invasion, and wound healing assay. First of all, we wanted to verify that transduction with SH LIMA1 lentiviral vector would lead to efficient knockdown of LIMA1. As you can see here, transaction with our shRNA, SH LIMA1 lentiviral vector led to a 92% decrease of LIMA1 expression, and that applied to both alpha and beta isoforms, as you can see there. We verified the efficient LIMA1 knockdown both at protein level by doing confocal microscopy, and as you can see, the intensity of LIMA1 expression is significantly reduced. I hope you can see that there is almost no LIMA1 expression there. And when you do the stats, the above knockdown was reduced by, the expression was reduced by 45%. We then went on to see what happens with migration and invasion. So following, knockdown of LIMA1 expression led to a significantly increased, primarily invasion at 24 hours, but as you can see, migration is also increased. And the same applied to the wound healing capacity. When LIMA1 was knocked down, you can see that cells were able to close the gap quite quicker. Within 8 hours, and the percentage of wound closure was 75% after 3 independent experiments. So far we managed to see that knocking down LIMA1 induces cancer characteristics in a normal cell line. However, we failed to show an increase in the proliferation following LIMA1 knockdown. As you can see here, and following 6 experiments, the proliferation in both cases is almost the same. We then wanted to delineate the molecular mechanisms that are responsible for the induction of those characteristics, so we proceeded to RNA sequencing. So we observed that there is a quite big number of genes whose expression is with differential expression. In particular, and as you can see in blue, there were genes whose expression was significantly decreased following LIMA1 knockdown, and those genes were related to cell cycle as shown by DSEA. In red, you can see the genes whose expression was upregulated following LIMA1 knockdown, and those genes were found to be involved in the epithelial to mesenchymal transition, EMT, and this, which precedes cancer metastasis, and the specific process is something that we're currently focuses on. We've done the knockdown experiments in the normal keratinocytes. Now we went on to carry on the opposite experiment. So we overexpressed LIMA1 in the cancer cell line C33A. For this reason, we employed the plasmid vector, and we went on to transfecting C33A cells using lipofectamine. We selected the cells using pyrimycin and went on performing the same assays as before. As you can see here, transfection with a specific plasmid, overexpressed eplin, and specifically the alpha isoform, to nearly six-fold. When it comes to migration and invasion, we observed reduced migration and invasion at 24 hours, while when it comes to wood healing capacity, at least for the time point up to 24 hours, we didn't observe any difference. As you would expect, proliferation was significantly reduced when LIMA1 was overexpressed, possibly due to a higher percentage of cells in the G0-G1 phase and a smaller, lower proportion of cells at D2M phase of the cell cycle. Last but not least, we do immunohistochemical analysis in samples that cover the whole spectrum of cervical carcinogenesis using anti-eplin antibody. The type and number of different samples that we've used are shown in the are shown here. So first, we verified the high LIMA1 expression in normal cervical epithelium. As you can see, there is a high, in both images, there is positive staining and high intensity. And when it comes to cervical intraepithelial neoplasia, CIA1, we observed that there a correlation between eplin-1 expression and CIN stage. So LIMA1 expression was increasing as the stage was progressing. When it comes to cancer, there, as you would expect, we've observed a decreased expression of LIMA1. Most of our samples were positive for LIMA1. However, the intensity of LIMA1 staining was lower than in the case of normal cells. So overall, there is a unique pattern of LIMA1 expression during cervical carcinogenesis. There is high LIMA1 expression in normal cells, and then LIMA1 expression reduces when we go to CIN1. There is a correlation between CIN stages, and then we see reduced expression of LIMA1 in cancer. So to summarize, knockdown of LIMA1 expression in normal keratinocytes induces cancer characteristics such as increased migration, invasion, and wood healing capacity. Also, it leads to reduced expression of genes involved in cell cycle and increased expression of genes involved in EMT. When it comes to what happens when we overexpressing LIMA1, there we observed reduced migration, invasion, and proliferation, primarily due to prolonged G0, G1 phase. What we observed from the immunocytochemical analysis, we showed that there is correlation between LIMA1 expression and CIN stages, and also reduced expression in cancer cervical epithelium. Taken together, we can say that downregulation of LIMA1 plays an important role in the initiation, progression, and metastasis of cervical cancer, and can be used as a prognostic marker or a potential therapeutic target. Thank you very much for your attention.
Thank you very much for the interesting data. Any question? There is time for just one question. Can I ask you something very... are there any data on LIMA1 expression from in vivo models, from in vivo cancer models?
Not yet.
Not yet.
Not yet. So this is something that we are planning to do, and obviously this would give us a lot more information of how LIMA1 could be used therapeutically.
Okay, because you need somehow to correlate the low expression of LIMA1 that doesn't affect proliferation, whereas upregulation of LIMA1
There
reduces.
is an explanation
Yeah.
for that, I think. It has to do, apparently, eplin alpha isoform is the key player. So in C33A cancer cell line, the eplin alpha isoform was quite low, and we overexpress it using the plasmid vector. In the normal keratinocytes, the endogenous alpha isoform was already very high. So by using the shRNA, of course, we did reduce the expression. However, it was very high to start with. So must be must be an explanation.
Thank you very much. Thanks.
Thank you.
Yes, sure.
Thank you. We knocked down Lima-1 in normal, and we overexpressed it in cancer cell line. So we did the, yeah. Okay, thank you so much.
Okay, we move to the fourth presentation by Dr. Pasudi of the Dr. Yanakis group, in vivo base editing to induce -113 adenine to guanine HPFH mutation results in deficient hemoglobin F reactivation in humanized mouse models for beta hemoglobinopathies. Dr. Pasudi will present that.
Hello. Today I will present our work regarding in vivo base editing for HPF reactivation in humanized mouse models. The conventional treatment for beta hemoglobinopathies includes blood transfusion, iron chelation, and pharmaceutical induction of gamma globin in the case of sickle cell disease, with hematopoietic stem cell transplantation being the only curative option. The great advance in gene therapy field enables the development of innovative therapeutic strategies, including the gene addition of anti-sickling beta globin or gamma globin gene, as well as the application of genome editing strategies in order to that either induce gamma globin reactivation by blocking repressive mechanisms, or allow the HDR-based in situ correction of sickle cell disease mutation. Despite the quite promising results of clinical and preclinical genome editing applications, concerns and limitations still exist due to the generation of potentially harmful off-target effect and on-target consequences related to the induction of double-strand breaks. The recently developed base editor seems to be a safer and effective approach for installation of base substitution. There are two main categories of base editors, cytidine and adenine base editors, that catalyze the conversion of cytosine to thymine and adenine to guanosine, respectively. Avoiding the induction of double-strand breaks, base editors allow the installation of disease-modifying mutations. Over the last few years, in collaboration with Dr. Lieber's lab in the University of Washington, we have developed an in vivo gene therapy approach based on in vivo transduction of mobilized hematopoietic stem cells by helper-dependent adenoviral vectors. This vector targets hematopoietic stem cells after the recognition of CD46 receptor on their surface. This approach would be ideal to deliver base editors into hematopoietic stem cells due to increased payload capacity and episomal transgene expression that increase the safety profile of the approach. Aim of the study is the ex vivo and in vivo reactivation of gamma globin expression via introduction of the -113A to G HPF8 mutation in CD34 positive cells from beta thalassemia and sickle cell disease patients using a non-integrated adenoviral vector encoding an adenine base editor. The vector also contains a O6BG BCNU resistant gene for enrichment of edited cells. Following 48-hour transduction with adenoviral vector encoding the adenine base editor, CD34 positive cells from thalassemia and sickle cell disease patients were treated or not with O6BG BCNU and seeded in erythrodifferentiation medium. As you can see, selection of transduced cells with O6BG BCNU resulted in 60% conversion rate, both in thalassemia and sickle cell disease cells, and the conversion rate remained stable during erythrodifferentiation. The increased conversion rate was also translated into significantly higher percentage of HbF positive cells, both in thalassemia and sickle cell disease group, with the difference being higher in the case of selected cells, in the total cell population, and in enucleated cells. These results was also confirmed by HPLC, where we also observed increased production of gamma globin chains. This increase in HbF levels resulted in marked phenotypic correction, as we observed an apparent reduction of oxidative stress levels and improved cell morphology at the end of erythrodifferentiation. Next, we developed a humanized disease model. After the transplantation of CD34 positive cells from thalassemia and sickle cell disease donors into NBSW mice, 6 weeks after transplantation, the mice were successfully mobilized, and at the end of mobilization, we administrate the adenine base editor vector in all recipients. And 1 week later, the mice were treated or not with O6BG BCNU, and 20 weeks after transplantation, the mice were sacrificed. After bone marrow analysis, we didn't observe any differences in engraftment rate and multilineage reconstitution after in vivo transduction and selection with O6BG BCNU. Interestingly, we observed increased rate of HbF positive cells within the GLIA positive population of the chimeric bone marrow, with the difference being higher in the case of mice that received both adenoviral vector and in vivo selection in THAL humanized model and in sickle cell disease model. After ex vivo erythroid differentiation of the chimeric bone marrow, again, we observed increased percentage of HbF positive cells, again in the group that received viral vector and the in vivo selection. Further analyses are ongoing, including the evaluation of the editing rate in vivo, the HPLC analysis of the GLIA positive population in the chimeric bone marrow, and of course we have already performed secondary transplantation in order to estimate the engraftment rate and HbF levels in secondary recipients. Overall, both ex vivo and in vivo transduction in humanized thalassemia or sickle cell disease models with helper-dependent adenoviral vector encoding the adenine base editors induce the HbF-like mutation and reactivate HbF in clinical relevant levels. As well as we observed improvement of the disease phenotype. Therefore, base editing for beta hemoglobinopathies has the potential to be an alternative, safe, and efficient therapeutic approach for either ex vivo or in vivo gene therapy. Thank you for your attention.
Thank you, Dr. Pasud, for being on time and for the nice work. In your in vitro experiments, the evaluation of the phenotype by using, did you look for apoptosis, for the improvement of apoptosis, or just used for the ROS only.
Sir, could you please repeat the question because I didn't hear the...
Did you check for any parameters for improving the phenotype in vitro following the transfer? Yeah.
Until now, no. But we have to check all this that I mentioned before.
Okay. Any other questions? Okay. Thank you very much. Okay, we move now to the fifth presentation. A novel gene therapy vector for beta hemoglobinopathies with a selective expansion cassette by the group of Dr. Psathan. Dr. Kirtsu will present.
Καλησπέρα και από μένα. Εγώ θα σας μιλήσω για το σχεδιασμό και τον έλεγχο ενός νέου οικού φορέα θετικής επιλογής για τις β' αιμοσφαιρινοπάθειες. Η ex vivo γονιδική θεραπεία έχει δώσει την απάντηση σε πληθώρα γενετικών αιμοποιητικών νόσων και περιλαμβάνει την κινητοποίηση των στελεχαίων αιμοποιητικών κυτάρων του ασθενή και τη συλλογή τους από το περιφερικό αίμα έπειτα από την κινητοποίηση. Στη συνέχεια τα κύτταρα καλλιεργούνται και τροποποιούνται γενετικά ex vivo πριν την επαναφορά τους στον ασθενή. Συγκεκριμένα, οι β' αιμοσφαιρινοπάθειες έχουν χρησιμοποιηθεί ως μοντέλο για τη γονιδιακή θεραπεία σε στελεχαία αιμοποιητικά κύτταρα με πολύ θετικά μέχρι στιγμής αποτελέσματα. Ωστόσο, υπάρχουν ακόμα σημαντικοί περιορισμοί σε ό,τι αφορά τους οικούς φορείς που χρησιμοποιούνται. Οι οικοί αυτοί φορείς έχουν μεγάλο μέγεθος, το οποίο οδηγεί σε χαμηλή παραγωγή οικών τίτλων, μειωμένη διαμολυσματικότητα στα στελεχαία αιμοποιητικά κύτταρα και πολύ συχνά σε αποσιώπηση του θεραπευτικού διαγωνιδίου μετά τη μεταμόσχευση ή πιθανή εισαγωγή εισχωρητικής μεταλλαξιγένεσης, καθώς στους περισσότερους από αυτούς τους οικούς φορείς δεν προστίθεται κάποιος μονοτής τύπου φραγμού. Για να ξεπεραστεί το πρόβλημα της μειωμένης διαμολυσματικότητας και να αυξηθεί το ποσοστό των γενετικά τροποποιημένων κυτάρων, μέχρι τώρα χρησιμοποιούνται κάποιες μέθοδοι, οι οποίες βασίζονται στην αρνητική επιλογή των μη διαμολυσμένων κυτάρων μέσω χημειοθεραπείας. Ωστόσο, υπάρχει και μια άλλη μέθοδος με την κασέτα του F36VMPL, με την οποία μπορούμε να πολλαπλασιάσουμε τα γενετικά τροποποιημένα κύτταρα, χωρίς να προκαλείται κυταρικός θάνατος στα μη επιδιορθωμένα. Αυτό επιτυγχάνεται με την πρόσδεση ενός παράγοντα διμερισμού και συγκεκριμένα του AP20187 ή αλλιώς CAD. Μετά την πρόσδεση του παράγοντα, η χημερική αυτή πρωτεΐνη διμερίζεται και σηματοδοτεί το κυταρικό πολλαπλασιασμό. Και ενώ αυτή η μέθοδος έχει χρησιμοποιηθεί σε ζωικά μοντέλα για την έκπτυξη των γενετικά τροποποιημένων κυτάρων, δεν συνδυάστηκε ποτέ με ένα θεραπευτικό διαγωνίδιο για τη γονιδιακή θεραπεία των β' αιμοσφαιρινοπαθειών, κυρίως λόγω του μεγάλου μεγέθους των διαγωνιδίων που υπήρχαν τη δεδομένη στιγμή. Στο διάγραμμα βλέπουμε την έκπτυξη των γενετικά τροποποιημένων κυτάρων, ενώ κάθε κορυφή αντιστοιχεί στα time points στα οποία χορηγούταν ο παράγοντας διμερισμού. Και βλέπουμε έκπτυξη των διαμολυσμένων κυτάρων. Σκοπός λοιπόν της παρούσας μελέτης είναι να δημιουργήσουμε και να ελέγξουμε ένα νέο θεραπευτικό φορέα με ικανότητα θετικής επιλογής των γενετικά τροποποιημένων κυτάρων μέσω της κασέτας F36VMPL και του παράγοντα διμερισμού, που θα περιλαμβάνει παράλληλα και ένα θεραπευτικό διαγωνίδιο για τις β' αιμοσφαιρινοπάθειες. Ο νέος αυτός θεραπευτικός φορέας θα φέρει κάποια σημαντικά χαρακτηριστικά. Θα φέρει έναν μικρό αλλά εξίσου ισχυρό ερυθροειδικό ενισχυτή, όπως ανέφερε και η κυρία Ψαθά στην παρουσίασή της προηγουμένως, τον PVT1, έναν μονοτή χρωματίνης, το shRNA του καταστολέα της γάμμα σφαιρίνης BCL11A, που είναι μια πολύ μικρή αλληλουχία, και έτσι η μείωση στο μέγεθος του οικού φορέα μας επιτρέπει να προσθέσουμε και την κασέτα επιλογής του F36VMPL. Με αυτόν τον τρόπο πιστεύουμε ότι μπορούμε να πετύχουμε έναν οικό φορέα, με τον οποίο μπορούμε να πετύχουμε μεγάλους τίτλους, μπορεί να εισέλθει στα κύτταρα και με την κασέτα του F36VMPL μπορούμε να ενισχύσουμε τον αριθμό των κυτάρων που έχουν πάρει τον αριθμό των διαμολυσμένων κυτάρων. Επιπλέον προσθέσαμε και το γονίδιο GFP για την κανονικοποίηση των αποτελεσμάτων. Αρχικά ελέγξαμε το συγκεκριμένο οικό φορέα σε μια κυταρική σειρά ερυθροειδικών κυτάρων. Έγινε η διαμόλυνση, τα κύτταρα καλλιεργήθηκαν παρουσία και απουσία του παράγοντα διμερισμού και ελέγχθηκε ο πολλαπλασιασμός των γενετικά τροποποιημένων κυτάρων με κυταρομετρία ροής για το GFP και έκφραση της γάμμα σφαιρίνης. Είδαμε ότι η κασέτα επιλογής δεν επιδρά αρνητικά στη λειτουργία του ιού και καταφέραμε να πάρουμε μια καλή επαγωγή της γάμμα σφαιρίνης. Και εδώ παρατηρούμε στα αριστερά ότι το ποσοστό των GFP θετικών κυτάρων που διαμολύθηκαν με φορέα ελέγχου που δεν είχε την κασέτα επιλογής, ακόμα και με την προσθήκη του παράγοντα διμερισμού, έμεινε σχεδόν σταθερό σε όλη την πορεία της καλλιέργειας. Στα δεξιά, σε αντίθεση με τη συνθήκη απουσίας του, όταν προσθέταμε τον παράγοντα διμερισμού, στα κύτταρα που είχαν διαμολυθεί με τον ιικό μας φορέα, παρατηρήσαμε σημαντική αύξηση στο ποσοστό των GFP θετικών κυτάρων. Στη συνέχεια, έλεγξαμε τον ίδιο θεραπευτικό φορέα σε αιμοποιητικά στελεχεία κύτταρα από υγιείς δότες με παρόμοιο υπεραματικό σχεδιασμό. Διαμολύθηκαν τα κύτταρα, καλλιεργήθηκαν σε μέσο ερυθροδικής διαφοροποίησης, παρουσία και απουσία του παράγοντα διμερισμού και ελέγξαμε την έκρηξη των γενετικά τροποποιημένων ή GFP θετικών κυτάρων και την έκφραση της γάμμα σφαιρίνης. Παρατηρούμε λοιπόν ότι και στην περίπτωση των ανθρώπινων στελεχαίων αιμοποιητικών κυτάρων υπάρχει μια σημαντική διαφορά στην έκρηξη των γενετικά τροποποιημένων κυτάρων με την παρουσία του παράγοντα διμερισμού. Αυτό το βλέπουμε και δεξιά, όπου ακόμα και με ένα εξαιρετικά μικρό MOI και πολύ μικρό transduction, με την προσθήκη του παράγοντα διμερισμού, ο πληθυσμός αυτός γίνεται διακριτός. Εξετάζοντας την έκφραση επιφανειακών δικτών, είδαμε ότι δεν επηρεάστηκε σημαντικά η διαφοροποίηση των CD34 θετικών κυτάρων από την προσθήκη του παράγοντα διμερισμού, αλλά ούτε και κλωνογενής τους ικανότητα προς ερυθρές και μυελικές αποικίες. Και επίσης παρατηρούμε και μια πολύ σημαντική αύξηση στο ποσοστό των HbF θετικών κυτάρων, με παρουσία του παράγοντα διμερισμού, τόσο συγκριτικά με τη συνθήκη απουσίας του, όσο και με τα αρνητικά control. Τα αποτελέσματα αυτά επιβεβαιώθηκαν και με HPLC, στα οποία ελέγξαμε την παραγωγή των γάμμα σφαιρινών και το λόγο τους προς την ενδογενή α και β σφαιρίνη. Τέλος, πραγματοποιήθηκαν παρόμοια πειράματα και σε αιμοποιητικά στελεχεία κύτταρα από δότη με τρεπανοκυταρική ανεμία. Στα οποία και πάλι, όπως βλέπετε, έχουμε σταδιακά αύξηση του λόγου των GFP θετικών κυτάρων στη συνθήκη παρουσίας του παράγοντα προς τη συνθήκη απουσίας του. Η διαμόλυνση με τον ιικό φορέα και η προσθήκη του παράγοντα διμερισμού δεν επηρέασε τη διαφοροποίηση των κυτάρων ούτε σε αυτή την περίπτωση. Και παράλληλα αυξήθηκε το ποσοστό των HbF θετικών κυτάρων παρουσία του παράγοντα, το οποίο και πάλι επιβεβαιώθηκε με HPLC. Συμπερασματικά, λοιπόν, καταφέραμε επιτυχώς να εκπτύξουμε τα διαμολυσμένα με τον ιικό μας φορέα κύτταρα παρουσία του παράγοντα διμερισμού και δείξαμε ότι η μέθοδος είναι αποτελεσματική τόσο σε κυταρικές σειρές, όσο και σε ανθρώπινα υγιή και ενδρεπανοκυταρικά CD34 θετικά κύτταρα. Επιπλέον, δείξαμε ότι ο ιωκός αυτός φορέας αυξάνει το ποσοστό της έκφρασης της HbF στα διαμολυσμένα κύτταρα, το οποίο σε συνδυασμό με την αύξηση του ποσοστού των διαμολυσμένων κυτάρων στο συνολικό πληθυσμό οδηγεί στην ενίσχυση του θεραπευτικού αποτελέσματος. Σας ευχαριστώ πολύ.
Ευχαριστώ πολύ, κύριε Κέζου, για τα πολύ ωραία πειράματα αυτά. Ο τίτλος, είπατε, είναι γύρω στα 1/10 στην έκτη.
Ο τίτλος ήταν 36/10 στην έκτη, tighter units an ml.
Έχει περιθώρια βελτίωσης αυτό, γιατί με την κασέτα αυτή που έχετε θα πρέπει να ανέβει λίγο ο τίτλος.
Υπάρχουν ακόμα και με ένα πολύ χαμηλό transduction, βάζοντας την κασέτα αυτή, μπορούμε να πετύχουμε ακόμα περισσότερο την έκπτυξη των γενετικά τροποποιημένων κυτάρων. Αυτός είναι ο στόχος.
Ναι.
Γιατί αν ξεκινήσουμε με ένα υψηλό transduction από την αρχή, δεν χρειαζόμαστε τη βελτίωση.
Το MOI σας ήταν...
Το MOI ήταν πάρα πολύ μικρό, ήταν περίπου 8.
Αυτό έγινε ενσυνείδητα, όχι, δεν υπαγορεύτηκε από τον τίτλο. Κατά κάποιο. Λέω, το χαμηλό MOI δεν υπαγορεύτηκε από το σχετικά χαμηλό.
Όχι. Έγινε γι' αυτό το λόγο.
Επί τούτου. Μάλιστα. Και ο φάκελος του ιού?
Ήταν ο VSVG.
VSVG, okay. Οπότε, ναι. Ευχαριστώ.
Ευχαριστώ πολύ.
Προχωρούμε στην επόμενη ομιλία. Απολογούμαστε για τα μεικτά ελληνικά-αγγλικά, αλλά είναι το είδος του ΣΕΣ, το manual. Όχι, παρουσιάζουν είτε στα ελληνικά είτε στα αγγλικά, οπότε και εμείς ακολουθούμε ανάλογα. Λοιπόν, η επόμενη ομιλία είναι από την ομάδα του κυρίου Ανάγνου, Effective Targeting and Killing of Myeloma Cells by the IFM Vita HF Lentiviral Vector. Παρουσιάζει η κυρία Καλαφάτη.
Καλησπέρα. Η εργασία που θα σας παρουσιάσω αφορά την αξιολόγηση ενός λεντικού φορέα της Ιντερφερόνης Β ως εναλλακτική θεραπευτική προσέγγιση στο πολλαπλό μυέλωμα. Το πολλαπλό μυέλωμα είναι ένα κακόηθες νεόπλασμα των πλασματοκυτάρων. Χαρακτηρίζεται από υψηλή παραγωγή και έκρηση μιας μονοκλονικής ανοσφαιρίνης που ονομάζεται παραπρωτεΐνη και αποτελεί περίπου το 15% των αιματολογικών νεοπλασιών. Παρά το γεγονός ότι οι τρέχοντες θεραπευτικές προσεγγίσεις έχουν βελτιώσει τόσο την ποιότητα όσο και το προσδόκιμο ζωής των ασθενών, ένα σημαντικό ποσοστό από αυτούς υποτροπιάζει, εμφανίζοντας ανθεκτικότητα στη θεραπεία, ενώ ο μέσος όρος επιβίωσης παραμένει χαμηλός. Το 2017 παρουσιάστηκαν τα αποτελέσματα μιας κλινικής μελέτης, στην οποία χρησιμοποιήθηκε σε ασθενείς με πολλαπλό μυέλωμα το στέλεχος Έντμονστον του ιού της Ιλαράς και είχε ενθαρρυντικά αποτελέσματα. Το στέλεχος αυτό φέρει τις γλυκοπρωτεΐνες HF στην επιφάνειά του, οι οποίες στοχεύουν στους υποδοχείς CD46 και SLAM. Ο SLAM εκφράζεται σε όλα τα εμπύρινα κύτταρα, αλλά υπερεκφράζεται στα καρκινικά. Ο CD46, εκφράζεται σε όλα τα εμπύρινα κύτταρα και υπερεκφράζεται στα καρκινικά, ενώ ο SLAM εκφράζεται στα λεμφοκύτταρα, τα μακροφάγα και τα δεντριτικά. Η Ιντερφερόνη Β είναι μια πλειοτροπική κυτοκίνη με αντικαρκινική δράση. Δρά μέσω του μονοπατιού JAK-STAT και πάγει την απόπτωση και ενεργοποιεί το ανοσοποιητικό σύστημα έτσι ώστε να δράσει έναντι του όγκου. Στο πλαίσιο εξέβρεσης μιας νέας θεραπευτικής προσέγγισης για το πολλαπλό μυέλωμα, κατασκευάστηκε ένας λεντικός φορέας της Ιντερφερόνης Β, ψευδοτυπωμένος με τις γλυκοπρωτεΐνες HF του στελέχους Έντμονστον, οι οποίες ήταν γενετικά τροποποιημένες έτσι ώστε να αυξηθεί ο τροπισμός και αξιολογήθηκε θεραπευτική δράση τόσο σε μυελωματικές κυτταρικές σειρές, όσο και σε πρωτογενή κύτταρα ασθενών. Εδώ βλέπετε το συνολικό πειραματικό σχεδιασμό. Στην παρουσίαση όμως θα αναφερθώ μόνο στα δύο πρώτα μέρη. Αρχικά, σε πολλαπλότητα μόλυνσης ίση με 1, επιτεύχθηκε σημαντική ικανότητα μεταγωγής, ενώ ο αριθμός αντιγράφων του φορέα ανακύτταρο ήταν σχεδόν ιδανικός. Όσον αφορά το θεραπευτικό αποτέλεσμα, δοκιμάστηκαν διαφορετικές πολλαπλότητες μόλυνσης και ακόμα και στη χαμηλότερη πολλαπλότητα μόλυνσης, ίση με 0,01, παρατηρήθηκε σημαντική μείωση της βιωσιμότητας των μυελωματικών κυττάρων, ενώ στο υπερκείμενο της καλλιέργειας παρατηρήθηκε δοσοξεξαρτώμενη αύξηση της Ιντερφερόνης Β. Παράλληλα, αυξήθηκε και το ποσοστό των αποπτωτικών κυττάρων, προκειμένου να διερευνήσουμε το μηχανισμό που επάγει τον κυτταρικό θάνατο. Επιλέξαμε γονίδια σημαντικά για το μονοπάτι της απόπτωσης και όπως βλέπετε στα διαγράμματα σε επίπεδο RNA αυξάνεται η κασπάση 3, η κασπάση 9, η οποία υπογραμμίζει και το ρόλο του ενδογενούς μονοπατιού της απόπτωσης και μειώνονται τα BCL2 και BCLXL, τα οποία είναι εντοπιστικά. Αξίζει να αναφερθεί επίσης ότι παρατηρήθηκε αύξηση στα γονίδια στην έκραση των γονιδίων ENDO-G και HTRA, τα οποία εμπλέκονται στο ανεξάρτητο από τις κασπάσεις αποπτωτικό μονοπάτι. Τα παραπάνω αποτελέσματα επιβεβαιώθηκαν και σε επίπεδο πρωτεΐνης. Στη συνέχεια μελετήσαμε την έκφραση γονιδίων τα οποία εμπλέκονται στο μονοπάτι της αυτοφαγίας, της BECLIN1, του ATG4β και του ATG5, που εμπλέκονται στην έναρξη, την επιμήκυνση και την ορίμανση του αυτοφαγοσώματος. Είδαμε μείωση της έκφρασης αυτών των γονιδίων στην πλειοψηφία των μυελωματικών κυτταρικών σειρών, ωστόσο δεν μπορούμε να μιλήσουμε για μείωση της αυτοφαγίας και η επιβεβαίωση θα έρθει από πειράματα που βρίσκονται σε εξέλιξη με συναισθιακή μικροσκοπία. Στη συνέχεια προχωρήσαμε σε συγκαλλιέργεια μεταγμένων και μη κυττάρων μέσω Transwell και παρατηρήσαμε ότι μετά από 24 ώρες μειώθηκε σημαντικά η βιωσιμότητα των μη μεταγμένων κυττάρων, ενώ στο υπερκείμενο της καλλιέργειας παρατηρήθηκε αύξηση των Ιντερφερονών. Έπειτα, μελετήσαμε τη συνεργιστική δράση του φορέα με τρέχοντες θεραπευτικούς παράγοντες και όπως φαίνεται στο σχήμα, το θεραπευτικό αποτέλεσμα ενισχύεται στην περίπτωση που συγχορηγείται το πορτέζομι, το καρφίλζομι, δηλαδή οι δύο αναστολές του πρωτεασώματος και η λεναλιδομίδη. Το παραπάνω αποτέλεσμα επιβεβαιώθηκε και σε μια δεύτερη κυταρική σειρά. Όσον αφορά τη μελέτη της θεραπευτικής δράσης σε πρωτογενή κύτταρα ασθενών, προχωρήσαμε σε μεταγωγή με το φορέα σε υψηλότερη πολλαπλότητα μόλυνσης και παρατηρήσαμε αύξηση τόσο της ιντερφερόνης Β όσο και της ιντερφερόνης Γ. Μειώθηκε η βιωσιμότητα των κυττάρων και παράλληλα αυξήθηκε η απόπτωση, ενώ αξίζει να σημειωθεί ότι αυξήθηκε και το ποσοστό των κυττάρων που βρίσκονται στη φάση G1-G0 του κυτταρικού κύκλου, αναδεικνύοντας τον κυτταροστατικό ρόλο της ιντερφερόνης Β. Τέλος, υπολογίσαμε τον τίτλο των αντισωμάτων 1-DHF ψευδοτυπωμένων λεντικό φορέων και είδαμε ότι είναι χαμηλός και χρησιμοποιώντας αυξανόμενο MOI δείξαμε ότι ο ανοσολογικός φραγμός μπορεί να ξεπεραστεί. Συμπερασματικά, ο φορέας της ιντερφερόνης μπορεί να μετάγει αποτελεσματικά τα μυελωματικά κύτταρα, μειώντας τη βιωσιμότητά τους και πάγοντας την απόπτωση. Η απόπτωση επάγεται κυρίως μέσα από το ενδογενές μονοπάτι της απόπτωσης. Η ιντερφερόνη Β που παράγεται από τα μεταγμένα κύτταρα είναι ικανή να μειώσει τη βιωσιμότητα των μη μεταγμένων κυττάρων. Ο φορέας ιντερφερόνης Β μπορεί να δρά συνεργιστικά με τρέχοντες θεραπευτικούς παράγοντες, ενισχύοντας το θεραπευτικό αποτέλεσμα. Και τέλος, ο τίτλος των αντισωμάτων έναντι αυτών των φορέων είναι χαμηλός, ένα ενθαρρυντικό αποτέλεσμα για τη μελλοντική χρήση του σε κλινικό επίπεδο. Ευχαριστώ.
Ευχαριστούμε πολύ, κυρία Καλαφάτη, για τα ενδιαφέροντα αποτελέσματα. Κάποια ερώτηση από το ακροατήριο. Κυρία Γιαννάκη.
Ευχαριστώ πάρα πολύ. Ήθελα να ρωτήσω πώς φαντάζεστε την κλινική μετάφραση αυτής της προσέγγισης, δηλαδή πώς θα χορηγείται αυτός ο οδικός φορέας και ποια κύτταρα στοιχεία.
Αρχικά αυτή τη στιγμή δουλεύουμε πάνω σε πειράματα τα οποία θα αναπτύξουμε ένα in vivo μοντέλο συστημικής νόσου του πολλαπλού μυελώματος, όπου έχουμε επιλέξει να χορηγήσουμε ενδοφλέβεια τον φορέα προκειμένου να δούμε τον τροπισμό του. Επειδή η υποδοχέα CD46 εκφράζεται σε όλα τα εμπειρικά κύτταρα, αλλά υπερεκφράζεται στα καρκινικά, και σε πειράματα που έχουμε κάνει και δεν τα έχω συμπεριλάβει, έχουμε ως 100.000 φορές αύξηση του υποδοχέα CD46 στα καρκινικά σε σχέση με τα φυσιολογικά. Οπότε ενδοφλεβίως, εφόσον αποδεικτεί ο τροπισμός από τα in vivo πειράματα που είναι σε εξέλιξη. Σίγουρα και από τον SLAM στοχεύονται και τα δενδριτικά και τα μακροφάγα, πέρα από τα λεμφοκύτταρα, αλλά είναι ένας πιο επιλεκτικός φάκελος σε σχέση με άλλους. Ναι, θα το δούμε στο in vivo.
Πάνω σε αυτό θα ήθελα να ρωτήσω, αν μου επιτρέπετε, σε συνέχεια της ερώτησης κυρίας Γιαννάκη, πώς μπορείτε να αποτρέψετε ο ιός αυτός να σκοτώσει άλλα κύτταρα, μη καρκινικά. Γιατί η ιντερφερόνη Β μπορεί να οδηγήσει σε απόπτωση και γειτονικά κύτταρα ή να έχει άλλα κυτταροτοξικά φαινόμενα.
Αρχικά, ο υποδοχέας IFN-R1 δεν εκφράζεται στα κύτταρα τα οποία έχουν υψηλή πλαστικότητα. Οπότε, π.χ. στα CD34 που είχα ψάξει, η έκφραση του είναι μειωμένη. Όσον αφορά τα υπόλοιπα κύτταρα, σίγουρα ο τροπισμός του φακέλου, που είναι και γενετικά τροποποιημένος, αλλά και ένας ιστοειδικός υποδοχέας, ίσως ένας υποκινητής των β-σφαιρινών, θα μπορούσε να οδηγήσει σε ένα πιο στοχευμένο αποτέλεσμα.
Ωραία, ευχαριστώ πολύ.
Προχωρούμε στην τελευταία παρουσίαση για τη συνεδρία αυτή, πριν από την απονομή των βραβείων. Preclinical validation of hemoglobin beta, IVS 110 G2A mutation, specific line editing as advanced therapy for beta thalassemia από τον κύριο Πατσάλη, την ομάδα του Carsten Lederer.
Okay, yeah, hello everyone. So my research focused on the gene therapy for beta thalassemia, targeting the globally common, in particular in Cyprus, the IBS 110 missplicing mutation. This mutation leads to the incorporation of 19 nucleotides from intron 1 into the mature mRNA, leading to the production of a non-functional mRNA. However, even though some functional beta-globin mRNA is produced, the vast majority of the IBS 110 homozygotes suffer from severe transfusion-dependent thalassemia major. Our gene therapy approach aims to correct missplicing through genome editing by disrupting aberrant regulatory elements using designer nucleases such as the TALENs and CRISPR-Cas9, and the targeted double-strand break at the site of the mutation then triggers the non-homologous enjoining repair pathway to fix the problem in a non-precise way, creating insertion and deletions. And these genome modifications, we believe that they have the capacity to disrupt the abnormal regulatory elements and prompting the spliceosome to use the normal splice acceptor size and therefore have restoration of splice. We have already demonstrated the proof of principle of this approach using CRISPR-Cas9 and TALENs in vitro on patient-derived CD34 positive cells, showing correction at the RNA, protein, and late stage of erythroid differentiation level. We proceed to preclinical validation, comparing our mutation-specific approach with a universal HbF-induced approach, targeting the erythroid BCL11 enhancer element using CRISPR-Cas9, which is currently in clinical trials, on the NBS-GW immunodeficient mouse model. In our study, patient-derived hematopoietic stem cells were nucleoaffected with CRISPR-Cas9 as ribonucleoprotein complexes, while TALEN and GFP were delivered as mRNAs. After 2 days, cells were divided to in vitro work, where we assess the functional correction and the erythroid and myeloid lineage potential of the edited cells, and to in vivo work for the assessment of long-term repopulating capacity of the cells in primary and secondary NBS-CW recipients 16 weeks post-transplantation. Starting with the in vitro experiments, this graph shows on and off targeting, on targeting levels on day 9 post-nucleoaffection, where both IBS 110 CRISPR-Cas9 and TALEN showed high targeted disruption efficiency, 89 and 60% respectively, and without any off-targeting on the top in silico predicted off-target sites, while the CRISPR-Cas9 that targets the BCL11A enhancer element showed low genome editing efficiencies. Late-stage erythroid differentiation was measured as percentage of CD235 positive 36 negative cells during erythroid differentiation, and both IBS 110 CRISPR-Cas9 and the TALEN showed exhibit improved late-stage erythroid differentiation, and especially the IBS 110 CRISPR-Cas9 showed correction starting from erythroid differentiation day 11, whereas the CRISPR-Cas9 targeting the BCL11A enhancer element showed modest improvement compared to the untreated control. Looking of late-stage erythroid differentiation level as enucleation, again, cells edited with the IBS 110 CRISPR-Cas9 and the TALEN showed increased levels of enucleation, and in the case of the IBS 110 of the BCL11A targeted cells showed slight increase in enucleation. Analysis of reactive oxygen species, mean fluorescent intensity in the enucleated cells, showed more than 35% reduction in the CRISPR-Cas9 targeting in the IBS 110, CRISPR-Cas9 in the tail ends compared to the untreated, whereas the BCL11A edited cells didn't show any reduction of the ROS levels. Looking at the beta globin-like globin chain proportions in these pie charts, you can see that again the IBS 110 CRISPR-Cas9 and the tail end showed increased the beta globin chain levels were increased by 50% and 34% in the case of IBS 110 CRISPR-Cas9 and tail ends, and there was a slight increase where the beta globin is in blue. And there was a slight increase of the gamma globin chains in the case of the edited cells with the BCL11A targeting the BCL11A enhancer element compared to the untreated. Similar findings are shown also in these bar charts, where we see the beta and gamma ratio to alpha globin chains. Analysis of erythroid and myeloid lineage potential of the cells by scoring DFUEs and CFUGM colonies in clonogenic assays, respectively, showed similar percentage across all samples, indicating that genome editing did not affect the erythroid and myeloid lineage potential on the edited hematopoietic stem cells. Moving to the in vivo experiment, 16 weeks post the first xenotransplantations, the animals were sacrificed for the assessment of the long-term repopulating capacity of the edited cells and proceeded to the second xenotransplantation. Analysis of the bone marrow and peripheral blood chimeras in the primary NBSGW recipients by flow cytometry showed high bone marrow engraftment, up to 80% for the tail ends, around 10% chimeras for the CD34 positives, and around 20% of CD44 positive in the peripheral blood. Statistical analysis of the bone marrow and peripheral blood chimeras didn't show any statistically significant difference between the genome-edited cells and the non-edited cells, indicating again that the genome editing didn't affect the long-term repopulating capacity. Genome editing on day 9 post-nucleofection and on primary bone marrow cells showed a drop of on-targeting down to 52% for the IBS 110 CRISPR-Cas9 and down to 14% for the tail and edited cells, while no editing was observed in the bone marrow cells that they were edited with the CRISPR-Cas9 targeting the BCL11A enhancer element. Overall, the IBS 110 CRISPR-Cas9 demonstrated significantly higher genome editing in the bone marrow cells compared to the tail ends, indicating its superiority despite that tail end edited cells had slightly higher bone marrow chimerism. In summary, all of our in vitro and in vivo data up to now indicate IBS 110 CRISPR-Cas9 as the best approach and tool for the treatment of IBS 110 beta thalassemia in terms of genome editing, functional correction, and maintenance of long-term repopulating capacity of the genome-edited hematopoietic stem cells. However, in order to have conclusive results, we still have to evaluate the long-term populating capacity of the edited cells in the secondary NBSDW recipients, and importantly, we need to assess the biosafety of genome editing, of the genome editing tools by on and off targeting analysis using targeted deep sequencing and chromosomal rearrangements via Cas. Thank you. I would like to thank the department, of course, the George Papanikolaou Hospital, our collaborators, for their hospitality, the University of Freiburg, Tony Cadome and Claudio Musolino, and of course the patients for their support.
Thank you very much. Since we have to run for the next two presentations, we cannot accept any questions. Thank you very much anyway. So we move to the next stage of.
Τώρα προχωρούμε στις τρεις εργασίες που έχουν βραβευτεί, δύο ισοδύναμες προφορικές και μία αναρτημένη ανακοίνωση. Εκτός από το τιμητικό χαρτί, πώς λέγεται, το βραβείο, τέλος πάντων το δίπλωμα, οι δύο προφορικές, δηλαδή ο πρώτος συγγραφέας, θα κερδίσει και μία συμμετοχή σε Ευρωπαϊκό Συνέδριο κατά προτίμηση της Ευρωπαϊκής Εταιρείας Γονιδιακής και Κυταρικής Θεραπείας, και αυτός που κερδίζει το πόστερ σε αντίστοιχο συνέδριο της ελληνικό, δηλαδή αυτής της εταιρείας ή της αιματολογικής εταιρείας ή της ελληνικής εταιρείας βιοχημίας και μοριακής βιολογίας. Το ένα από τα δύο ισότιμα προφορικά βραβεία αφορούν την εργασία Development of a tailored cell-free therapeutic approach based on MSC exosomes for acute hepatic failure, και αποδίδεται στην ομάδα της κυρίας Ρουμπελάκη. Ο ομιλητής η κυρία Ψαράκη.
Καλησπέρα σας. Η συγκεκριμένη ομιλία είναι για την ανάπτυξη μιας προσωπημένης μη κυταρικής θεραπευτικής προσέγγισης βασισμένης στα εξωμάτια προερχόμενα από μεσημεματικά στρωματικά κύτταρα MSC στην οξεία ηπατική ανεπάρκεια. Ως οξεία ηπατική ανεπάρκεια αρχικά χαρακτηρίζεται η εμφάνιση ηπατικής βλάβης χωρίς προηγουμένως κάποια γνωστή ένδειξη και χαρακτηρίζεται κυρίως από υψηλό ποσοστό θνητότητας, εκτεταμένη δυσλειτουργία υπατοκυττάρων, ενώ κύριες αιτιολογίες είναι οι υπατοτοξικά φάρμακα, η αυτοάνωση, η επίθεση ή η κοιμόλυνση. Μέχρι σήμερα η κύρια γραμμή αντιμετώπισης οξείας ηπατικής ανεπάρκειας παραμένει η μεταμόσχευση του ύπατος. Όμως, οι περιορισμοί που σχετίζονται κυρίως με τον περιορισμένο αριθμό των δοτών, τις μεγάλες λύσεις αναμονής και τις επιπλοκές ανοσοαπόρριψης, απαιτούν την έβρεση νέων εναλλακτικών θεραπείων για την οξεία ηπατική ανεπάρκεια. Σε πρόσφατο άρθρο, ανασκόπησεις έχουν αναλυθεί τα μειονεκτήματα της χρήσης της μεταμόσχευσης υπατοκυττάρων σαν θεραπεία για την οξεία ηπατική ανεπάρκεια, καθιστώντας αναγκαία την έβρεση νέων εναλλακτικών κυταρικών πηγών για τη θεραπεία της οξείας ηπατικής ανεπάρκειας, όπως είναι τα μεσεγχηματικά στρωματικά κύτταρα MSC, τα οποία μπορούν να προέλθουν είτε από ενήλικες είτε από εμβρυϊκές πηγές. Σε πρόσφατο άρθρο έχουν αναλυθεί τα πλεονεκτήματα της χρήσης MSC ως εναλλακτική θεραπευτική στρατηγική για ορισμένες ασθένειες του ύπατος σε κλινικές μελέτες. Όπως επίσης σε προκλινικό επίπεδο έχουν αναφερθεί σε προηγούμενες μελέτες του εργαστηρίου τα θεραπευτικά αποτελέσματα της χρήσης του κρυτώματος προερχόμενου από MSC και υπατικά προγονικά κύτταρα HPA σε ποντίκια με υπαγόμενη με τετραχλωράνθρακα οξεία ηπατική ανεπάρκεια, τα οποία, όπου παρατηρήθηκε συνολικά η βελτίωση του υπατικού φαινοτύπου, η μείωση των επέδρων της φλεγμονής, όπως επίσης και η αύξηση του ρυθμού μεταβολισμού των ονοειδών κυττάρων. Επιπλέον, σε πρόσφατο άρθρο έχουν αναλυθεί και τα πλεονεκτήματα της χρήσης εξοκυττάρων και στιδίων προερχόμενων από MSCs, τα οποία περιέχονται στο κρύτωμα, τα οποία δρώντας μέσω διαφορετικών σηματοδοτικών μηχανισμών έχουν ως αποτέλεσμα προσδίδουν θεραπευτικά αποτελέσματα σε διάφορες ασθένειες του ύπατος, καθιστώντας τα μια εναλλακτική λύση για την οξεία ηπατική ανεπάρκεια. Σύμφωνα με τα παραπάνω, οι στόχοι της συγκεκριμένης εργασίας ήταν πρώτον η διερεύνηση του θεραπευτικού ρόλου των εξωσωματείων προερχόμενων από MCs, υπατικά προγονικά κύτταρα HPL και υπατοκύτταρα HL σε προκλινικά μοντέλα οξύς υπατικής ανεπάρκειας, η ταυτοποίηση του πρωτοαιμικού προφίλ των εξωσωματείων προερχόμενων από τους 3 κυταρικούς τύπους, η διερεύνηση του βιολογικού ρόλου των επιλεγμένων μορίων που απαντώνται στο φορτίο των εξωσωματείων στην εξέλιξη της οξύς υπατικής ανεπάρκειας, όπως επίσης και η διερεύνηση των σηματοδοτικών μηχανισμών στα οποία εμπλέκονται τα εξωσωμάτια στην εξέλιξη της οξύς υπατικής ανεπάρκειας. Για τον σκοπό αυτό, αρχικά MSCs κύτταρα διαφοροποιήθηκαν σε υπατικά προγονικά κύτταρα και στη συνέχεια σε υπατοκύτταρα, και από κάθε τύπο κυττάρων απομονώθηκαν εξωσωμάτια τα οποία χαρακτηρίστηκαν στη συνέχεια μέσω nanosite και ηλεκτρονικής μικροσκοπίας, με το μέγεθός να κυμαίνεται μεταξύ 50 και 200 νανομέτρων. Στη συνέχεια χαρακτηρίστηκαν μέσω Western blot και περατομικής ανάλυσης για χαρακτηριστικούς δείκτες όπως είναι η Floatlin 1, η CD63, η CD9, η CD81 και η HSP90α1. Στη συνέχεια, εξωσωμάτια σημάνθηκαν και χορηγήθηκαν ενδοϊπατικός και ενδοβλεβίος σε ποντίκια με παγόμενη με τεραχλωράνθρακα οξύ υπατική ανεπάρκεια, όπου η ενδοϊπατική οδός χορήγησης αναδείχθηκε ως η καταλληλότερη οδός χορήγησης των εξωσωματείων, ενώ η χορήγηση εξωσωματείων προερχόμενη από τους 3 κυταρικούς τύπους είχε ως αποτέλεσμα τη βελτίωση του φαινοτύπου, τη μείωση των περιοχών νέκρος, όπως επίσης και τη μείωση των περιοχών σώρευσης λιπιδίων, γεγονός που επιβεβαιώθηκε μέσω χρεώσεων με τάνελ και oil redot, όπως και μέσω μοριακών μεθόδων για συγκεκριμένα γονίδια, όπως είναι το BCL2 και το PPARG. Παράλληλα, ex vivo παρατηρήθηκε η αύξηση του ρυθμού πολλαπλασιασμού των νοηδών κυττάρων, παρουσία εξωσωματείων προερχόμενων από τους 3 κυταρικούς τύπους, κατά την 3η μέρα καλλιέργειας. Στη συνέχεια, πραγματοποιήθηκε και η πρωτοαιμική ανάλυση των εξωσωματείων προερχόμενων από τους 3 κυταρικούς τύπους, τα οποία αναλύθηκαν με βάση δεδομένων, όπως επίσης και μεταξύ τους, και αναδείχθηκαν 6 κοινές και στατιστικά σημαντικές πρωτεΐνες, μεταξύ των οποίων και εκείνη της MFG8. Τα επίπεδα της MFG8 αναλύθηκαν στο εκρήτωμα και στα εξωσωμάτια των MSCs και των HPL κυττάρων, όπου παρατηρήθηκε στατιστικά σημαντική αύξηση των επίπεδων της MFG8 στα δείγματα των HPL εξωσωματείων. Στη συνέχεια, θέλοντας να διερευνήσουμε τη δράση της, ανασυνδυασμένη πρωτεΐνη recombinant MFG8 χορηγήθηκε σε ποντίκια με παγόμενη με τεραχλωράνθρακα οξύ υπατική ανεπάρκεια, όπου και διαπιστώθηκε η πτώση των επένδρων αμινασών στο ρο του αίματος των ποντικών, όπως επίσης και η βελτίωση του φαινοτύπου ιστολογικά, με μείωση των περιοχών νέκρωσης και της διεύθυνσης φλεγμονωδών κυττάρων. Παράλληλα, η χορήγηση εξωσωματείων προερχόμενη και από τους 3 κυταρικούς τύπους είχε ως αποτέλεσμα την αύξηση του ρυθμού, την αύξηση στην έκφραση ενδογενούς MFG8 στο υπερποντικών με οξύ υπατική ανεπάρκεια, το οποίο βεβαιώθηκε μέσω ανοσοθορισμού και μέσω μοριακών μεθόδων. Τα χαμηλά επίπεδα έκφρασης MFG8 συσχετίστηκαν με βιοψίες ασθενών οξύς υπατικής ανεπάρκειας σε σχέση με τους αρνητικούς μάρτυρες, ενώ επίσης η χορήγηση της ανασυνδυασμένης πρωτεΐνης MFG8 σε ποντίκια με οξύ υπατική ανεπάρκεια είχε ως αποτέλεσμα επίσης την αύξηση της ενδογενούς MFG8. Ακολούθησε πληροφορική ανάλυση των κοινών πρωτεϊνών του εκρητώματος και των εξωσωματείων προερχόμενων από MSCs και HPL κύτταρα, όπου αναδείχθηκε το PI3K ACT σηματοδοτικό μονοπάτι ως σημαντικό για την εξέλιξη οξύς υπατικής ανεπάρκειας. Και στη συνέχεια, μέσω μοριακών μεθόδων για συγκεκριμένα γονίδια που εμπλέκονται στο σηματοδοτικό μονοπάτι, παρατηρήθηκε η μείωση της ενεργότητας του σηματοδοτικού μονοπατιού PI3K ACT, έπειτα από τη χορήγηση εξωσωματείων προερχόμενων και από τους 3 κυταρικούς τύπους, όπως επίσης και έπειτα από τη χορήγηση της recombinant MFG8 στο υπαρποντικών με οξύ υπατική ανεπάρκεια. Σε δεύτερο χρόνο, πραγματοποιήθηκε η πρωτοαιμική ανάλυση στο υπαρποντικών με οξύ υπατική ανεπάρκεια έπειτα από τη χορήγηση recombinant MFG8, όπου παρατηρήθηκε μέσω βιοπληροφορικής ανάλυσης η αύξηση της ενεργοποίησης των μονοπατιών που σχετίζονται με το μεταβολισμό, το σηματοδοτικό μονοπάτι της ινσουλίνης, όπως επίσης και της φερόπτωσης, και η μείωση των σηματοδοτικών μονοπατιών που σχετίζονται με την επεξεργασία του mRNA και την αναδιοργάνωση του πυρηνικού φακέλου. Παράλληλα, αναλύθηκαν για τα επίπεδα έκφραση σημαντικών δικτών υπατικής βλάβης, όπου παρατηρήθηκαν να ακολουθούν παρόμοιο πρότυπο με εκείνα των υγιών ποντικών έπειτα από τη χορήγηση recombinant MFG8, ενώ επίσης παρατηρήθηκε η μεταβολή της έκφρασης πρωτεϊνών που σχετίζονται με τη φλεγμονή, όπως είναι οι καθεψίνες και οι σαρπίνες. Στη συνέχεια, αναλύθηκε εκτενώς και το μονοπάτι της φερόπτωσης και συγκεκριμένα αναλύθηκαν οι πρωτεΐνες που απαντώνται στην πρωτοαιμική ανάλυση και εμπλέκονται στο μονοπάτι της φερόπτωσης, όπου παρατηρήθηκε η εμπλοκή της φερόπτωσης στη δράση της MFG8 στην οξύ υπατική ανεπάρκεια. Στη συνέχεια, διερευνήθηκε και η δράση της αυτοφαγίας έπειτα από τη χορήγηση recombinant MFG8, όπου μέσω πρωτομικής ανάλυσης και μεταγραφικής ανάλυσης για συγκεκριμένα γονίδια δείκτες, όπως είναι η ETG7, η ETG5, η FSGY, η Becklini 1 και η LC3β, παρατηρήθηκε αύξηση ενεργότητας στις αυτοφαγίες έπειτα από τη χορήγηση ρεκόμενων MFGH στο υπερποντικών με οξύ αιπατική ανεπάρκεια. Η αποσιώψη της MFGH στα MSS κύτταρα μέσω λιτικού φορέα και κατ' επέκταση στα εξωσωμάτια επιβεβαιώνει τη θεραπευτική δράση της MFGH στην οξύ αιπατική ανεπάρκεια, όπως επίσης και την δράση των εξωσωματίων μέσω της αυτοφαγίας και της φερόπτωσης. Συνολικά, τα εξωσωμάτια προερχόμενα και από τους τρεις τύπους κυττάρων είχαν ως αποτέλεσμα την βελτίωση του φαινοτύπου in vivo, όπως επίσης και την αύξηση του ρυθμού πολλαπλασιασμού των οειδών κυττάρων ex vivo. Τα εξωσωμάτια προερχόμενα και από τους τρεις κυτταρικούς τύπους κατάφεραν να αυξήσουν τα επίπεδα έκφρασης της ενδογενούς MFGH στο υπερποντικών με οξύ αιπατική ανεπάρκεια. Τα χαμηλά επίπεδα έκφρασης MFGH συσχετίστηκαν με βιοψίες ασθενών με οξύς αιπατικής ανεπάρκειας με τους αρνητικούς μάρτυρες. Η MFGH, η οποία περιέχεται στο φορτίο των εξωσωματίων προερχόμενων από MSCs, χαρακτηρίστηκε ως σημαντικό μόριο για την εξέλιξη της οξύς αιπατικής ανεπάρκειας μέσω της πτώσης του σηματοδοτικού μονοπατιού PI3K και αντιπροσωπεύει έναν μελλοντικό προγνωστικό βιοδείκτη της εξέλιξης οξύς αιπατικής ανεπάρκειας. Η φερόπτωση και η αυτοφαγία αναδείχθηκαν ως σημαντικά σηματοδοτικά μονοπάτια για την δράση της MFGH, όπως επίσης και για την εξέλιξη της οξύς αιπατικής ανεπάρκειας, ενώ συνολικά η συγκεκριμένη μελέτη αποτελεί μια προσαρμοσμένη με θεραπευτική προσέγγιση βασισμένη στα εξωσωμάτια, μη κυτταρική, μη τοξική και μη νοσογονική, η οποία προσφέρει νέες πληροφορίες για τις μηχανιστικές πληροφορίες, όπως και για τη δράση των εξωσωματίων. Σας ευχαριστώ για την προσοχή σας.
Μια ερώτηση. Ναι, κύριε Ανάγνου. Καλύτερα να σε ακούνε όλοι. Εγώ θα σε ακούω, αλλά δεν έχει σημασία αυτό.
Συγχαρητήρια για την πολύ καλή δουλειά. Με βάση και αυτά που είπε και η κυρία Ρουμπελάκη χθες όσον αφορά την ετερογένεια αυτών των μεσοχηματικών πληθυσμών και υποπληθυσμών, οι οποίοι χαρακτηρίζονται αφενός από την ικανότητα των πλαστικών ιδιωτήτων, από την άλλη και την παρακλινική τους δράση. Αυτή τη στιγμή δεν υπάρχει κάποια ασφαλής μέθοδος η οποία μπορεί να προβλέψει, επειδή στον τίτλο σας γράψατε tailoring, δηλαδή μπορεί να προσαρμοστεί ανάλογα με τις ενδείξεις κάθε παθήσεως. Θα μπορούσε κανείς, υπάρχουν ενδείξεις ότι κάποια μόρια όπως είναι το twist one νομίζω, το οποίο συνδέει αυτές τις δύο ιδιότητες τις βασικές, πλαστικής επιδιόρθωσης και παρακλινικής δράσης, ούτως ώστε να φτιαχτεί μια κλίμακα με βάση αυτό το twist και που συνδέει αυτές τις δύο ιδιότητες, ούτως ώστε αυτό να καθορίσει καλύτερα μια ποσοτικοποίηση των ενδείξεων σε σχέση με τις παθήσεις που ενδεχομένως να ωφελούνται δυνητικά από αυτή την προσέγγιση. Έχετε κάποιο σχόλιο?
Γενικά, τα εξωσωμάτια πια μπορούν να προσαρμοστούν για την κάθε πάθηση, μπορούν να γίνουν modified, είτε ως προς το φορτίο που περιέχουν, είτε ως προς τα κύτταρα που στοχεύουν. Οπότε, σίγουρα αυτό είναι ακόμα πιο... μπορεί να αποτελέσει ένα μεγαλύτερο πλεονέκτημα για τη θεραπεία συγκεκριμένων ασθενειών στο μέλλον, στοχεύοντας σε συγκεκριμένα κύτταρα και ενεργώντας σε συγκεκριμένα σηματοδοτικά μονοπάτια.
Τι θα ακολουθήσει μετά την έρευνά σας αυτή?
Ακόμα δεν το γνωρίζουμε. Τώρα, σε αυτή τη στιγμή προσπαθούμε να διερευνήσουμε ακόμα περισσότερο τη δράση της MFG8 ως μόριο στην εξέλιξη οξύς αιπατικής ανεπάρκειας, όπως επίσης και το ρόλο που έχει στην δράση των εξωσωματίων. Ίσως μια εναλλακτική λύση είναι και η τροποποίηση των εξωσωματίων να είναι enrich της MFG8 ως μια άλλη θεραπευτική προσέγγιση για τις ασθένειες του ύπατος.
Ωραία. Ευχαριστούμε πολύ. Ελάτε προς τα εδώ. Έλα και εσύ. Εδώ στη μέση, επειδή είναι μια πολύ ωραία εργασία, συγχαρητήρια.
Και τώρα θα περάσουμε στην δεύτερη ισοδύναμα βραβευμένη εργασία από την κυρία Πασχούδη, από το εργαστήριο της κυρίας Ψαθά. A synthetic erythroid enhancer successfully replaces micro-LCR in gene therapy vectors for beta hemoglobinopathies.
Hello again. I will talk about our work regarding the generation of erythroid enhancer, synthetic erythroid enhancer, that successfully replaced the micro-LCR. The gene therapy for beta hemoglobinopathies includes the collection of mobilized hematopoietic stem cells from patients' peripheral blood, the ex vivo transduction of these cells with lentiviral vectors carrying beta globin or gamma globin gene, and the transplantation of transduced cells back to the patient after myeloablation. Notwithstanding the high success rate that emerged from lentiviral-mediated gene therapy, the limitations of the currently available vectors minimize the applicability of the approach. The introduction of long beta-globin LCR, spanning almost 3 kb in size, reduces the viral titers and HCC's transducibility. Additionally, the activation of LCR elements in early HSPCs could potentially reduce the safety profile of these vectors, even though no genotoxicity events have been observed in the clinic. As Dr. Psatha mentioned before, in a previous study, we identified 2 potent compact erythroid-specific enhancers with differential enhancer activity. These erythroid enhancers displaying the strongest activity at the early and late stages of erythroid maturation and were identified as bona fide enhancers of PBT1 and PPAR-alpha gene respectively. In this study, we explored the possibility to combine these 2 potent compact erythroid-specific enhancers to generate a stronger synthetic super enhancer able to successfully replace the micro-LCR. To this aim, we generate a reported vector bearing both enhancer sequences. This vector was compared with respective vectors with single enhancer elements in ex vivo experiments, where normal CD34 positive cells were transduced at the same MOI with all 3 vectors, as well as a non-enhancer control vector. We assessed GFP expression intensity during ex vivo erythroid differentiation, and we observed that the synthetic enhancer achieved a higher and possible synergistic effect. Comparing the lentiviral titers of this vector with enhancer concatemer to relevant cassette incorporated the long micro-LCR, we observed a 5-fold increase in virus production. Next, we transduced CD34 positive cells from normal donor with micro-LCR and enhancer concatemer GFP vectors at the same MOI. With the use of synthetic enhancer, we were able to achieve a significantly higher percentage of transduced cells, and this increased in GFP expression remained stable during erythroid differentiation. The next step was to compare these 2 erythroid enhancers in therapeutic vectors. We therefore generated a shRNA BCL11A vector and an additional vector containing the gamma globin cDNA. As expected, the viral titers of BCL11A shRNA vector were significantly higher with the use of the synthetic enhancers. However, we did not observe a similar effect in the case of gamma globin vector. Transduction of HUDEP2 cells with BCL11A shRNA or gamma globin vector resulted in significantly increased and almost identical HbF reactivation in all conditions. Next, CD34 positive cells from 3 thalassemic patients were transduced with micro-LCR and enhancer concatamer vector, enhancer concatamer BCL11A or gamma globin vector at the same MOI, and the cells were seeded in erythroid differentiation medium and in methylcellulose culture. As you can see here, transduction with lenti vectors, both BCL11A shRNA vector and gamma globin vector, did not affect the clonogenic capacity of CD34 positive cells, as we don't observe differences between the total numbers of erythroid and myeloid colonies. Additionally, transduction of CD34 positive cells did not affect the immunophenotype characteristics during erythroid differentiation. Interestingly, during erythroid differentiation, we observed an evident, however not statistically increased, in the expansion rate of enhancer concatamer vector transduced cells, both in BCL11A shRNA group and in gamma globin group. Despite the fact that we didn't observe differences in vector copy numbers, at the end of erythroid differentiation, we observed a significantly increased percentage of HbF positive cells in all transduced conditions, with, in this case, enhancer concatamer is performing at least the same, if not even better, to the gold standard micro-LCR. And also we observed that with a BCL11A shRNA vector, both with micro-LCR and with enhancer concatamer, we achieved a higher percentage of HbF positive cells compared to gamma globin vectors. The increase in HbF levels was also translated in reduced levels of oxidative stress and improved cell morphology. Overall, for the first time, we generated a synthetic, potent, compact, erythroid-specific enhancer able to successfully replace the micro-LCR in gene therapy vectors for beta hemoglobinopathies. This enhancer was implemented in clinically relevant viral vectors, and we don't observe, and this induction did not negatively affect the viral titers. Also, in the case of enhancer concatamer transduced cells, we observed induction of HbF expression and improvement of disease phenotype. Overall, we were maybe at no. We were planning a new generation of lentiviral gene therapy vectors for beta hemoglobinopathies. And sorry, thank you so much.
Thank you very much for this very nice work. Are there any questions from the audience? If not, and because of saving some time, we will proceed.
Στη φωτογραφία. Και δυστυχώς φτάσαμε στην τελευταία.
Last but not least.
Είναι ωραίο να δίνεις βραβεία, το ξέρετε. Λοιπόν, είπαμε για την καλύτερη εναρτημένη εργασία, η οποία έχει τον τίτλο Control Human iPSC-Derived Astrocytes Rescue the Regenerative, δεν βλέπω καλά, συγγνώμη, Phenotype of A53T A-SYN iPSC-derived neurons generated from Parkinson's disease patients. Η ομιλήτρια είναι η κυρία Αποκότου από την ομάδα της κυρίας Παπαστεφανάκη στο Παστέρ.
Thank you very much for the introduction. I'm very glad to share with you my work on neuron-glia interactions in Parkinson's disease. First, some introduction. Parkinson's disease is primarily characterized by the loss of dopaminergic neurons, resulting to both motor and non-motor symptoms. The main histopathological feature of the disease is the presence of intraneuronal inclusions rich in alpha-synuclein, termed Lewy bodies and Lewy neurites. The PA53T mutation in the gene of alpha-synuclein causes a familial form of PD with early onset and a very severe phenotype. Here, for this study, we generated astrocytes and dopaminergic neurons from induced pluripotent stem cells derived from patients harboring the PA53T mutation and healthy donors, and combined them in a co-culture system at all possible combinations. We primarily observed compromised neuronal viability as well as impaired neuritic outgrowth of healthy neurons co-cultured with PD astrocytes. Interestingly enough, this phenotype was reversed in PD neurons in the presence of healthy astrocytes. Moreover, we managed to quantify PD-associated neuropathological features such as Lewy body-like formations, Lewy neurites, retraction bulbs, etc. And we noted that the neurodegenerative phenotype in healthy neurons was exacerbated in the presence of PD astrocytes. And moreover, this phenotype, this neurodegenerative phenotype, was significantly alleviated in PD neurons co-cultured with healthy astrocytes. I'm sorry, something came up here. Okay. Last, we used rabies virus-based retrograde monosynaptic tracing system in order to track the synaptic connectivity between neurons. What we observed is that PD neurons co-cultured with PD astrocytes displayed impaired synaptic connectivity that was restored by healthy astrocytes. And of course, accordingly, the synaptic connectivity of healthy neurons was significantly impaired by PD astrocytes. In conclusion, our data support a critical role of PD astrocytes, mutant astrocytes, in the neurodegeneration process of healthy neurons, and also a remarkable ability of healthy astrocytes to rescue the neurodegeneration of PD neurons. And with that, I thank you for your attention.
Υπάρχει μια ερώτηση? Είμαστε πολύ πίσω, ευχαριστούμε που δεν υπάρχει. Κυρία Παπαστεφανάκη, έρχεστε κι εσείς.
Ευχαριστούμε πολύ και συνεχίζουμε με τα δύο keynote lectures.
Guard. Okay. If you don't mind, I better start with the second speaker. So probably Stylianos will come and introduce Hildegard. So the session, this session is two special lectures. There are two distinguished speakers. I will start with Dr. André Lieber. He's a professor of medicine in the University of Washington and an elected member of the American Society for Clinical Investigation. So Dr. Lieber has spent more than 25 years in research on adenoviral vectors, both basic and also translational research. As regards his translational approaches, it has two main pillars. The first one is refers to cancer. So he has developed adenovirus recombinant proteins for treating cancer, and two of these products are currently under IND. And very recently he has also developed an in vivo approach for targeting hematopoietic stem cells with adenoviral vectors. And this expands not only to hemoglobinopathies but also to several other target diseases. So this approach involves the hematopoietic stem cell transplantation, the in vivo transduction of hematopoietic stem cells, which then migrate back to the bone marrow and produce normal cell lineage after gene correction. So as you realize, by targeting in vivo hematopoietic stem cells, and in particular thalassaemic stem cell, creates a pioneering approach for treating beta hemoglobinopathies by avoiding leukapheresis, conditioning, transplantation expertise, and also by significantly lowering the cost. And this is will be a great progress towards making the gene therapy for hemoglobinopathies accessible to all patients. So we are very honored to have Dr. Lieber with us. He will talk on in vivo gene therapy for hemoglobinopathies and beyond.
Thank you, Lila, for giving half of my talk already. I mean, at least conclusions. I'm not sure why I'm second last. I was preparing to be the last speaker. But anyway, I would like to thank the organizing committee and all of you for inviting me. And again, thank you for staying for so long. Okay, so these are my disclosures. Yeah, as Lila explained already, we have developed an in vivo HSC gene therapy approach that involves the mobilization of HSCs from the bone marrow into the periphery And while they are circulating in the periphery, we are intravenously injecting an HSC-tropic adenovirus vector, transduced cells or cells that are transduced in the periphery to a large degree home back to the bone marrow. And as Lila explained, this avoids a lot of issues that are associated with ex vivo HSC gene therapy. So I would like to walk you through some of the more important components of this approach and then give a couple of examples for using the approach for treatment of hemoglobinopathies. So to target HSCs, we are relying on the natural tropism of adenovirus vectors, specifically of serotype 35 and 3. As you might know, there are more than 100 different serotypes. S35 uses CD46 as a receptor, which is known as a complement inhibitor, while S3 uses desmoglein 2, which is widely known as an epithelial junction protein. So it was quite surprising that we found both of these receptors expressed on human CD34 plus cells and also on rhesus CD4 plus cells. So what is, what is important is, what is important is that, for example, for CD46, the density of this receptor is higher on more primitive cells, for example, on CD34 plus, CD90 plus cells. In the human system, we are also testing the approach in transgenic mice that express human CD46 in a pattern similar to humans and on an adequate model. And in this case, we see higher expression levels of CD46 on LSK cells in the bone marrow. Interestingly, both of these receptors are also upregulated on dedifferentiated cancer cells, which indicates that these proteins might have a function in maintaining stemness. So our vehicle is an adenovirus. Specifically helper-dependent adenovirus or gutless adenovirus. All viral genes are removed, so there's lots of space for inserting transgenes. The capsid has been heavily modified. It contains so-called fibers that target, that have enhanced affinity to either CD46 or DSG2. The capsid is derived from serotype 6, which largely circumvents pre-existing immunity in humans. It minimizes hepatocyte transduction, and the vector can be produced at very high yields. And this is an important aspect if you talk about in vivo gene therapy using intravenous injection. As I will outline later, most of the injected vehicle is actually lost due to unspecific sequestration. The vector usually remains episomal. It is extremely efficient in transferring its genome into quiescent cells. So in the bone marrow, HSCs are surrounded by extracellular matrix proteins, laminin, collagen, produced by stromal cells, and they are not really accessible to intravenously injected ligands like our HD-ED vector. So we decided to use HSC mobilization using drugs, GCSF and AMD 3100 plerixafor, to bring them into the periphery. So these drugs are used to harvest HSCs. In our case, we leave the HSC in the periphery, and we are trying to transduce them. So in mice, the peak of mobilization would be around 40 minutes after AMD 3100. In monkeys, it's somewhat later, it's around 8 hours. So what was somewhat surprising was that a large fraction of HSCs transduced in the periphery returns actually to the bone marrow and spleen, and this is shown in this simple study here, where we inject a GFP vector 40 minutes again after AMD 3100. So at 2 hours after injection, we see about 15% of LSK cells transduced in the periphery. And these cells then by day 3 disappear and they reappear in the bone marrow and spleen. The spleen is a secondary hematopoietic organ, but in terms of size, in terms of total number of HSCs, it's smaller than the bone marrow. Because we have a higher receptor density on LSK cells, we see more transduction of primitive cells. And again, I want to point out that without mobilization, we basically transduction of HSCs in the bone marrow analyzed at day 3 is not efficient. This is a situation in the monkey. Again, we inject the same vector at a peak of mobilization. At days 3 and 7, we see about 6% of, again, primitive CD34 plus, CD90 plus cells expressing GFP. So viruses, and specifically adenoviruses, have not evolved to be injected intravenously at high doses. So they trigger innate responses. Since the tragic death in 1999, arguably adenoviruses have been the best studied vectors in terms of triggering pathways that lead to innate responses. And two of the key players are IL-1 and IL-6. So we decided to use drugs that have been used before for rheumatoid arthritis or also in the CAR T-cell field to block these two cytokines. And the effect was quite dramatic. And here are some examples from monkeys, which we injected at 2 different doses here with HD-AD vectors, and we are measuring serum IL-6 levels, which is an indicator of innate toxicity. Without anakinra, we still see significant cytokine release, especially after high-dose injection, but if you combine both IL-6 and IL-1 blockers, we basically blunt cytokine releases. So as I mentioned, adenovirus vectors are episomal. To stimulate integration, we use a Sleeping Beauty transposase system, which consists of 2 vectors and results in a random integration of a transposon that is delivered again by this vector here. In vivo, co-infection of HSCs with 2 vectors is inefficient, so this is one of the reasons why we have incorporated an in vivo selection system, which is based on MGMT mutant, which confers resistance to methylating drugs like O6PG, BCNU. And this has dramatic effects, which is shown here in mice, again with a GFP vector. So 3 doses, 3 injections of low-dose O6PG BCNU results in GFP marking and these, in PBMCs, and these PBMCs are derived from HSCs in the range of about 100%. And we see marking in all lineages, including LSK cells. And also we see GFP expression in, for example, in tissue macrophages and F480 macrophages in the brain, intestine, and liver. So we have used this approach. We have shown the safety and the efficacy of this approach in a number of models for different mouse models of different diseases, hemoglobinopathies I will talk about later, hemophilia, infectious diseases, cancer. And we have shown the safety and decent efficacy of the approach in more than a dozen rhesus monkeys. So my first example for therapeutic application of the approach is a gene addition approach. again using the Sleeping Beauty system to integrate or to add a gene. In our case, this cassette is pretty large. It contains a beta-globin LCR and a human gamma globin gene. And we are comparing it here with what you call, I think, a micro LCR, again driving a beta-globin gene. So we do our in vivo injection, mobilization, IV injection of both vectors, and then 3 or 4 rounds of O6BG BCNU selection. This shows the percentage of human gamma globin positive RBCs in the periphery. Red is the long LCR, black is short LCR. These are the levels of gamma globin measured by HPLC, human gamma globin over mouse alpha. So we are clearly the long LCR gives you higher levels. They are in the range of about 20%. And this leads to phenotypic correction. So there is no reticulocytosis, no hemosiderosis shown here in the spleen, no extramedullary hematopoiesis, which results in normal spleen sizes. As I mentioned earlier, integration is random. It's truly random, which is slightly different from AAV and lenti vectors, potentially more even safer. And we also have shown that this vector system works in monkeys, so we see gamma globin marking in this monkey in the range of about 90% after O6BG BCNU selection. Yeah, now at some point we decided to get away from integrating vectors, even if they are, well, theoretically safe, and focus on in vivo genome editing. And my first example is an approach that uses an adenine base editor, ABE8E, which is a very active base editor that can convert, that can actually destroy a BCL11A binding site and reactivate gamma globin. You have heard about this story before. I don't need to go much into it. So what we have now is a single non-integrating HD-add vector to reactivate gamma globin. This is the single vector. The vector remains episomal. It is lost over time, even in slowly dividing LSK cells. These are some efficacy data. Again, we mobilize the mice. We inject 1 vector IV. We do 3 rounds at earlier time points of O6BG BCNU selection. And then we measure different parameters, including editing at the target site, which reaches about 70% and stays stable in secondary recipients. This results in reactivation of gamma globin. Oh, I forgot to mention that this study was done in beta-yak mice, which contain a large piece of the beta-globin locus and which are an adequate model for globin switching. So we see a switching and activation of human gamma globin in these mice, so about 80% or 65% of red blood cells are human gamma globin positive. The level of human gamma versus beta gamma is about 20%. And as you have heard earlier, base editors do not trigger or not efficiently trigger double-strand breaks. So the level of indels is below 1%. And using 2 approaches to predict potential off-target editing, we were not able to detect off-targeting. So this was a study that was done by Kyriaki. She just showed the data, and as you have seen, she has also data in humanized mice. Another example of in vivo editing is shown here. Here we use a prime editor, PA5 Max, provided by David Liu, which is able to repair the sickle mutation, basically the root cause of sickle cell disease. This prime editor contains 4 elements, and they fit all into 1 vector. We are using here a model for sickle cell disease, Townes mice, again mobilization, single vector injection, 2 rounds of O6BG BCNU selection, and we see about 50% of editing, and editing here means actually repairing the sickle mutation. And there's a mixture, if you look at single cell derived colonies, if you look at single cell derived colonies, there is a mixture of monoallelic and biallelic editing. Again, you would need only monoallelic editing to treat a disease. We use 3 different methods to measure beta-globin in erythrocytes at week 16. All 3 methods showed that about 60% of HBS was converted into HbA, and this led to a phenotypic correction, as shown here in the sickling assay. There's no leukocytosis, normalization of spleen size. We spent quite a lot of time and money to look for off-target effects, which has become a major concern in the field of genome editing. And again, using 2 different prediction approaches, we did not see off-target editing. So a third example is a Makassar base editor. You also have heard about this earlier. Why we like this base editor more than the prime editor is that it is more efficient, has a higher editing efficacy in primary CD34 plus cells compared to the prime editor, and it does not need a dominant negative MLH1 protein, which transiently suppresses DNA repair. Again, this study was done by Chang Li and will be published soon. Again, we have a single vector here. We see a nice correction and also phenotypic amelioration. So what I want to point out here with all 3 methods, human gamma globin gene addition, gamma globin reactivation, and correction of sickle cell mutation, we are all more or less in the range that would theoretically be therapeutic in humans, based on current ongoing clinical trials with lentivirus vectors and ex vivo approaches. So I want to spend a few words about optimizations. GCSF mobilization would be contraindicated in sickle cell disease patients. So we collaborated with John DiPersio, who developed a small molecule and VLA4 inhibitor called W106. And we combined it with AMD 3100 and injected 2 hours later our PA5 max vector into these Townes mice. So we see efficient mobilization, as efficient as with GCSF. We see less leukocytosis, no cytokine release, and efficient editing. Another red flag for clinical translation would be the use of O6BG BCNU. So we are trying different approaches that are based on epitope editing. So we would use base editors here to destroy an epitope of a receptor that is present on primitive cells, in our case here on common myeloid erythroid progenitors. And then we would use an antibody drug conjugate to kill cells that have not been edited. In our case, we have a second guide RNA that would actually confer a therapeutic edit and reactivate gamma globin. So the ADC here is Mylotalk. There are other approaches being developed that are based on CD117 and CD45 by our collaborators. These are some data that we have with Mylotalk-based selection of edited normal CD34 plus cells. So if you differentiate them into myeloid cells, you see enrichment of edited cells over time. After erythroid differentiation, you see higher levels of, you see also higher levels of editing and higher levels of gamma globin. The last optimization I want to talk about is an issue we have seen in monkeys, but not as much in mice. The observation was that cells transduced in the periphery, HSCs transduced in the periphery, largely return to the spleen and not to the bone marrow. So we decided to transiently overexpress CXCR4 from a promoter that would be silenced after a week or two. So shortly after we do our in vivo transduction, these transduced cells would express CXCR4, which should increase rehoming to the bone marrow, and we have tested this in monkeys. And as you see here, these are the percentages of GFP positive CD34, CD90 cells, and these numbers basically double after CXCR4, using the CXCR4 approach. So we have, again, as I mentioned, we have greatly improved the vector platform. So we are using now so-called HDL6-3 vectors. They're very easy to produce at very high yields. Theoretically, they are less neutralized by pre-existing antibodies. They seem to be safer, less cytokine induction, less thrombocytopenia. And in combination with CXCR4 overexpression, we are reaching about 0.35 vector copies per cell in the bone marrow, which is a good starting point. I mean, before in vivo selection, which is a good starting point for further editing approaches. So in my last slide, I would to indicate some barriers, and some of them have been mentioned to date already. Clearly, one issue is pre-existing immunity against virus, but also against LNPs, especially in people who got vaccinated for COVID. A major issue is unspecific sequestration. And this starts with soluble blood factors, complement coagulation factors, blood cells, and then macrophages in the lungs, spleen, and liver, and other tissues. And this removes the vast majority of vectors that were injected intravenously, and also activates innate immune responses. In the bone marrow, I mean, the virus has to get into the bone marrow, and it has to diffuse in the bone marrow. However, as I mentioned, there are physical barriers, so we try to solve this by mobilization. Optimally, the virus should be targeted to HSCs. We are trying to work here on this and trying to combine different two receptors to transduce HSCs. What viruses are really good at is, again, transporting their payload into the nucleus. But overall, I believe the future of in vivo gene therapy will be a rationally designed nanoparticle. But until they are ready, our strategy is to rely on products of natural evolution and improve them. So I would like to thank the people in my lab, especially Chang Li, who did most of the editing work. We are collaborating with Hans-Peter Kiem and Dr. Yannaki's group in Thessaloniki. I also would like to thank Thalia Papayonopoulou, who has helped us very much in the beginning, and our sponsors. Thank you.
Thank you, Dr. Lieber, for this exciting talk. Please address your questions to the microphone. I can start. I have a couple of questions. So, what is your belief or envision what will be the best approach for this in vivo strategy. I mean, which vector, which in vivo selection system, which mobilization, what it would be the optimal, you think?
Well, I think we are trying to pull different things together here. So let's start with the vector. I think we have a vector platform that is promising. We are focusing on GCSF-free mobilization. In terms of payload, we really like this ABE8 base editor for its safety, for its efficacy to reactivate gamma globin. In terms of expansion and in vivo selection, we are working hard on finding alternative approaches. I mean, I mentioned CD33 myelotalk-based approach. We also have good data for an expansion approach that relies on a truncated HMGA2 protein, which works without any drugs. Again, we are trying to pull these things together in a monkey study. Hopefully, by the end of the year, next year, we will have some data for this, and which could be the basis for clinical application.
Do you think that the regulatory authorities would have any concern about the IV delivering other viral vectors?
This will be an uphill battle clearly. I mean, this would be new to the FDA, and it clearly would need some effort. And I see this as a problem.
Any other question, please, to Dr. Lieber? If not, Dr. Lieber, the society wants to give you a small gift. Thank you very much.
It is now my great honor and pleasure to introduce to you Professor Hildegard Bühning from Hanover, Germany. Hildegard Bühning is a professor for infection biology and gene transfer, and also deputy head of the Institute for Experimental Hematology at a medical university, Hanover. She also served as president of the German Gene Therapy Society, but also until recently as the president of the European Society for Gene and Cell Therapy. Hildegard Bühning actually contributed seminal work on AAV biology, how AAVs infect our cells and traffic. Also developed vectorology tools and platforms for improving AAV vectors based on directed evolution, but also rational-based design. So we are very happy to listen to your presentation with the title Tailoring Adeno-Associated Viral Vectors for In Vivo Gene Therapy. Please, Hildegard.
Thank you very much, Delius, and I would like to thank the organizers for the invitation. It has been a wonderful meeting so far, and also today it was just fantastic. So I'm very pleased that you stay. I know it's the last talk, but I hope I can hijack you and fascinate you towards a little virus, the adeno-associated virus. It's, as I said, a little virus. It's belonging to the parvovirus family, and so far there's no disease known for AAV. Nature is so kind to has provided us with a lot of serotypes to play with, which is very good if you are working in gene therapy and would like to tailor them as vectors. All of them do have a broad tropism, but they have certain preferences, and this, of course, you can use then when it comes to the vectors. So in brief, what you see with AAV is it's a non-envelope protein capsid, basically this nice drawing here, and it delivers a single-stranded DNA. If we have a look at the DNA, then we see that we do have here specific structures called inverted terminal repeats. They are serving as packaging signals, origin of replication, and so on and so forth. We have the information here on multifunctional non-structural proteins, which serve for the replication and transcriptional control. And we have here the information on how to build the capsid and actually the information for the capsid proteins. And we will hear a lot about this gene region later. And basically, if you now would like to hijack the whole story, then you would like to get rid of the all viral open reading frames, the rep and cap, and replace it by a transgene expression cassette and package this into one of the serotype capsid. And that's basically the first generation of AAV vectors, which are used in all the market-approved AAV gene therapies on monogenetic diseases. So, work done. So can we go to dinner? I think not yet, because I have still some minutes to convince you that we can do still better than nature. And I think to do so, we really have to look at both sides of the coin. So we first also have to look onto the cell and see where can we use which AAV serotypes best, and also see there, when there are barriers, to learn how we can then engineer the vector to make it better. And I would like to share with you one I'm too quick, because I first would like to tell you that we have to be better. Sorry. So before I come to this slide, and I would like to remind you that we are roughly having 3.7 times 10 to the 13 cells. We are not counting the bacteria in our gut. But if we now use the naturally occurring AAV serotypes for in vivo gene therapy, and we really stick now to their main targets, which is in most of the cases the liver, and we think about therapeutic approaches for the liver, then this is the doses which we apply. And now just calculate that these are human beings which are adult. And this is the best case. And if we now would like to use AAV, such as in spinal muscular atrophy, where we would like to convince AAV9 to go into the brain and transduce motor neurons, well, it prefers to go to the liver, then we really end up with a very high dose. And then, of course, our immune system with this is not happy, and we will get immune responses. So now, come back. How can we modify this? And as I said a minute ago, because I forgot about these 3 slides, is we have really also to look into the cell. And this is the example I would like to share with you now. So Nadja Moiman, a very talented postdoc in my lab, wanted to know if we can use AAV vectors for treating cancer, hepatocellular carcinoma, and she went for this into a transgenic mouse model. So she injected AAV2 vectors delivering a transgene, so GFP, and then she had a look into the healthy part of the liver. And I think you agree with me that this is not really a wonderful transduction, because here you see this one is a transduced hepatocyte. Not really much, right? But then she was very surprised when she looked into the tumor part. This is a transduction you can work with, and this as well. So the question was, because we saw this not only when we went in the MYC-driven model, but also in this case in a RAS-driven model, and here you also see the difference. This is where the tumors are. That really somehow there seems to be a preference of AAV transduction to the tumor tissue. And the first thing you are thinking about if you inject these vectors intravenously, it's maybe more vectors are found in the tumors. So what we did is we looked into the HCC part as well as in the healthy part, or here in this case in the tumor-bearing mice versus the non-tumor-bearing mice. And we basically saw that there is not a preference of entering into the cell. So we did our homework, did a lot, and ended up in the nucleus. And AV is so tiny little virus that AV with its genome can enter the nucleus. And the uncoating, so the release of the genome, takes place in the nucleus. And then, when it is released, then it forms these episomes, which are the stable forms from which transgene expression occurs. So the question was, if we do our PCR analysis, we just look to the whole sum of all the AAV genomes, and there we didn't see a difference. But what about if there is a difference in this area, so in the uncoating? And for this we used an enzymatic trick, so we isolated the DNA and then digested it with an exonuclease, where we get rid of everything which is not in the stable episomal format. And when we came back to our mice, and we saw that indeed, in the constellation where we do have the tumors, we see that the uncoating is taking place much more efficient. So we learn here that indeed, depending on the condition, the microenvironment in a cell, AV does better or not. And this is not only a mouse issue, you can also see this here in this patient sample. But now let us go back to what we can do to the virus. So you may know that we like to engineer the viral capsid. And this you can do by rational design. It's not only our group, a lot of groups like to do this. And basically what you can do is, these are the surface most exposed positions, these are the regions where AAV interact with receptors on the cell surface. So what you can do here is, you put here receptor binding ligands with then mediating the transduction of the cell. You can also engineer there nanobodies, DARPins, adapters, or whatever. You can do it genetically or even non-genetically. And that this really makes a change, I would like to discuss with you on an example on inner ear disease. So you listen to all the talks today, and to be really able to get what you hear into your brain, we have to use a relay system, right? So what happens is we have the sounds which enter the ear through the outer ear, where it vibrates to the eardrum. This is relayed then to the middle ear and to the cochlea. And there you have the organ of Corti, where we have the hair cells, which then transform this vibration via something which they sense as a fluid movement into a signal, which then can be transmitted to the brain. And you can imagine that a lot of things can happen here, which then causes that we are not able to hear. So Jenny of my group, together with Hinrich Stecker, our collaboration partner, wanted to think if we can use AAV and develop AAV in this regard, because it's also a very tiny little organ where you can do gene therapy. And the experimental setting is here that we use a mouse, and that the vectors, which are also very little doses, as you can see here, are applied locally into the posterior semicircular canal, and that then after 7 days look which cells in the organ of Corti have been transduced. When you use a natural occurring AAV serotype and look into histology, then it looks a little bit dark under the microscope, because we transmitted GFP as a transgene, and you are with me that this transduction efficiency is not really high. So luckily, Hinrich had performed an FH display library selection approach, and we're coming up with some peptides. And so, rational design based, we inserted the peptide at this highest peak, second highest peak of the AAV capsid, and tried if this one then can help mediating transduction. And indeed, you see now that indeed, if you have the right key inserted on the surface of the capsid, you can open the door to the cells. And what you see here is that indeed here the spiral ganglion cells and also the hair cells were transduced. So GFP is a GFP. Can we do more than that? And you know that there are hearing aids around, so cochlear implants, but how well they're performed is a little bit unpredictable. And what is believed is that it is unpredictable how well they have performed, because we are losing somehow also the spiral ganglion cells, because they are non-activated. And the idea would be to provide neurotrophins. As a first step in that direction, and a simple test for our mutants, we wanted to apply these in the context of ontotoxicity. So how does this work? So you have your poor little mouse and you apply ontotoxic drugs, and then a week later you provide our AV mutant with the neurotrophin, and then you look what happens. And so we have the group, the control group, then 2 conditions of ontotoxicity, even this one, the more severe one, and then what happens if we try to rescue this with AV. And if you just look at the images, or here more closer in the investigation results, then you indeed see that by this simple approach, you can indeed rescue the neurons. Besides using a rational design, you can also look what libraries, what evolution is doing, and learn from this, and therefore you can generate libraries. You remember that I told you that in the CAP open reading frame of the AV viruses, everything is written what is required to generate a capsid. So what is done is that you introduce mutations just randomly, and then by your selection strategy identify those which are good for your approach. You can use just error-prone PCR approaches via the capsid gene. You can also use the different capsids of the different serotypes, chop them apart and ligate them, so shuffle them around. Or you can decide for one capsid and introduce into these surface exposed positions peptides of a random sequence, and have by this, of all of these approaches, you generate a very huge library where this virus here differs from that virus just by what happened to this virus. And with this pool, which is commonly greater than 1 million, you do your selections. Ex vivo or in vivo, you should first think about what the best features would be of your virus, later on of your variant, and you do some rounds of selection, always looking for the best in the race. Coming back to the liver, Nadia performed exactly that. So we were ending up with an AAV capsid peptide display library where we had the random peptide inserted here at this peak. These libraries were tailbane injected into the mouse, and then we ended up with these highly selected variants because we wanted to go into hepatocytes. Nadia produced them as vectors encoding for luciferase, so these 2 mutants, and compared this to the parental AAV serotype 2. And you nicely see, I think, that the efficiency in hepatocyte transduction was greatly increased by this approach. To get this high expression, we needed less vector genomes, and this is shown here. See, this is a log scale. And what is done here is you isolate from your target organ as well as of off-target organs the DNA and test for the amount of vector genomes delivered to this. So the mutants required less vector genomes in the liver to give this bright signal, and this was impressively less than what you needed for the AV2 or the gold standard, which is AV8. What is also very nice is here that you see less vector genomes in spleen, liver, spleen, lung, or heart. Okay, you don't want to cure a mouse, so what about human cells? Nadia first tested her vectors then in human, primary human hepatocytes, again luciferase, and by the signals it's easy and you compare just the bars. They work in human primary hepatocytes and outperform what was out there. The ultimate goal is, of course, to check this in vivo, and so we teamed up with Leszek Lidowski. And this is a humanized mouse, humanized liver, and it was a side-by-side comparison between the variants, so LIV-K and LIV-A, as well as AV8, which is a gold standard for liver transduction in a mouse, and AV2, the parental serotype. They were packaged with identical vector genomes. You can only distinguish the 4 different vectors by a little barcode, which is found in the genome. You mix them and inject them in the same mouse, so really a direct comparison. And after that, Leszek and his team isolated the human and the murine hepatocyte and looked for DNA and for RNA, so for expression and for DNA distribution. And again here, when you just look at AV2, then you see that really entry, so being in the hepatocytes, is not an issue for AV2, but really expression is not good. And this is the same results that we saw in the HSC models. However, if we now have a look at the two variants which Nadia has developed, then you see that we have a very impressive expression in the human and the murine hepatocytes, showing that they are really suitable to really do a preclinical development all the way through the clinic. And this maybe is because it's nicer to see it as an image. I think you nicely can appreciate that they really do the job also in the human hepatocytes. We heard a lot about CD34 cells, and so I would like to show you the first steps we are doing in this direction together with Hans-Peter Kiem and his team. And so we selected our library in NHPs and ended up with a set of variants, and we first tested them on human CD34 positive cells. And these are, for AAV, these are very low particles per cell numbers, and we really checked for the long-term progenitor cells and expression, and also in the multipotent progenitors and the lymphoid progenitors. And you see these are pretty good results for AAV, and we didn't see any toxicity, which you sometimes see with AAV6. But what about, do they do their job in vivo? So we first did a humanized mouse model, shrinking them, the 30 variants down to this set, and compared this set then to the parental and the gold standard, in this case AAV6. And we looked for expression and biodistribution. And this is the DNA data, so the CD34 positive cells, white blood liver, this is DNA, and this is the exo. And you nicely see that we have two variants which seem to do the job quite well in the humanized mouse. This is just a bit more of investigations. So we took them now back to the rhesus macaque. Again, we used the two best vectors here in comparison to the parental and the gold standard. Did expression as well as further analysis. It's still ongoing, but as you can see, we see indeed expression in the long-term progenitor cells. And at this stage, because we also did here a side-by-side comparison of the four vectors, only distinguishing them by the barcode, we couldn't say who was doing the job here. So we had to wait for the analysis of the barcodes. And this is what we are at the moment at. So what you can nicely see is that so this is on day 3, so while the animal was still alive, on day 7 they were sacrificed. And what you see is that indeed our variants perform quite well. We in particular are very proud on this result, what you see here for the long-term progenitor cells. And this is in particular important for us because we see nearly no expression in the liver, but really a change towards reaching these cells. So we have 6 AAV gene addition therapies which have in total obtained market approval. We have a lot of things to do. I showed you that by capsid engineering you can improve the transduction efficiency, modulate the tropism, but you can even do more. And this is the sole data slide I would like now, or set I would like to share with you before closing. And this is that you can use capsid engineering for another purpose, and that is vaccine development. So you have the protein capsid, and you can imagine that you could put your antigen on the surface and use then the coding information, the vector genome, for also overexpressing your antigen later on from the transduced cell. So when you inject this vaccine, which we call antigen-decorated AAV vector-based vaccines, the immune system can already learn and can get primed to the antigen, which is on the scaffold of a virus. And then later on, as I said, the transgene will be expressed from the muscle and is available for the boost. To test this, we used ovalbumin, a well-described antigen, and we teamed up with Sahil Adrush from Rouen and Ulrich Hacker from Leipzig. This study was performed by Anne-Christine in my lab. So what did we do? So we had an AAV2 just encoding for GFP, we had an AAV2 just encoding for the antigen, and we had our prime boost vector in 2 different doses, having the antigen on the surface and encoding for the antigen. It was injected in the mouse, and then we challenged the mouse with tumors expressing the antigen. So first of all, on the fly, we looked if we had an antigen-specific immune response in the blood, and indeed, look always to the blue and the red, because these are the prime boosts. You see, it worked quite nicely. Not only the T cells, also we got antibodies which were antigen-specific. And when we had a look at harvest, what about the function of the T cells, you did see these antigen-specific T cells indeed do their job. However, what about the tumor? So this is tumor growth when we just use AAV encoding for GFP. This is when we use an AAV which is encoding for the antigen, so nothing on the surface. You see that in the poor little mouse, the tumors are growing. However, when we have the prime boost vaccine, nada, no tumor growth. Okay, you can say it's 15 days after vaccination. What about challenging the system? So we moved forward, same approach, but then challenged. So the tumor injection was done at day 80 post-vaccination, and we used two different tumor models, one of which, the B16F10, is quite aggressive. And this is the result here. And I think these are the prime boost situation, and I think it's quite impressive what you can do with these new approaches. So let us sum up. So you can indeed also, with the capsid engineering, move beyond gene addition therapy. And I think with all these tailoring, you indeed end up with an AAV vector which you can use as a biological nanoparticle for gene therapy, cell therapy, vaccine development, and of course, basic research. And the last two slides are the most important slides because I'm only here to report on what a wonderful team back in Hanover did. And the work I could report today on was done by Nadia, by Jennifer, and Anne-Christine in my group. And here you see them, so Jennifer, Anne-Christine, and Nadia. And I'm grateful to our funding agencies, and a huge thing is going to my collaboration partners, for which I can stand here to report on the results. So the CD34 targeting approach is done together with Hans-Peter Kiem's group. Big thanks to Stefan, Keith, Daniel, and Anne-Sophie, as well as Christopher. And in a year, I already I told about Hinrich, here the cancer immunotherapy with Sahil. And by this, I really would like to thank you, and I'm looking forward to our discussions.
Thank you very much, Hildegard, for this excellent presentation and also for sharing this fresh data with us. So, other questions from the audience? So, let me start by asking something about your prime boost approach. So, what do you think is the reason why the combination, so your decorated capsid plus the encoded antigen, works better than the AAV2 wild-type capsid with the same antigenic sequence in the genome?
So the reason is that when you present the antigen really on the surface, you can already start with the immune response and really get it into the right direction. And the nice thing is really also this timing between priming the immune system and boosting the immune system, because this is what we saw in prior investigation. It's really that you get a mature immune response. So it's really this unique composition, and then it could be that in particular these particles are really able to really transduce also dendritic cells, which you cannot do with wild-type capsids.
Thank you very much. It was a fantastic talk. I really enjoyed it. Regarding the last part, I was a little bit curious. Two questions. The first one, if you have tried to compare your decorated AAV with an AAV expressing the transgene or whatever the antigen. And also I was curious, your AAV was decorated, if I understood correctly, but forgive me, because your AAV was decorated with OVA, right? And then your B16F10 model was also the F10 expressing OVA, or otherwise, how did
Yeah. Sorry, maybe I was not clear. Of course, the tumor cells were expressing the antigen. Okay. Otherwise, it's not possible, right? Okay. And maybe I have also to add, Stelios, to your question that, of course, if you let the system longer run and if you have sufficient antigen expression by AAV to normal capsid really boosted as much as possible, then you get, of course, also an anti-tumor cell response. But here it's really tiny little, as you would do in a vaccine.
Very nice. And have you characterized the memory T cell, the quality of the memory T cell that you are generating? And also, I saw the paper was accepted. Where was it accepted? Is it already online? Or?
It's online, yeah. It's online. So they are not exhausted. Okay. And if this is your question, maybe.
Okay.
Thank you for the fantastic talk. They are really great data. So I was wondering, what kind of antigens can we use to load the AAV with? Can we use, ovalbumin is a really immunogenic antigen. Yes. Can we use antigens which are specific for tumor, but they are not so TSA tumor-associated antigens, which are less immunogenic? And given that AAV are also presenting their immunogenic antigens, is there going to be a competition, an antigenic competition, so that the immune system will result on having T cells against the AAV and not against the not so good antigens?
Thank you for this question, because this is a discussion Vincenzo and I do have in the coffee breaks. If it is better to start with an adenovirus where you have somehow a road which you follow because the immune system is recognizing this very well and starting a very strong innate response, or starting with a mild immunogenic one like AAV. And our strategy is really to use the toll-like receptor 2 and 9 recognition, which happens to AAV2, but which is compared to other vectors quite low. And really build on that to drive it into the right direction. So with regard to the antigen, the next step for us is, of course, to go to real antigens to get a challenge. And what is really nice is that we, as I said, we can add more features to this particle to drive it even in the one or the other direction if it is necessary. Yeah.
May I ask a question? First, just of curiosity, which will be the first disease that you will target through hematopoietic stem cells? And if this targeting is specific, or may you get spread to other tissues?
So this is really fresh data. So we have just received the tissue prior to the ASGCT meeting. So this is a tissue which we have analyzed so far. So we looked into spleen, liver, into what we received from the bone marrow and from the peripheral blood. And what you see in this table, this was the investigation on expression. And there it was really very impressive to see that variant 24 was nearly off in the liver, which is very good for an AAV, and really went to and expressed, it was expression data in the hematopoietic stem cells, in the long-term stem cells. What is with the other tissues, I cannot tell you because it's ongoing. So we are in the NGS analysis at the moment. So we received from Seattle, or we are currently receiving, I have to say, all the other tissues in a parcel from Seattle.
Okay, are there any other questions? Okay, then I would like to thank again all the speakers, both speakers. And before we close this session, Hildegard, please stay on the stage. The Hellenic Society has also a small gift for you to honor your contributions. Thank you.
We now close the session and this long day. I would ask the members of the society to stay. The General Assembly will follow, and we need to discuss several things as the board of directors will be changed for the next 2 years. So please, the members need to stay.